Otherwise, if not, I think we can go to the first introduction, which is Eric and Jeannie. So Eric, please go ahead.
Thank you, Terry. Welcome, everyone. It's great to see you. And for those of us who are joining us remotely, thanks for joining. And I just want to start off front by expressing my appreciation for the folks who organized this meeting. And my appreciation for NHR's communication group, some of which are sitting behind me, who are making sure that we have good AV. Zoom is going to get the word out. Also, they're recording this, so we will make this entire proceedings publicly available. I'm going to just make a few brief context-setting remarks and then turn it over to my colleague sitting to my left. The origin of this entire series of meetings dates back to 2011, actually, when the Institute put out one of its series of strategic visions. It was the second one it put out since the completion of the Human Genome Project. And in the 2011 strategic vision, we specifically called out the desire for the Institute to move more aggressively towards the applications of genomics in medicine. And at the time we actually wrote it, Jeff Ginsburg, who's sitting to my left, was the one that planted the seed of idea to Terry, Manolio, and I that maybe we should set up. Of course, it sounded great. Yeah, we want to pursue genomic medicine. And we even wrote a really nice article that was published in Nature, aided by our council and many people in the community, including people sitting around this table. But Jeff wisely pointed out, we need a group that's going to really tackle this and move ahead and really give advice to the Institute if possible. And that seemed perfect for a working group of our advisory council. And shortly thereafter, we created the Genomic Medicine Working Group, which has been an amazingly productive and effective group that I have relied on extensively over the years, various members of which are sitting around this table. And among the various things they've done was to help organize or conceive of and participate in this genomic medicine series. Now, as you read in Roman numerals, number 16. And Terry, in a little bit, is going to sort of go through a little bit about those meetings, the history, to further set a context about it. The working group, which meets pretty much monthly, I join quite often when I can. They do a variety of things and have been incredibly helpful on other fronts. I would just put in a plug that one of the things they've been doing recently is to once a year write a short article highlighting I think it's about 10 highlights in genomic medicine in terms of papers that were published in genomic medicine implementation. It's called Year in Review. The American Journal of Human Genetics published it, and it just came out like a week ago or something like that. So that's a shameless plug, but it does illustrate the effectiveness of this working group. So we are always brainstorming for ideas of what should we do for the next genomic medicine meeting. And this idea bubbled up, the idea of the very interesting aspects of genomics associated with infectious diseases. Now, the National Human Genome Research Institute has tended to stay away, appropriately so, from the infectious agent side of it. We tend to stay more on the human genome side. That's in our name, that's in our mission. And we have an outstanding other institute at the NIH that has ably handled research, including genomics research of infectious agents. But of course, there's interactions that go on between the host and the infectious agent. And we've always wondered there'd be great opportunities to explore it. And when Jeannie Marazzo arrived on the scene as the new director of the National Institute of Allergy and Infectious Diseases, besides personally hitting it off and really getting to know each other quite well and enjoying each other's strategic and ideas working together, but also a friendship, it occurred to us that, wow, maybe we should really explore ways for our two institutes to interact around a topic such as this. I think it was about the same time the working group was thinking this would be a fertile area, and it didn't take very long to just say, yeah, let's just do it. And so that's the history that has brought us here to this meeting. It is wonderful, I will just say, to doing one of these meetings with another NIH institute. And it's been delightful to organize this and to have Jeannie and her colleagues be able to bring a new people into that maybe we don't know. I mean, it's part of what this is, a little bit of a mixing of communities. And we have no preconceived ideas what'll come out of it. We never do when we start one of these genomic medicine meetings. However, when you hear Terry's presentation, you will see there's a very strong track record of very productive things coming out of these meetings. So with that, I'm going to get out of the way. I'm going to turn this over to my colleague and good friend, Jeannie Marazzo, and she's going to give us a bit of an overview from the perspective of the work going on at her institute.
Thanks, Eric. And if I could have the slides, that would be great. I'll just start by welcoming everybody and thanking the team who put this together, particularly Terry and Lucas, who have been, from my institute side, who've been very, very working really hard to assemble, I think, what's a fantastic meeting. There are many wonderful things about coming to the NIH, some of them being very evident from this meeting. One is meeting people like Eric, whose unbounded enthusiasm is contagious and really energizing, and also learning more about genomics from colleagues like mine, Steve Holland, for example, who has been doing this work at the clinical center for a long time. So it's been a fantastic journey, and I look forward to seeing what we're going to accomplish in the next couple of days. There's a lot of time for discussion, you may note. So I think this will probably not be a shy group, and we'll have a lot to talk about. So I just have a few slides to point out some things that are probably pretty obvious, but since there has not been one of these meetings devoted to infectious disease, I'll just kind of set the stage here. You know, infectious diseases have had an intimate dance with our immune system and our genome for millennia, right? And we know that genetic variation can lead to resistant or susceptible phenotypes to various pathogens. So understanding how these infectious diseases and other environmental selective pressures have impacted human evolution really is important into understanding the pathogenesis of infectious diseases and also potential treatments which might relate to some of those genetic adaptations, as we have sort of dreamed about with precision medicine. I don't think we're quite there yet, but it is, I think, a goal. So, oh, sorry, hearing myself in stereo, which is always a little freaky. So I think what this meeting is really going to get into is this exciting intersection of genomic medicine and resistance and susceptibility to infection. And I should just point on the top, there's some very nice kind of examples of diseases that clearly, from malaria to TB to HIV, have really been impacted quite a lot by some of these events over the course of human evolution. We know that some of the genes that have conferred resistance to some of the worst scourges in the infectious disease world actually come at a cost. They have unwelcome trade-offs. And some of the best examples, I think, are that some pathogen alleles that confer resistance to things like HIV, malaria, leprosy, sleeping sickness, and others may lead to other non-infectious diseases with a genetic component. So the adaptations to fight off these infections are not always free in terms of their genetic currency. So a couple of examples shown there. I think the most obvious one, and perhaps one of the most famous one, is that G6PD deficiency. We know that G6PD encodes an enzyme that helps prevent reactive oxygen species from accumulating to toxic levels. And when you have G6PD deficiency, you can get hemolysis with certain triggers, including certain drugs and foods such as fava beans and stress. So very, very close interaction there. The challenge with G6PD deficiency is, I think, best shown with the infection of malaria. So we see this deficiency in parts of the world where malaria is endemic or has been endemic. So Africa, Asia, Mediterranean, Europe. And there is a clear association between certain G6PD variants and protection against uncomplicated malaria. Again, a trade-off here that the host has to figure out if this is worth it in terms of benefit. Unfortunately, some of the antimalarials we use, like primaquine, can actually trigger hemolysis. So you really need to understand what's going on here to optimally treat infections that can be potentially deadly. The other area that I think is really worth mentioning here are inborn errors of immunity. And we know that these can have devastating outcomes, including susceptibility to infection, as well genomic medicine has great promise to help patients with these disorders. One of the things we would like to see come out of this meeting is a discussion of a development of clinical recommendations to treat infections in these patients, because you can't always use what you might use in a patient who does not have these disorders. A good example is Chapel disease. Some of you may have heard this. Eric kindly invited me to his council several months ago, and I talked about Chapel disease. It's a fantastic story of how interrogating the mechanism of presentations of some of these most devastating inborn errors can help you figure out how things work and how to deal with it. Chapel disease is characterized by complement mediated hemolysis and thrombosis and a very severe protein losing enteropathy. People also get recurrent respiratory infection and can get hypogammaglobulinemia. So all of those things can suggest Chapel disease, but none of them are particularly specific. So this can be something that is not necessarily noticed. So I will just point out that, as I said before, one of the very fun things about this job has been getting to learn the unique capacity and the unique resources that we have, particularly at the clinical center here, to evaluate and to treat some of these immune disorders that are related to genetic dysfunction or disorders. This was launched in 2017 to help address interrelated challenges in clinical care and in our division of intramural research, which, as I mentioned, is led by Dr. Holland, who's here. And some of the focus areas of this program are to provide clinical genetic diagnostics for Mendelian disorders and medically actionable secondary findings, many of which, of course, are infections. These investigators also undertake routine genomic analyses in people who come into the clinical center and are on our protocols in an IAID, but also offer that opportunity to patients who are admitted under other protocols from other institutes. And the idea here is to do whole genome sequencing to look for known and novel genetic defects associated with immune disease, including polygenic models of disease etiology and progression. Sometimes involves family members, often goes into sort of the familial patterns of where some of these fall. And this is a program that has contributed greatly to the evidence base on the processes and outcomes of the clinical genomics and genetic counseling services. So a whole program has really been built up around this. You're going to hear more about this during the meeting, but I just wanted to put it out there as I think a really nice model of how these programs can lead the way in defining this complex interaction. So I'm going to stop there and turn it over to Terry. Very brief introduction.
Super. Thank you so much. And I did forget one housekeeping item. So those of you who might like to get on the webinar, especially if you're myopic like me and sitting way at the back, it's Bethesdan B is what you want. And then you want TBH in capital letters. That's for the Bethesdan Hotel. Oh, there it is right there. Bring it back. Bring it back. TBH 8120, which is the address of the hotel, and then exclamation mark. So that should get you on. Super. And I'm just very quickly going to, okay, if you could go now to my slides, please. Jacob, that would be great. And just thanking our thanking the organizers of the meeting, of whom there are many. Several of our colleagues in the back are doing the recording and webcasting of this. And also, everyone who helped set up today. So, and then next, I can next. You've been seeing the objectives there on the agenda. I won't go over them, but really what we're focusing on here is implementation. What we're not going to be addressing is discovery research. So there are lots of big questions. One of the pre-reading has identified a whole ton of them in terms of what are, associated, what are the variants that are related to X, Y, and Z. We can identify those and sort of, put a pin in them. We're hoping to have another meeting with our NIAID colleagues that will address that specifically. So really focusing on implementation today. Many thanks, as Eric mentioned, to our genomic medicine working group members. These are some of the meetings that they have helped us organized. This is the first 12. They won't all fit on one slide any longer. But these are the other three. The last one was held about a year ago on population screening. And then this is obviously the 16th. From these meetings has come a whole host of initiatives that we've developed. From the first one was the ClinGen Consortium, the Clinical Genome Resource, which has been, I think, really instrumental in developing a standard framework for variant curation, and has then grown into the GenCC consortium or coalition, which is throughout the world. The second one led to our Ignite Implementing Genomics in Practice consortium, which began as small implementation projects, now is large clinical trials, including the GUARD US, which is a nephrology, a study of ApoL1 and its relationship to renal failure. I can't show them all, but the fourth one on training led to our Inter-Society Coordinating Committee on Practitioner Education in Genomics, which is very active in disseminating and developing training materials for clinicians. The fifth one, I think, or was it the sixth? The sixth led to the Global Genomic Medicine Consortium, and then the International 100,000 Cohorts Consortium, both of which are very active in gathering information from worldwide cohorts and implementing it in clinical care. In the eighth one, we had a notice. It doesn't really say what the notice is. Sorry about that. At least I can't read it. And then, and then let me just see. Yeah, that's right. The genomic medicine modules, and then the variant function and disease one as well. And the 10th, I think, ended up being the adopt. Thank you. Eric is like giving these to me. I think. So, adopt PGX is a pharmacogenomics trial. It's part of the Ignite consortium. The 11th was on implementation. It led to a whole meeting on employers' role in health and wellness and genomic screening. That is still sort of ongoing conversations. The 12th was on polygenic risk. The eMERGE network then moved into genomic risk assessment, and the PRIMED consortium is developing ways to assess polygenic risk in diverse populations. And let's see, the 10th one, sorry, that was the 12th one. The 13th, I just lost the 13th was on informatics, led to a notice on that. The 14th was on genomic learning health systems, and that led to both two programs that are in electronic consults as well as genomic learning health systems. Both of those are about to, well, actually, genomic learning health systems has started, and the e-consults is about to get underway. And then population screening was from the 15th one, and the applications for that have just been received. So the 16th one, I'll have to remake this slide, which I'm not looking forward to because it's really hard. But at any rate, we're looking forward to. It takes like 2 hours to do this. Well, you have to scrunch everything down. It's really a challenge. But at any rate, so really looking forward to this. And yeah, that's right. There are bigger problems than that. Just to let you know that we do post all of the slides and the videos and the meeting summaries from these meetings on our webpage. And you can see here, we've already got the agenda and that posted for Genomic Medicine 16. This is the webpage for 15, which shows you where the summaries are as well as the videos, et cetera. So with that, I know we're a teeny bit behind, but I will turn this over to Eric and Jeannie to moderate.
Oh, good. Thanks. Thank you so much.
I know we're just jumping into discussion, or we're going to jump into the speakers.
If people wanted discussion, we didn't leave time for discussion. So if you just want to go right into the groundwork.
Okay, so I think we should jump right into. We have a couple of keynote speakers that are going to sort of help set up. Jennifer, are you going to do it from the?
Yeah, we thought it might be easier to do it from your seat since it's such a long way to get to there. Yeah.
My only other idea that if people were speaking at the podium, if they start to go overtime, this feels like a bowling alley. And I just thought Terry and I, engineer, were going to land and inevitably we'd go in the gutter and it would take out the speaker. But maybe that's not a good idea. So we'll do it.
That was Eric's idea. I had nothing to do with that.
Yeah, you know, it's just so our first speaker is a colleague of mine going back to the early days of the intramural program at NHGRI when Jennifer Puck was one of the founding investigators that arrived a year actually before I did. So this is a bit of a bringing her back to Bethesda, but she's now on faculty at UCSF and has been an expert in this area of inherited disorders of immunity for decades. So Jennifer, take it from here.
Okay, so can people hear me? All right. Thank you so much. And I'm very excited about this, the topics today, and also just seeing sort of where things have come from. So I think they're echoes of all these previous meetings that are going to be useful here. So just to start off with a group that has taken on cataloging all of the single gene inherited disorders of immunity and publishing every couple of years or so. And this is I think from 2 years ago, the International Union of Immunologic Societies Committee on Human Inborn Errors of Immunity, which is a big mouthful. And actually, when they published this, they had tipped the graph 90 degrees, but I turned it back this way because this is the way I think it should look, to show you the exponential increase in the number of single gene disorders over time since the very first were discovered in sort of serendipitously in the late 60s, and then the genomic revolution thanks to the Human Genome Project, and of course lately a big increase because of everybody's learning how to use the genome to look for these rare disorders. So they are individually rare, but if you add them all up, it's actually a significant health problem, and I think we need to keep that in mind. So the way this IUIS committee tracks the diseases is that they try to put them in tables that capture certain specific immune disorders, like the number one immunodeficiencies affecting cellular and humoral systems. And I just put the link to the update from 2024, which has a whole lot more diseases that weren't there in 2 years ago. So they're well over, like, pushing 600 diseases now. And the trends are just continuing from these slides. So you can see that there are all different modes of inheritance, and they affect all different types of immunity. And it's sort of interesting to look at the graph on the right. The autosomal dominant conditions are coming up, coming into their own. Those are more difficult to find and define, and they're also in many cases quite variable in their presentation. And the geneticists know how dominant diseases have issues with expressivity and penetrance and so on. So the immunologists are learning that their own way. So when we think about the immune system, of course, there's the adaptive and the innate immune system. And Steve Holland and I love to argue about which is more important. Of course, the adaptive immune system is, I'll just tell you. As Robert Good showed, that this is where the specificity for particular molecular recognition lies, and also immunological memory. So this is the immunity of T cells and B cells. And the innate immune system limps along without that specificity and does its best to and there are defects in granulocytes and macrophages and cytokines and other things. And we're discovering a lot more of these. And they actually, when these components are not working properly, diseases do result. So yes, they are worth something. Okay, so, and I just want to point out that T and B cells are special because they're the only cells that rearrange DNA. So we all know how splicing of RNA occurs all the time in every cell, but T cells and B cells have a whole raft of alternate related sequence components that get spliced together sort of at random to and not only at random but also in a sloppy manner so that even more diversity is generated at the joining points and so this generates a huge amount of diversity, and each cell has its own specific sequences that are then encoding a surface receptor that can see an antigen or a molecule in the environment and react to it. So this is how the specificity and memory happen. And of course, it's some very mind this very special aspect of our wonderful adaptive immune system. Okay, and then just back to the list I showed before, it turns out that different components of this system are associated with protecting us or kinds of pathogenic uh, um, diseases. So for example, if you're missing T cells, well, I sort of think of them as the conductors of the orchestra of the immune system. And so without a conductor, there's chaos and all kinds of infections ensue. And in particular, we think about opportunistic infections that don't make healthy, normal people sick, but things like pneumocystis and unfortunately, vaccine strain live rotavirus vaccine, that's a lifesaver for infants around the world, keeping them free from secretory diarrhea. But if you don't have an adaptive immune system, this causes terrible diarrhea. So we have to sort of keep in mind all of these aspects of the immune system as we're going along. And then in the innate category, there are very fascinating diseases that result from defects in each of these components that I won't take time to talk about, but they're listed here. Okay, so when we're in the clinic, what makes us think of an immune deficiency? Well, first of all, infections. But not just any old infections, because all infants get infections. That's how you fill up that memory bank of your adaptive immune system by meeting and making memory responses. So, and actually the American Academy of Pediatrics has data that the typical baby has about 8 infections in the first year, and so I think anybody who's had kids or grandkids knows very well. And when you're in daycare, it's even more. So it's not just that they're frequent or recurrent, but they're persistent. They don't go away. They are not controlled with the standard course of amoxicillin for an ear infection. And so despite standard treatment, they are still there. They're severe, and they can be caused by opportunistic pathogens. And I think of the people who are affected developing sort of a toxic bouquet of pathogens that don't go away. And so you can have many different organisms at the same time. And of course, the result of this is failure to thrive. So one of the reasons for growth failure in children is immune deficiency. And then the flip side of immune deficiency is immune dysregulation. So if the immune system is not regulated properly, then we get autoimmunity and inflammation. And that is becoming better and better understood. And all of us know that we should take a family history. We learned that in our early genetics courses, but unfortunately, it's not informative as often as we would like, and fewer than 20% of cases actually far fewer than 20% are going to have a positive family history. And then we also have to look out for syndromic features because many syndromes are accompanied by different degrees of immune problems. Okay, so I'm going to talk just a little bit about severe combined immunodeficiency as an example of a single gene immune disorder category. So these are infants born with absent adaptive immunity, no or very few T lymphocytes. And even if they have B cells, the B cells don't work because B cells need T cell help. And so these infants typically would experience weight loss from age 2 to 4 months when their maternally transferred IgG wanes, and they get all kinds of infections, as I already mentioned, and this is fatal in the first year of life unless we can establish an effective immune system, which actually can be done with bone marrow transplant. And so the little guys here are at an immune deficiency foundation meeting, having all had transplants in infancy for severe combined immunodeficiency. So the defect is intrinsic to the hematopoietic progenitors of lymphocytes, and therefore transferring hematopoietic stem cells can alleviate or even cure the disease. The optimal donor is an HLA matched sibling, but very few people these days have an HLA matched sibling. Some patients are cured, others not completely. And we've learned that full function of the immune system requires making some space in the bone marrow for the normal stem cells to reside. And that's usually done with chemotherapy such as busulfan. So now I have to put in a plug for the Primary Immune Deficiency Treatment Consortium, or PIDTC, funded by NIAID for the last 15 years and also by NCATS. And so this has been a very important group of now 46 different centers in the US and Canada to pool their data because these diseases are so rare that no institution will have enough cases to make robust statistical studies. But when SCID cases were pooled, in this case, it was a retrospective study of still modern era treatment, and it shows the age of bone marrow transplant is a biphasic curve. So there's a minority of very young transplants, and then the big majority are transplanted later than 3.5 months. And so it turns out that the early minority were those who had a positive family history, an affected brother or cousin that enabled the family and physicians to recognize the condition very early and treat it promptly before infectious complications. The big later peak are the sporadic cases generally that presented because of infectious problems. And guess what? There's a big difference in survival. So I don't want to look at all of the little lines here, but on the very top are the ones who are under 3.5 months, and also ones who are even older but happen not to get any infections by the time of their transplant. And their survival is excellent. And on the other hand, that orange line shows that even today, transplanting these infants in the face of an active infection is deadly in half of the cases. So that raises the question of newborn screening, because since there is no family history in most of these, that would be the only way to avert the tragedy of late diagnosis in the big majority. And it turns out that there is a biomarker, these T cell receptor excision circles, or TREX, that are formed when those very smart adaptive immune cells are generating the mature linear gene that's going to be their T cell and B cell receptors for antigen. The leftover pieces get turned into circles, and those circles are stable because they don't have DNA ends to get chewed up. And we can do a PCR reaction across the joint of the circle. And if we see a product, then we know that T cell generation is happening just fine. And if we don't, that's a baby we have to meet. So this has turned out to be a very effective, ooh, it's going the wrong way. Help, help. Oh, I know. Okay. It won't go back. Now it went back too far. All right, we will get there. All right, so everybody wants to do newborn screening using the dried blood spots that are already collected because we can actually afford that. This is these treks can be analyzed in DNA from the dried blood spots that every baby is already getting.
2 minutes, Jennifer.
Okay, and the results of this are in the next slide. So I'm just showing on the left the old kind of SCID presentation, a poor baby with thrush who happened to have pneumocystis and all kinds of other problems. And on the right is a baby who had a diagnosis made after newborn screening, never turned a hair, and is alive and healthy with a completely fine immune system today. So of course, newborn screening, we believe has been very successful, but we actually have data from the PIDTC to prove that, which is actually hard to come by in many newborn screening contexts. And so this shows the survival of SCID transplants from this great big consortium by decade. And we thought that, like childhood leukemia, we might see incremental improvements in survival over the decades as treatment got better. And we were very surprised to see it was absolutely stagnant from 1980, 90, 2000, up to 2010, and then suddenly there was a jump. So what happened after 2010? Newborn screening for SCID started to be instituted. And we actually kept track of which babies had newborn screening and showed a very significant improvement in survival. And of course, screening doesn't just detect the thing you're after. So yes, it detects SCID, but also, as shown in this graph, syndromes, secondary causes, and other infants who are born with very few T cells. These are on a graph here from the newborn screening in California, ones we consider actionable, that is, ones with fewer than 1,500 T cells at birth who should not, for example, get a live rotavirus vaccine. And just to end up, we've also found that SCID is not one disease. Every single genotype has its own characteristics and different survival. So this is data from that PIDTC showing the survival of these different genotypes of SCID, and the worst one, the DCLRE1C or Artemis, is a DNA repair defect. And this has perhaps the worst survival because in addition to not being able to do the VDJ recombination that's necessary for adaptive immunity, these patients are sensitive to radiation and alkylating agents that are often required for chemotherapy. And so that has led actually to a successful trial of gene therapy for Artemis SCID. And one of the big problems here now is how to get a trial that has succeeded beyond our wildest dreams from the phase 1/2 research setting to an approved therapy, and that's a whole other meeting. And
Can you wrap up, please?
I'm gonna stop there.
Thanks so much, Jennifer. So we will have time after our next speaker to have a discussion. So with that, I'd like to introduce Ivan Chin. Ivan is the director of Children's Hospital and has also at Baylor and has also been the PI or the investigator on a number of the programs that we're talking about today, including the Clinical Genome Resource, the Undiagnosed Disease Network, and the United States Immunodeficiency Network. And we're going to hear from him about variant pathogenicity in rare diseases. Thanks, Eric.
All right. Well, thank you for the introduction. I thank the planners and organizers for inviting me to come and talk to you. So I bring greetings from the ClinGen SCID Variant Curation Expert Panel, which we call a VSEP. So many of you in this room know what ClinGen is, but I'll just summarize. It's the clinical genome resource. It's funded by the NIH. Its goal is to really curate the evidence behind the validity of certain whether genes or variants. For clinical practice, we order genetic testing. As Jennifer just mentioned, there are now hundreds of inborn errors of immunity genes, and you get a variant and you have to decide, is this benign or pathogenic? And really the first step is the clinical laboratory has to decide if it's benign or pathogenic. And how do they do that? So everything is done within the ACMG framework. These are the guidelines that were published by Richards et al., the latest guidelines. This is not meant for you guys to see, but it's just to show you again that there are various types of evidence ranging from population data to functional evidence and familial co-segregation and so forth. So all of these pieces of evidence are taken into account. You add up each code. So there are various codes for each one of these types of evidence. You add up all the codes, and that tells you whether or it gives pathogenic or likely pathogenic. So, as Jennifer mentioned, each inborn error of immunity is quite rare. So when you're trying to decide whether a variant, when you're curing the evidence behind a particular variant, right, then, we're using this framework. Then you have to decide if it's pathogenic or so rare. So I'll bring you back to SCID as an example, as Jennifer has already mentioned, and I'm not going to go into all the details, but actually the first molecular cause of SCID was identified in 1972. So we've known about this disease for many years. And the rarity, again, is shown there, 1 in every 50 to 60,000 births in North America. So the ClinGen SCID VSEP has been tackling curating variants in 7 of the 19 SCID genes that were listed in the 2022 IUIS document. So those 7 genes are the ones you see listed there in red. You'll notice almost all of them are autosomal recessive except for IL-2RG, which is X-linked. So, how do we actually curate these? Well, actually, each gene needs its own set of rules or specifications tailored towards that gene. So the VSEP develops these rules, and these rules are formally approved by ClinGen after some pilot testing. And how we develop these rules really falls within ClinGen guidances. So ClinGen has guidances for how you apply each code. They publish essentially guidances for every single code. Here's an example for PVS1 of the algorithm that they published. What you see there is, it's not meant for you to read, but the things that are in yellow, those are the things that the VSEP is encouraged to specify or modify. The things that are not in yellow, we're actually not really encouraged to change much at all. So what have we learned about trying to do variant curation in rare diseases? So I'll give you the examples from the SCID VSEP. And so here are some things. So challenges: lack of sufficient data to meet ClinGen requirements for PS3. That's the functional evidence code. Few published cases for PM3 or PS4. Those are the codes that we use for essentially how many probands or how many affected patients have been published in the literature. application of PM5 and PS1, where that's where you might have a same amino acid as one that's already been described as pathogenic. That's statistically unlikely in a disease that's that rare. And the phenotype for our diseases is usually or often very specific. Um, and so let me give you some examples of some of these challenges. So the PS3, BS3, this is the guidance from ClinGen. PS3 and BS3 applied experiments in model systems. So the PS means strong. The ACMG code is by default strong. However, the guidance from ClinGen is that the strength of that code shouldn't be strong by default, but really depends on use of benign and pathogenic controls that have been validated using that experimental assay. And they use this system called the odds path or odds of pathogenicity. So as I'm a clinical immunologist, I'm not a geneticist. So what would we consider to be a well-controlled assay? So this is a paper published back in 2004 about JAK3 deficiency. And what you see there is that JAK3 will phosphorylate itself if it's working properly. And so here's an experiment assay where they transfected a cell line with JAK3, either wild type or certain mutants. Very classic assay. You see wild type on the left. You see a known null or non-working variant, and so that's your positive control. I guess or your pathogenic control. You have your empty vector, and then you have your experimental unknowns, and those are the three variants on the right. And you see that none of them autophosphorylate, so those are functionally not working. And we would consider this to be a very strong assay. However, for ClinGen, what you actually need for strong validated assays, you have to actually include a lot of known benign variants in that assay. You have to test them as well as some pathogenic ones. So not just one, as I just showed you in that other slide. So that's how you calculate your odds path. And because a lot of these experiments that we're looking at from the literature are old and didn't do a lot of benign or known pathogenic controls, that has really impacted our specifications. So you see here that none of our PS3 codes for our specifications actually can reach the level of strong without using animal models. The best we can get is to moderate, and that's for only 2 of the genes. All the rest of them are stuck at the supporting level. So there is one slight victory in this. So I have to talk about challenges and victories. So here's one victory because we knew about this. So there are ongoing experiments for FOXN1 variants that are being done by Nikolai Van Oors at UT Southwestern. And so he actually he threw in a bunch of benign and pathogenic variants into his model system. And so with that, we were able to get an odds path of 5, and we're able to use PS3 at moderate strength for curation of FOXN1 variants. So let me walk you through a couple of additional examples. Both of them come from IO2RG. So here's a variant that we're provisionally curating. So this variant is absent from nomads, so that gets us PM2 at supporting. And in a experimental assay published in literature, you don't get expression of the gamma chain. And so, but again, as I showed you, this assay has not had a lot of benign or pathogenic validated controls, so the best we can get is supporting. Now, for PP4, that's our phenotypic criteria. This is not meant for you to read, but PP4, that we use a point scoring system for how many phenotypic criteria and what type of criteria does the published patient meet, and you get points from that. You add up all the points, and that's your PP4 strength. So default is supporting, but you can actually, you see up there, you get up to 8 points. You can get strong. So for this variant that I just showed you, the variant, we add up all these different points, and this is curated by, we have actual curators that do all this work. 2.5 points total, which gives you a moderate level of evidence. PS4, so we also use the ClinGen guidances for applying PS4. And so these numbers that we use are what most VSEPs use for PS4. And so we just use these here. So how many probands, excluding the one that you use for PP4, have been published in the literature? And so depending on how many have been published, you get a certain amount of points, and that gives you your strength of evidence. So for this patient, the variant has been reported in 5 probands. But of course we use 1 for PP4, so there's 4 probands that can be counted. And so the result that we get here is PS4 moderate. So when you combine all these codes for this variant, right, you get 2 moderate, 2 supporting. That gives you a classification of likely pathogenic. Now, that's fine and dandy, but really it seems unlikely, very unlikely, that 5 different SCID patients published across 4 reports would have this exact variant, right? They all have SCID, and yet somehow it's only stuck at likely pathogenic. And so, this brings up the need, right, for these really rare diseases. We have to kind of maybe relax some of these criteria for, especially for proband counting in the literature. And so here you can see we adjusted the points, we lowered them tremendously. This is provisional, it has not been approved by ClinGen yet. But, with this result, this would bring us to a very strong category classification. And the final classification would be pathogenic. I'll give you a second example, also from IL-2RG. This is a different variant that's also being provisionally curated. This variant is absent from NOMAD. There are 3 probands published in the literature. We used 1 for PP4, which I'll come to in a second. And so, again, these were the PP4 criteria. And we do have approved specifications for all of these. As I mentioned, they are being revised. So, so we have all these different points. And, if you get to, above 2 but less than 8, you get moderate. Above 8 points, you get to strong. And included within that, are some functional, like, testing criteria on actual patient cells. And what do we see in the literature? So the program we use for PP4, the variant, there was a flow cytometry assay performed that showed absence of surface expression of CD132 and failure to phosphorylate STAT5 in patient immortalized EBV transformed B cells. So we add up all that, we get 3 points. I think I can barely read that. It is moderate, right? And so this is all the evidence that we have for this variant. Moderate supporting, it becomes a variant of uncertain significance of VUS. So, that brings up the point that the CD132 STAT5 defects in the patient cells are actually quite specific for either IL-2RG or JAK3 deficiency. I mean, it could be one or the other, but here we have this variant in IL-2RG. So this brings up the need to maybe increase the strength of PP4 for some of these rare diseases, especially when you don't have the PS3 functional data, as happened in this case. So you can see all the modifications that we proposed for increasing some points to give more evidence to some of these functional assays that are being performed in patient cells. And so with that, this would actually increase some of those things to 5 points each and give 11 points and raise the strength of evidence to strong. And now we can classify this variant as likely pathogenic instead of a variant of uncertain significance. So other things just to throw out there for rare diseases and IEIs. So if some of these genes do come with incomplete penetrance, and that brings up the need to downgrade BS2 in some situations. And I gave some examples there. So for, we're also curing RMRP because that can cause low T cells and abnormal newborn screening tests for SCID at birth. What we've learned with that is expressivity, right, for some of these inborn errors of immunity it can be highly variable. And this is becoming more and more appreciated, especially with, as Jennifer mentioned, some of these autosomal dominant conditions that are being newly reported or discovered. And the other thing to remember is for some of these newly reported genes, the entire phenotypic spectrum just remains unknown because they are so new. And then the last thing is that not all IAIs are monogenic, and that raises different problems. So my summary for rare diseases using inborn errors of immunity and SCID as an example is that a wealth of published data, you just can't expect that to be found in PubMed. The PS3 strength classification based on the odds path might be a little bit too stringent. It's something to think about. Each case that's identified in the literature is highly valuable. Or if it's in population databases on the benign side, right? It really just carries a little bit extra strength, I think. Highly specific phenotypic features can be compelling, and guidances from both geneticists and disease experts is absolutely essential. And so I want to, show you the current ClinGen SCID VSEP. So I'm a co-chair. I'm the immunologist co-chair. My co-chair is Britt Johnson from LabCorp Invitae. So she's a geneticist, and actually she's the one who, I tend to like to stick to rules and guidances and guidelines quite closely, and she's actually the one who's a little bit more relaxed, like, oh, maybe we should relax some of these guidelines a little bit. So it's helpful because otherwise I wouldn't know exactly how far to go in terms of relaxing things. And you see a number of people on this panel, especially on the right side. We have geneticists and immunologists, actually SCID experts on this panel. So this is, again, my summary that I want to leave you to. I want to thank the support from ClinGen, which is being funded by the NHGRI. And all of this VSEP work actually is being supported by NICHD down the lower left corner. So I'll leave it there.
Okay, well, thanks to our two keynotes, we now have time for discussion, a good healthy block of time for discussion. I will tell you it is impossible to read names on the placards at the other end, but what I think people could do is which we often do, is just if you have a question, take your placard, just put it up. And we'll call on you. We will fail, except I know Bob, so I will have his name. But in the case, if we don't call you by name, it's because there's no way we can read at the other end. But and if we're not seeing you and you've put your placard, just wave and Jeannie or I will see you and we will call on you. So we open this up for discussion. Bob?
One quick thing, Britt Johnson's now at GeneDx, head of medical affairs. She left LabCorp. The comment I wanted to make has to do with some pilot experiments that were done in Invitae while I was still there, using laboratory data, not just published data, and using machine learning approaches to variant interpretation. And without going into a lot of details, the bottom line is we are way too conservative in calling variants of uncertain significance. The vast majority of them, we're talking about 85, 90, in some cases 95% of variants being called VUSs are benign. And that mostly comes from looking at patients referred with particular phenotypes. And in the area of the immune deficiencies, you have so many excellent assays, phenotypic assays, for being able to determine whether a variant is deleterious or not. We are underutilizing that information and causing too many VUSs.
Mark, online. Oh, there's Mark Williams.
Hi. Sorry I can't be there. Very nice presentations. This is a question for Ivan, and it relates to the phenotype specificity, because this is certainly something as a geneticist that when we're dealing with variants that come back and trying to make a determination as to whether or not they are clinically relevant, we do rely on that. But at least in terms of non-laboratory phenotyping, it can be very difficult to establish what really constitutes specificity. It's subjective. But, for something like severe combined immunodeficiency, it's not only that the phenotype is pretty specific, but it's also a very rare phenotype. It's not something that you see. It's not like a congenital heart lesion or deafness or something of that nature. So one question is, how within your VSEP are you thinking about clinical phenotype specificity? And then the second question is, the other thing we deal with is that in the literature cases, a lot of times the phenotypes are under-described. And there's not consistency to the description. So is there a role for the VSEP to say, hey, if you're publishing an immunodeficiency case, here are sort of the minimum criteria that we need to have in the paper to be able to establish phenotypic specificity?
Yeah, so that's a great question. What phenotypic features really count towards PP4, right? And at what level of strength? We think about it all the time. I mean, Jennifer's on this VSEP, so we think about it all the time. The unfortunate thing, as you mentioned, is old papers are old papers. I mean, there's nothing we can do about that. It does become incumbent upon people to really, especially when you're reviewing papers, to really push upon authors to provide more information, phenotypic information. But the other thing is we are able to use data for our variant curations from commercial labs. So the more information a commercial lab can get from the provider who ordered that test, that becomes highly valuable for our VSEP.
Terry?
Yeah, Ivan, I was wondering if you could comment on, the average practitioner who is working with these variants, et cetera, may not know how to pursue them necessarily. And I know there are opportunities for people to actually contact the VSEPs and say, hey, I have this variant, can you help me, to classify it? So do you do that with, rare diseases? Do you have outreach, or do you have people contact you about, what do I do with this variant?
That's a good question. So we don't have outreach because we do have that grant funding, but it only goes so far. But we have had at least one person contact us about the case that they've had. So we do have biocurators that can take upon the task of curating a variant. If perhaps upon request.
Yeah, I ask because I've had an experience recently with Modi, so a different, a totally different disease where, basically there was a variant that was almost there for pathogenicity, and because I happened to know the head of the committee, et cetera, could contact them and say, hey, we have this case, could you reclassify it, which they did, and there was a diagnosis, and we could then go into treatment. And so you sort of wonder, and Jonathan, I don't know if you want to comment on this, you wonder if there's a role. I mean, ClinGen is doing so much, 2,500 investigators, 60 countries, et cetera, but is there some way to be reaching out or to at least accept comments from from primary care practitioners or others in the field? So
I just wrote it down as a grant name for our renewal. I mean, I think it's a really interesting idea. So I mean, one of the challenges is we've got an incredible breadth to cover across all the different domains. And so making the specifications for one gene takes so much work. We've got to get scaling past that. So there's a lot of thinking about how do you sort of scale the variant specification process, but at the same time be able to scale how do we provide that resource to people to be able to, ask those questions, right? Here's an interesting variant, what does it mean? So I think it's a worthy sort of consideration of those two goals. Can I ask my other question? So I had a question for Jennifer. So just back of the envelope calculations, it looks like probably 2 to 3% of coding genes are involved in immunodeficiencies, something like that. And so I guess my question is, are we getting close to the full set, or how much more are we going to discover? And then are there places that we haven't looked, like non-coding? It seems to me like non-coding RNAs must be involved in regulation of the immune system, things like that? What are the potential areas that we're currently applying to?
Well, I think that's a great question, and since we're still growing at an exponential rate, I don't really have any idea where it's going to plateau. And certainly, there are non-coding sequences that are now associated with particular errors of immunity, and you know, I think the tools now to be able to start with a patient, do not just exome but whole genome sequencing, and look very, you know, look at the whole genome in detail. It is a great opportunity, and these inborn errors of immunity offer models to help elucidate a lot of mechanisms of disease beyond just the immune system too. And I think, you know, some of the things we never dreamed of are disorders of glycosylation causing immune problems. And I think it's worth remembering that the T cells and B cells proliferate a great deal, perhaps only secondary to GI proliferation. So that the functions that require a lot of proliferation are susceptible to a lot of different kinds of genetic lesions.
Thanks, Jonathan. Maybe we could just go down the row here. It looks like we have 4. So starting with you. Sorry, I can't see your, if people could put their name tag so that we can at least with binoculars see the names. I do see Scott. I can see Scott. And then we'll go down 1, 2, 3, assuming that Mike, yours is still up for a new question, but go ahead. Scott.
Scott Kenna, rheumatology. Just first a comment Dushan and I were talking about. I mean, if you've had one outreach to VSEP, I get about 10 a week for variant interpretation from people around the country, and I'm not even good at it. My question is really about whether VSEP or anybody gives guidance to clinical sequencers about reporting of VUSs, because clinically we get clinical sequencing data from a variety of different places in a variety of different forms. We get exome backbones, genome backbones, panels, and the guidance on what actually gets reported is incredibly variable even within companies. And so sometimes it's incredibly frustrating because, you know, you'll get something back, a whole exome, and it'll have no reported variants. And so then, you know, we go through the laborious process of trying to get the primary data, but
Ivan, it's hard to read your intentions here. What's that?
Question is, does anyone, like the VSEP provide guidance or at ClinGen?
So, guidance, you mean to the commercial labs, the clinical labs? I mean, we don't. You know, the specifications, you know, as far as like, let's say PP4, right? What phenotypic criteria are needed to make PP4 strong even? They are available on the ClinGen website for commercial labs to look at, but we just don't have the resources to tell all the clinical labs.
Thanks. The person next to Scott, whose name Stephen.
Thank you. For Jennifer, you showed some really cool data, Jennifer, on SCID related to outcomes. Maybe the most beautiful data I've ever seen related to improvement in outcomes in any field. And you showed convincingly, I think, that if a baby's diagnosed within 3.5 months, they have the best outcome, and if they're diagnosed before any infection, they do similarly well. Couple of questions related to that. You know, these days we can do better than 3.5 months. We can do 3 days or 5 days or 1 week. And so first question would be, you know, how much better can we get and how much room is there for improvement? And then the second question would be, that was for SCID, and SCID's a very specific subset of immune deficiencies. And I'm wondering whether there's similar data across all immune deficiency disorders.
Sure. So to start with your second question, I think there are many disorders of immunity where early diagnosis would improve management and outcomes. And we happen to be lucky with SCID to have a biomarker that you can just do a quick little PCR reaction and get a very clear answer. The other immune deficiencies don't have a wonderful marker. People have tried for B cell kappa chain of the kappa light chains of immunoglobulins also have circles in their production. The trouble is B cells are a lot more rare than T cells, and there's kappa and lambda, so the signal to noise is not favorable, and that's why those haven't been implemented. It's just the number of false positives you would get for every true positive makes this not really a feasible newborn screen. And after that, what can you do with a dried blood spot? Well, you can do a whole exome or whole genome sequence, but I think as Ivan already pointed out, we're always finding new variants, and we have not saturated the genes for variants for these rare diseases, not even close. And so you can't really interpret the genomic data yet in a way that would be that I could see would be feasible for screening. And I know this is a hot topic that a lot of people are thinking about, because we really would like to diagnose these conditions early. Now, your first question was, should we do better than 3.5 months?
Yes.
And is
yes,
even earlier better?
Exactly.
And so again, this data is about SCID. It's interesting that newborn screening is great. It's not perfect. And one of the big problems we found is that cytomegalovirus can infect infants with SCID who are breastfeeding, even in that first week of life when the dried blood spot is sent off to the lab and coming back. And CMV and SCID is a dreadful, lethal condition in many cases, despite effective antiviral treatment. We're not quite there yet, but I don't think it's, pushing back from 3.5 to 3 to 2.5 months. It's more what are the pathogens that we're missing, and how could we address that? And I know people are thinking about newborn screening for CMV. I'm not sure that would do it for this problem, because even by the time that newborn screen comes back, whether it's from CMV in the birth canal or CMV that's shed into breast milk, 1 or 2 virions seems to be enough to really do in a SCID baby.
You could screen the moms and do PCR of breast milk, or sort of, genital PCR for CMV, and that, or you could do serology before. I mean, there are other ways to, I think, figure out if there's CMV exposure in the infant. Maybe.
Now, people have tried, and they've also tried to eliminate CMV from breast milk by freezing, by pasteurizing, by heat shocking. And there's a big, sort of cottage industry of breastfeeding advocates who try to do this. None of them are FDA approved. And again, for preterm infants and their CMV disease or other conditions, than immune deficiency, this might be helpful, but for SCID, I wouldn't trust it.
As long as they're not giving them raw milk with H5N1 in it. Sorry, I had to inject a current topic on my mind. So let's go to Bob, and then we'll go to Gail.
I just wanted to comment on this issue of inquiries about VUSs and variants. Just as was said before, the commercial labs get dozens of these a week. Obviously not about SCID, but about everything. And I think the problem is that often results in going to the clinician who's making the request and saying, well, what more information do you have that you didn't make available to us in the initial TRF request form? And we get lots of more data. And then the lab says, oh, well, now that we know this, we can interpret things differently. That then gets percolated eventually through a reinterpretation through ClinVar. It's slow. We're talking about months and months and months. Going the other direction, it's available on the ClinGen website, how things have changed, but that really requires sort of constantly pinging it. And so what I'm thinking is that it would be great to establish stronger liaison between the ClinGen working group, particularly for variant interpretation, but also gene phenotype correlation with the clinical labs, and have it be a faster back and forth. The submissions to ClinVar are expensive. I mean, in terms of time. You know, at the lab where I used to be, it was basically half an FTE to just take care of ClinVar submission. And it only occurred like twice a year at most. So there's a lag when this additional information comes in and a variant gets reinterpreted.
Yeah.
Yeah, so Jennifer caught my attention when you said you're learning about variable expressivity in these disorders. And I'm thinking of this in the context of, you know, we see adults who have, you know, sort of a chronic low T cells or whatever. And as long as they're not getting sick, we kind of leave it alone. And are we learning about the, you know, milder disorders in adults related to children? Is that a different set of genes? When do we sequence an adult with one of these panels?
Oh, that's a great question. And I think people are paying more attention to immune disorders in adults, whether those are acquired or multigenic or single gene disorders is still up for grabs, I'd say. And I think it's probably all of the above. But on the other hand, sequencing is available and way less expensive than it used to be. And in fact, it's a lot cheaper to get a gene panel than to send lymphocyte subsets. And so I actually, in my own practice, sequence much earlier than I used to. And I think most of the time we don't learn much, but when we hit a home run, it's life-changing for the person involved. And I think it's something we should do much more of.
Steve?
I would just like to second that. I think the value of, for instance, lymphocyte subset testing, which is tremendous if you're dealing with somebody with HIV infection, to really see what the CD4 number is. Beyond that, I don't know in an adult. And so I would echo, I think the tyranny of infants, it's wonderful, and I'm all in favor of pediatrics, but so much of what we've learned about genetic disease has been with this paradigm of SCID and early life onset. And we may need a different set of paradigms for adult disease. I think so much of what we think about adult immunodeficiency has been heavily influenced by HIV, which is a tremendous model, but it may not be the right model for where we need to go from here. And I think if we're not sequencing, I don't know how we figure out what's really going on. And the amount of phenotypic effort that's put into adult immune deficiency, frankly, is a ridiculous waste of time and money. And I really think we've got to start moving, even if this hit rate is 20, 30, 40%, that's still higher than the hit rate I think we're getting for many adult immune defects.
Yeah, so I guess, you know, my question is the hit rate, you know, and for what patient population is the hit rate 10 or 20%, which I'd be thrilled to get back. It's really interesting to hear that the T cell subsets like cost more than the sequencing. But I also wonder, is it the same set of genes even, and do we even, and we're not going to know till we look, right? But we don't often start by looking in clinical populations. We start in research populations, and it's outside of my field, of course, but I'm not really aware of a lot of research on adults who don't have significant, problems with infections.
Well, of course, significant problems with infections are a large part, and I only know about the people who have that. But in that setting, by the time we see them, they've already been through lots of phenotyping, which has not been informative, which is why they keep looking for answers. And in that context, the hit rate, we'd have to go back and stratify by age, but the hit rate is significant enough. And the genes, there's an overlap, as you might guess. There are some late presentations of IL-2RG that either have reversions or are hypomorphic. And then there are other genes that really don't show up in infancy. For GATA2 as an example, virtually never shows up in early childhood, but does show up in later adulthood, which makes for a very complex, how do we, I think one of the things that's not factored into many of the stratifications is if you test people with some conditions at age 15, they're normal. If you test them at 50, they're not. How do we put that into the really ponderous efforts of ClinVar to say there's a time dependency in how we understand genetics?
Yeah, and I would say it's of course time and exposure. So there are conditions where your immune system can't handle a certain situation, but you'll never know until that situation arises. And then the other thing I would just bring up again is this flip side of immune dysregulation and immune deficiency. And they can go on in the same person or the same family and be attributable to the same gene and the same gene defect.
You're going to have to wave at us because if anybody has questions, we really cannot see placards. So please don't be shy. I see Bob again.
This discussion about people going through lots and lots of phenotyping without any kind of significant answers is just so reminiscent of intellectual disability in the young, and the fact that we've now come around really to the idea that stop the diagnostic odyssey, do a whole genome sequence first. It shouldn't be the last thing you do. And to me, it sounds like adults with immune dysregulation, we should consider doing whole genome first and save all the other phenotyping expenses. If you get 20%, that's sort of where things were originally with whole exome. And it's gotten better and better and better now with whole genome. I think we're up to 40, 50% hit rates, maybe even higher in some cases. We may see the same thing and learn a tremendous amount.
Jeannie?
Maybe we should think that.
Carol.
Yeah.
Okay, Carol, then Jeannie, then Terry.
Thanks. So Mark Williams actually has a comment in the chat that mirrors something that I had wanted to ask for Ivan, which is, so there are other tools like MIM Matcher and Gene Matcher and maybe Genome Connect that might have information related to, curating the variants and disease associations. And I'm wondering if those are employed. And then Mark also asked, entered the comment, perhaps a component of the feedback and communication could be tested within the new eConsult network.
Yeah, those are great points. I guess the problem with gene matchers, we would have to enter the gene in there and see what matches, right? And for like IL-2RG, I mean, who knows what you're going to get? But we do leverage the resources of whatever we can get. So again, we have Britt at GeneDx. We have biocurators who are actually working at Invitae and at Prevention Genetics. And so they're willing to volunteer, like searching their databases. And then we have the experts themselves, right, who maybe have unpublished variants that they've looked at through the decades in their lab. So, you know, we leverage as much of these resources as we can.
Maybe a little bit of a perspective from sort of the practicing infectious disease world. You know, I guess I'm curious about at what point, well, first of all, has there been much interaction between the community of infectious disease providers who are likely to see some of these folks who have persistent, recurrent, unusual infections, say your average patient who has invasive cryptococcosis without HIV infection? Clearly something's wrong there. Do we really know what that is? So has there been a concerted effort to increase the dialogue between that community and these kinds of resources? You know, I know Steve, so I would call him if we had somebody who, you know, I wanted him to work up, but that's a pretty extraordinary access point. So is that something that would be helpful? And I'm just thinking of our infectious disease meetings. We don't often talk about this. And I guess at what point could there be guidance about when you should call your friendly genetic consultant to ask about the point that Bob made, you know, before you go down these many months, sometimes many years of phenotypic characterization, when do you actually pull the trigger and think about as this work gets more advanced and you get more tools and recognition to actually do whole genome sequencing? That might be something really interesting to think about, for example, with IDSA.
And has that, I'm just gonna, Carrie, has that come up? I mean, that sort of scenario, we've done so many demonstration programs over the last decade around genomic medicine implementation. I guess that's never really sort of fit in neatly. I mean, this may be the meeting where that happens, but I'm just curious what our history has been.
Yeah, no, we really haven't done something like that. And, you know, and it even raises the broader question because, you know, Bob mentioned intellectual disability. Well, when do you push the button to say, okay, I need some help? And that was going to be my question is, you know, so thank you. We're of a mind here. Yeah. So, you know, can we come up with, and maybe that's an expert panel, maybe, you know, who knows? But when is an infection unusual? So last week I saw a 71-year-old with pertussis who had been vaccinated. It's the first time I've ever seen it. I mean, is that unusual or not? So it's not clear. I think it's a kid. Yeah, she said she thought she had gotten a Tdap as well. Yeah, but anyway, you know, so how would I, as a dumb internist, know that, oh, this is unusual, I should contact someone?
Well, and I think just to follow that, just to kind of build on other programs, right? I mean, some of the questions of how do you facilitate genetic testing is how can we identify those patients? So are there EHR phenotypes that can be generated that would sort of say, okay, this is a really, you know, suspicious case, so somebody needs to do genomes. That's genomic learning healthcare system. I think, you know, the e-consult network is mentioned as a potential way that those types of things could be tested and implemented. So I think it fits really nicely with the whole sort of set of programs that have been developed here.
And by the way, I'm just going to put this table for future discussion because Terry, I mean, I think one of the things, and maybe this would be one of the actions coming out of this workshop by the end of tomorrow is, I mean, is it just an expert panel or is it a research program? And, you know, and lots of other options in between. I mean, is it just a matter of getting people together and talking about it, or should we do that and then study it? And so,
yeah, so one of the challenges with obviously clinical medicine, as you know, is that, yeah, we can have a research project that takes years. We have patients in front of us now. So is there a way to, you know, start with the expert panel, get some kind of guidance, and then do the research?
do both. Of course, they're not mutually exclusive. So Casey, maybe you're next, or yeah, we'll do Casey and then we'll come back over.
Yeah, so thank you, Casey Taylor. My background's in informatics, so I'm learning a lot about these conditions. So one of my questions was, point was raised around family history, and it's low capture. And I just wanted to hear more about that, whether it's the knowledge of family history, or is it that it's not showing up where it can be known by the family? Because just thinking about tools that would help have richer like phenotypes that would be relevant.
Well, I think it's both. So I think sometimes when an immunologist or maybe an infectious disease person takes a family history, they say, you know, does anyone in your family have SCID? Or, you know, it's too narrow. And one of the things that we learned is you have to ask about deaths early in life. And I'll never forget a family I asked this question, and they said, well, we don't have any skid, but there were 4 male uncles who died of cystic fibrosis in their first year of life in the 1960s. And of course, looking back, I think, they had diarrhea, they had failure to thrive, they had pneumonia, and they died. And it was called cystic fibrosis. And what do you think it was? So when you ask about a family history, you have to ask with a way that you can capture the information that might be relevant. Having said that, though, as you know, even with X-linked SCID, 1/3 of the cases are the first manifestation of a new mutation, and we've seen that over and over. The old Haldane rule applies, and
What's the old Haldane rule?
the old Haldane rule. I always explain it as it's the bathtub of mutations. So you have a bathtub, and that's the population with a certain number of populations and of mutations on this gene, and there's a drain. So every time there's an affected male who doesn't reproduce, that goes down the drain, and then there's a faucet, and that's the new mutations coming in. And at equilibrium, you have equal new mutations from the faucet and draining. And so it turns out 1/3 of the mutations are new because we have males and females, and in a female, the mutation doesn't show up. So it's the carriers are asymptomatic, so their mutations persist in the population, but when it is in a male with only 1X, then down the drain.
Here, Scott.
Thanks again, Scott Kenner from Philadelphia. So I have a question, but a couple responses. So in my institution, the answering the question of like, how do you decide if this is weird is a multidisciplinary question. And so to do that, we've assembled, and I'll talk a little about this, a multidisciplinary immune dysregulation team. And we're working, we have a paper that's in press right now about how multiple groups, including ours, have done that and how that functions and how there's some barriers to how that functions. But the answer is almost always, yeah, we should sequence that patient if we can possibly get to it. But my question is really about, I think, all of us almost every day use Nomad as this amazing resource where, all these exomes and genomes live in the same place and they get reanalyzed. And I think that there are a few examples, the clinical sequencing program maybe being the best, of how having that kind of a body of genomic data in the same place that gets constantly reanalyzed is incredibly useful. The Vexus story sort of mimics that as well. And so I'm just wondering, are there any efforts to try to, other than what, individuals have been trying to do, collectively at the NIH to, take all of the sequencing that gets done, either research, or even some on a clinical basis, and get it all in one place so that it can be revisited, reanalyzed, and potentially sort of build on what's already been done.
Why don't we do this one and then we'll go to Mark Williams online. Oh, and then back to Steve. Okay, yep.
Jillian Hooker, I wanted to go back to, I think mostly Terry's question about how do we come up with guidelines for who to test. And in my day job, I spend a lot of time taking guidelines and turning them into payer coverage policies. And one of the biggest challenges we face is just the remarkable variation across specialties and how they approach testing guidelines and exactly this question of who to test. And some are so specific that in order to follow the guideline, to the earlier point, you might do $10,000 or $20,000 of evaluations just to find out if someone meets criteria for genetic testing. And that's not that practical, right? And I think it comes from a confusion of diagnostic criteria and who to test criteria. And I guess my main point would be, I think it's really important that we separate the difference between those two things. Like where a genetic test might be useful is not the same as saying somebody who has all of these clinical findings probably has this diagnosis and therefore we should do genetic testing.
Mark Williams online.
Thank you. As the discussions progressed, it strikes me that, we have resources that could really be tested to explore some of these questions. And Terry, I recognize that I'm perhaps straying dangerously into discovery, but I think that if we look at 100,000 cohorts where we have sequenced individuals coupled with phenotype, that we could approach a couple of these questions that seem to be very salient, which is, okay, we have a gene, we have variation in the gene, this is what the variation looks like. Is there, in fact, a consistent phenotype that could be seen with this? That could be something that could lend itself to a PheWAS study if we knew exactly what sort of variation we wanted to study. Conversely, if there are certain phenotypes that we think are highly predictive or specific for potential immune deficiency, we could take a look at individuals in our data, electronic health record data, to see if we can identify those. And some of them would be, organism-specific information that could be extracted, I think, relatively easily from microbiology reports, and say, okay, are we seeing variation in specific genes that could potentially explain this? Now, granted, this is not going to be as easy to do for autosomal recessive conditions, which tend to predominate in this space, but it would certainly be a way to identify some contributions from monogenic and X-linked conditions. So another possibility of how we can use existing resources to begin to test some of these questions that are emerging.
I want to inject a question I've been having for a while, because I think it piggybacks very nicely on what Mark Williams was just talking about, is do we feel we collectively, people at this meeting, maybe people online or joining us, are capturing international experiences in this arena? Especially I'm thinking about where there's activities like in the UK that probably are surging ahead of capabilities or what we have here right now. And I'm just not connected in this arena. Some of you might be. But if anybody has any perspectives of, are there opportunities taking advantage of ongoing programs or existing data in other places of the world that are looking at population scale data collection? Is that also another opportunity, or just we should make sure whatever we do going forward, we include those voices and those experiences? Just a thought. Anybody?
So these two people might.
Yep.
Yeah, well,
So-
I think Jennifer was going to answer.
Well, I just wanted to mention, taking off from Mark Williams, the Jeffrey Modell Foundation has a list of 10 warning signs for primary immune deficiency that they have been publicizing worldwide for decades. And, there you can pick at problems with this list, but there's actually a recent publication showing that although these things are non-specific, like, pneumonia that doesn't get better or whatever, how many infections in a year, non-specific, but a starting place that has been actually validated. And is published around the world.
I'll let you handle this side.
I actually was going to ask Steve to comment, because the question about global reach, and I did want to ask you, Jennifer, given that the SCID screening just started in 2010 in the US and Canada, how widespread is it globally and what does that look like? But before I do, let me just mention that, I would think that areas of the world where consanguinity is still practiced, still maybe not the prevalent practice, but still there, would afford a very rich source of this. And I think Steve and I have talked with colleagues in, particularly one colleague in our Middle East, North Africa, Turkey program, the MENAT group. And I think there is an effort to start doing that. And I wondered if you could comment on that, because particularly in Northern Africa, there are some places where this is still an issue.
Well, I think, there's the regions with high levels of consanguinity have long been known to have high rates of immune deficiencies, as well as many other conditions. And trying to make this available at a population level is certainly of great interest to the governments and the academic institutions there. and trying to work collaboratively and prospectively with many of those groups in countries throughout the region. I think many people from different institutions have been engaged in that, and it is a powerful tool to be able to identify some of those conditions and then to bring those patients to treatment. Everything from, as you mentioned, Chapel disease to SCID to many other forms of immune deficiency. And I think governments need to be thinking a bit about what are the consequences of identifying these things. Identifying them early is obviously better than identifying them late, but then how do we go about treating them? And we're very engaged in trying to do that with governments in the region. I wanted to come back for just a moment to the questions that always come up, and I'm speaking now just as a clinician. This desire to choose, the weird presentations, the difficult sequence. And I just want to reflect back, being old enough to talk about when HIV was first identified. And when early on, in order to test somebody from HIV, you had to get an ethics consult in order to do that. And then after a while, people said, well, okay, maybe we don't need to be quite that exclusive. And then it got to the point where for every person admitted to the hospital, we said, this is so important because there are years during which you're asymptomatic. We don't want to wait until you're on death's door to figure this out. We want to figure it out while we can do something. So we dropped that sort of, requirement. And I think to the great benefit of the world, my God, look at how effective that's been. And we've got treatments for at least around the immune deficiency space. We've got treatments. So I would urge my colleagues at institutions, I think as Scott was alluding, if you get to the point where two clinicians are wondering, is there a problem? Hell yeah, there's a problem. That's the time to sequence, not to say, well, I'm sorry, you need 2 more intubations before we think it's worth doing the sequencing. And we've got to start moving because the problems we're going to have of curation, of understanding the variants, these are only going to get more complex. And just like I don't think there's a clinician out there who would say, I only want the CBC with the eosinophil number. I don't want to see the rest of the diff. And there are very few clinicians who have ever had the opportunity to compare a chest X-ray to a CT scan to say, oh yeah, give me the lousy data so that I can, infer what I really care about. That's where we're going to go. And we got to start getting ready for what is, as the prices come down, it's inevitable that this is just a couple of years away. And I just think we better be getting ready for the torrent. And I gather that my colleagues in industry are already dealing with this, and how we prepare ourselves for what is on the way, I think is our task at hand.
Okay, I cannot read.
No worries, all good. So I'd like to echo Steve's and what I think is the prevalent opinion here. I think we need to be forward-looking. I think as we take vitals to a newborn, we need to do whole genome, and that becomes part of your epic record, right? And that alone is the beginning. Then there's everything else to solve, and we'll talk about that. The variants of non-significance, important, non-important, but we need that information, and it's at our fingertips. It's affordable to do. Some countries, to answer part of your question, I think Norway is trying to implement whole genome sequencing for all newborn babies. And as the price is coming down, I think we need that as much as I need my blood pressure tested when I see the doctor. And maybe not yearly, but somatic genetics is an important aspect. So we probably need to revisit that in adulthood as well. So it's never too early, if you ask me, to have that information, and then there's a ton of work to be done.
Price is coming down for the sequencing, but what about for the storage of the data, right? I mean, that's, you don't know.
Absolutely. Yeah, it's, I think UK Biobank has transformed the way we look at genetics and genetic disease associations, including infectious diseases. And that just gives us a whiff of what's possible. And maybe we need to do at a federal level, state level, partnering with private. There's a lot of private initiatives, sequence million individuals, right? So, I mean, 1 million is, 329 away from the whole US population.
Yeah.
So we,
So,
we
and
can get there.
storing the VCFs is not expensive. So it's storing the full files, which no clinician is going to use. So maybe one last.
Well, then we'll take a break. 2 more.
Stephen, and then the
Yeah.
person.
Just very quickly, to answer your question, Eric, and it's already been highlighted a little bit. There are now, I don't know, 30, 40 groups worldwide doing newborn screening by genome sequencing. Immune deficiency disorders are on everybody's list. I don't know that anybody has a comprehensive set yet, but many of those are tapping into populations that are just, we don't have reference population sets for currently. So that's very exciting, and that is a resource that is well worth looking at, even though their thrust is not primarily on immune deficiency disorders. It's going to become one of the groups of disorders that cost effectiveness shows to be worth looking at.
At the end?
Magda Walkiewicz, the NIH. I just want to make a comment about the family history and importance of that in analyzing the data and also disclosing or obtaining a pedigree. I think unless you ask very specific questions about a certain phenotype in a family, you're not going to get an adequate family history. People often don't think about diabetes or pancreatitis or other phenotypes that are associated with possibly autoinflammation as related at the time of that conversation. So I think I just urge all providers to just really ask pretty specific questions, and Morgan can comment on that a little bit more since she's the one who obtains those pedigrees. As far as analysis of the data, for all the people that analyze the genomic data, either from a panel or exome or genome perspective, I think it is really, really important to remember about the non-penetrance and variable expressivity. We often see variants inherited from unaffected parents that are missed or not considered just because of the fact that that variant seems to be inherited from a parent that doesn't express the disease at the time of the analysis. Very often when we go back to these families and we actually disclose these results, these parents are not necessarily not affected. They might have a cellular and subclinical phenotype. And I think it's important that when we analyze these data, the genomic data, we take these points to considerations in the immunology field.
Okay, I think we're going to wrap up this session. Thanks to our 2 keynote speakers to kick us off. Thanks for a vigorous discussion. It's been great. Terry, you want to tell us what's next?
So we're back at 11:05, please, and I think that was all. So thank you all very much. Let's applaud our speakers. Hi, everyone. We're going to start up in 1 minute. So 1 minute, please. So I need to call on Jonathan and Lucas, who I don't see. Oh, there's Lucas. And Jonathan, if somebody could shepherd Jonathan in, we're going to get started again, please.
I go outside.
Yeah, why don't you?
I appreciate it. I think it's straightforward.
So. Okay, Jonathan will be here in just a moment, I'm sure, but.
Good morning, everyone. Please take your seats. We're going to get started in just a second.
Great. Thanks, Lucas. And just if we could remind people, you really need to be on top of these microphones in order to be heard well. So as you can see, I'm almost swallowing it and you're still not hearing me. So, all right, we're going to get started. Here comes Jonathan. Go ahead.
Good morning, everyone. Thank you. We're going to move into session 2, pathogen first, phenotype first, and sequence first approaches to genomic medicine implementation and infectious disease. And we're going to start off with our very first talk, which is from Dusan.
Hi, everyone. Thank you for invitation and thank you for organizing this meeting. I'm enjoying it quite a bit. Do I have to be a little bit closer? Okay, I'll do that so you can hear me. Okay, so let's talk about rare infections and how we approach them currently in the clinic. How can we implement that, and maybe what are the challenges of things to come? So first, thank you to people who did a bunch of work in my lab. Inborn errors of immunity were described extremely well by Jennifer Puck here, but essentially we have about 500 genetic disorders that lead to them. Jennifer likes this graph as she pointed the other way around, and I don't disagree. But what this is a phenotype first approach. So we'd start with somebody who we think needs sequencing, and then we do the sequencing, identify, and those that are luckier than others, sometimes there is actually precision medicine intervention that's absolutely wonderful to see. Okay, so mutations in these immune genes can lead to essentially susceptibility to infections via bone marrow failure, cellular deficiencies, or antibody deficiencies that we've touched upon, and also they can lead to autoinflammation, immune dysregulation, which is really this immune dysregulatory phenotype that people have mentioned. So as I was putting this talk together, I was like, well, what is the right way to implement this? And just at the time, Scott actually emailed me and said, hey, this is what we use, right? And here is the immunodeficiencysearch.com, which is an algorithm. This is something that any provider can use in their decision making, whether or not to sequence an individual. And clear, as you can see, if the patient is not responding to therapy or not clearing the infection, I would say no matter the age. Of course, in children, genetics is going to be more penetrant, but now we have quite a few cases of adult onset disease. You can click here on, let's say, viral disease that this patient is not clearing and it's just persistently there. And if you click on that, the next thing that can show up is this CMV, is this herpes, is this very severe influenza, is this JC virus, BK virus, HHV8, varicella, HPV. And here for just simple purposes, let's say we click that this patient has, recurrent herpes that is not just labial, but is showing up on other parts of the skin, and what could be. So if you go and click there, this is what shows up. It can be NEMO deficiency, it can be, of course, Westcott outreach treatment, it can be STAT1 deficiency, TLR3 deficiency. So at that point, what a provider can do and should do is say, okay, so it could be genetic, let's send it for sequencing, right? And what do I do? How do I do that? Where do I send for sequencing? So Invitae panel, which we've talked about here, covers 429 genes currently that are known, mutations in which can cause some of these infectious and otherwise phenotypes. So results come back with a variant that is called pathogenic, case solved, meaning there is exact same variation in TLR3 that causes herpes simplex encephalitis. Let's say that's the case. And now we know that we have a genetic diagnosis and there is genetic counseling for that can be done for the family members. Most often what happens is that the results come back with a variant that is called unknown significance. So you have a VUS or variant of known significance, and that's really the stumbling problem right now when the results come back to the provider. This is where the provider says, what do I do? Right, so they can email Ivan, they can email Scott, they can email me, and usually it works through that. There's a couple of listservs that providers may or may not know about, depending how intimately they're linked with the research community to seek help for that. So NIH has heard us and funded two different programs. One is the New York Royal, the other one is NICER, which helps with this functional validation and ideally reclassification. Because for Ivan's team ever to look at this variant of unknown significance, it needs all these functional and all otherwise tests that need to be published for them to be part of the ClinGen, so then Invitae can call this pathogenic as opposed to variant of unknown significance, right? So there's really layers to that. And these two programs help accelerate the speed at which we can feed our data via publications and direct submission to ClinGen for this reclassification to happen so that provider gets a result back and need not consult us on more things. So that's one idea. So results come back negative. What happens is that if you decide 429 gene panel, it's probably good to refer to research, whether it's a whole exome or whole genome sequencing research that can then again start the same process with us identifying novel variants or even novel genes that can cause these infectious diseases or immunodysregulatory syndromes. Okay. So how can NIH initiatives expand use and diagnosis of infectious diseases via whole genetics? Currently, most variants in genes remain functionally not annotated. And this is true not just for genes that cause infectious diseases. This is true for genes that cause any disease. So one way, mostly we have 2 to 3 known disease causing variants. Most identified will be variants of unknown significance. So I'm of the thought that we need a forward-looking genomic database. It's a little insane, but we were insane before, and Whole Genome Project brought us to where we are today in medicine, which I think is absolutely unprecedented. So the next step in my book is to do forward-looking genomic database where we functionally annotate every and single SNP in every and single gene, and go beyond that and functionally annotate at saturation every single nucleotide so we can be forward-looking. So when somebody comes with a mutation, we already know functionally whether or not in vitro this is associated with a phenotype. This gets very complicated very fast, and it's probably beyond this meeting. This is just what I said. Okay. We had a human genome project at the beginning of the century. I think it's time for the human variome project for the next half of the century. That's probably bigger than the genome project. Okay. Another problem that Jennifer also alluded to is in the dominant disease especially, we have a incomplete penetrance. Incomplete penetrance happens when there is a carrier of otherwise known disease-causing allele that is not sick, or partial disease expressivity, meaning it's not as sick as some other family members. And we have a very nice term for it, incomplete penetrance, but we don't understand it. Here, I'm just showing you the example of a made-up family, but everybody carries the allele, and you can see that the father and the daughter have fully clinical disease. There is a partial phenotype in a sister, whereas the mother, who's wild type, and the son, who is a carrier, are completely healthy. So this is a classic pedigree of incompletely penetrant family. If this was the gene first to be looked at, any geneticist would look and say, this is not the disease-causing gene, right? Because it just makes no sense. Okay. So what I had to do recently is go back to textbooks and relearn that actually not all genes are expressed from both alleles. And of course, we know this from genomic imprinting, where you can be just expressing maternal or paternal allele throughout your organism. We know, of course, of X chromosome inactivation. Individuals with 2 X chromosomes, one gets randomly inactivated. We know about transcriptional bursting, when one allele gets preferentially transcribed really fast, and that's been described by actually Bill Paul here at NIH in the 80s. And what I'm going to tell you a little bit about today is the concept of monoallelic expression, which is different from transcriptional burst because it's actually mitotically stable and it's somatic. So you can think about it as transcriptional mosaicism. So you're heterozygous everywhere, but let's say only your T cells become transcriptionally homozygous, and that's actually pretty stable. So that suggests that if we think about incomplete penetrance, canonically we would see somebody with partial disease expressivity, no disease, or fully penetrant disease as equally expressing maternal and paternal alleles. But if we twist this thinking a little bit, we can think about somebody who's healthy that's predominantly expressing the wild type allele. Somebody who's a little sicker has more of the mutant allele expressed, and somebody who's very sick, despite equal genomic heterozygosity, is actually expressing mostly the mutant allele. Okay, so how did we come about this? We identified a patient with a mutation in JAK1. This was a de novo mutation, and it was a mosaic mutation at DNA level. So what we did is we did single cell RNA sequencing because within the individual, we had both wild type cells and cells that carried the mutation. And here is the result, if you can look at there. The wild type allele should be expressed in every cell because it's either wild type or a heterozygous cell, and 50% of cells should have it. What was shocking to us is that the other 50% of cells should be double positive for the mutant and the wild type. And what we ended up seeing is mostly the mutant allele expressed, which didn't make any sense because it's, you know, against what we were taught in very basic human genetics, right? Okay, so we went on a quest for about 5 years and looked at, formed first hypothesis that monoallelic expression contributes to phenotypic variability or incomplete penetrance in inborn errors of immunity. And we tested this in 10 healthy New Yorkers. And let me orient you around this graph. On the top is a donor. There is donor A, B, C, D, and F. And columns are number of clones of T cells that we expanded from a single cell to 200,000 cells. If you look at the color of the circle, the middle is yellowish and light blue. That means it's biallelically expressed. If it's completely blue, it means it's an alternate allele. If it's completely red, it means it's a reference allele. If you go to donor C, you can see how about half the clones are monoallelically expressing a silent SNP that this healthy individual has. If you go to donor F and PLC gamma 2, you can see how one clone is completely maternal, one is completely paternal, and the rest of the clones are biallelic. So this suggested to us that this transcriptional mosaicism, or the ability to somatically commit to the expression more so of one allele, is actually a common trait of human biology. And long story short, we know now that about 5% of genes can undergo monoallelic expression. In each individual that we tested, N is small, but we've never detected more than 0.5% of genes undergoing monoallelic expression, at least in the immune system. Does this influence incomplete penetrance? Here is a family where the grandfather, mother, and the granddaughter all carry a variation. The mother is the only one that's sick. Again, classic genetics would say this is not the allele that can cause disease because you have two healthy individuals, older, younger, they're not presenting with a phenotype. This is an interesting case because this is a female from the UK. She was healthy until toxoplasmosis at the age of 28. Following that, she had recurrent infections, CVID, hyper-IgM, splenomegaly, severe pancytopenia, thrombocytopenia requiring transfusion. It's very clear that toxoplasmosis doesn't cause this, but it correlated with it. So what we did here is with biochemical testing, something that we can send to Iman's group to evaluate with, and as you can see here, compared to the wild type, it's a very clear biochemical gain of function. There is no question about it. But the grandfather and the granddaughter also carry the allele with this very clear gain of function, and they're not ill. If you look at then what we did, the next thing is we sorted B cells, monocytes, NK cells, and T cells. The graph, the y-axis, is JAK1 mutant allele expression. If somebody's a heterozygous, all these dots should be around 50. And what we saw in the mother and the daughter, that predominantly mutant allele was suppressed, with the exception of maternal T cells, where this was flipped.
You have 2 minutes.
I'll be done in 1. So this really suggested to us that monoallelic expression can correlate with disease. We did the same thing with PLC gamma 2, discordant phenotype between two cousins. One's B cells work fine, and the other ones they don't produce IgG. We sorted these cells, performed calcium flux, and here are the latest set of results. Wild type is on the x-axis, mutant is on the y-axis. Wild type individual expresses only the wild type allele, whether cells are fluxing or not. Individual who makes normal IgG by B cells, cells are fluxing or not, they are biallelically expressed. And the individual who doesn't make IgG, you can see the cells that are not fluxing are solely expressing the mutant allele. And I'll skip this one, but long story short, in the father who has the variation, we detect only the wild type allele. The kid that's sick, we in classic monocytes and B cells, they detect biallelic expression. In conclusion, genetic analysis can guide therapy in primary care setting. We have that. I think we're heavily underutilizing it. We need human variome projects for the next century to come. We need to understand genetic penetrance and how genotype, and now a word we made up, transcriptotype, may differ because it may influence the disease penetrance. And a bunch of people who did this work and collaborators within and outside without whom this wouldn't be possible. Thank you.
All right. And great, we're going to hold questions to the end. So our next speaker is Scott Canna, Primary Immune Disorders and Specialized Infectious Testing.
I echoed Dushan. I turned it off. I echo Dushan's enthusiasm for this meeting. It's an honor to be here. I love how much time there is for argumentation / Q&A. Some disclosures that don't actually have any money behind them. You just heard from Dushan about, if you find certain infections or phenotypes, these are the immunodeficiencies to look for. And I was asked to go the opposite direction. If you have a patient with a known inborn error of immunity, or if I like primary immune disorder, because as you mentioned, not all of these are inborn, but it's going to take a long time to turn around. What kind of infections should we look for? And so my next disclosure is, who is this guy? I'm a peds rheumatologist. My research focus is mostly on inflammation that's not infectious. And so either I'm here because you couldn't find anyone else, or because I represent this immune dysregulation program, which very briefly is arose out of sort of clinical necessity because of too many argumentations over bedsides about what flavor of HLH someone might have. It's an inpatient and outpatient program. It almost functions like its own service. It has all these different specialists that you can see on the right, and is now sort of a institutional program of record. We're out of our sort of beta period, and serving as, I think, a little bit of a template for these around the country and how they might differ at different institutions. And so I drew upon my colleagues within what we call DIRT, Dysregulated Immunity Response Team, from the world of immunodeficiency and infectious disease. So thanks to them. Further biases are, we've talked about immunodeficiency as if it's its own thing. And I'm personally of the bias that there's really no such thing as immune dysregulation as a category. It's all used or heard of. I'm sorry you can't see, but like BioFire or Carious testing is metagenomic, and these can be used on liquid samples like plasma or CSF, or increasingly even from tissue samples if you know beforehand that that is what you want to send. So I thought I'd illustrate the sort of idea of going from monogenic disease to infection with a case. So this is a 5-year-old with cartilage hair hypoplasia, which thank you, Dr. Puck, for introducing the importance of that gene as a potential inverter of immunity. And the child had that rash for quite a while, which was obviously very disturbing. And so has this defect in what is actually a long non-coding RNA that's important for this RNase, but doesn't really tell us a whole lot about how you get the phenotype. And so these patients have both a sort of syndromic appearance as well as a really heterogeneous and potentially severe immunologic phenotype that can include autoimmunity and immunodeficiency and something that I call sort of dyshematopoiesis, including bone marrow failure. But most patients don't have all of this. But this particular patient did have low T cell numbers and hypogammaglobulinemia, and was on replacement and had, to all appearances, seemed to do pretty well. But this type of a rash had really been there somewhat continuously through 18 months, or from 18 months. And so, of course, it was biopsied, and it showed sort of unremarkable non-caseating granulomas that had been cultured, that had special stains, that did not respond to empiric antimicrobacterial antibiotics. And so what the heck is going on? Is this all just immune dysregulation? And it turns out that they went back and they found this is vaccine strain rubella. And this is all Kate Sullivan's story. So you can't read it, but it's down there. So if you take tissue blocks from these biopsies and you stain for rubella proteins, you can find either within simple or multifocal granulomas that this is a patient that has vaccine rubella reactivation that has caused this really hard to diagnose granulomatous skin disease. And it turns out that about 70% of primary immunodeficient patients with granulomatous skin disease, they find rubella. This is Kate Sullivan has grabbed slides from colleagues through USAIDnet and sent them off for special testing. It's actually done at the CDC. And so this is a breakdown of the kinds of immunodeficiencies associated with this sort of reactivation of rubella. Most of them had sort of combined immunodeficiencies, but you can see a decent number of just regular CVID. And so you can test for this. Now, if you have a suspicion for rubella, you might actually do rubella-specific testing, either with a targeted qPCR or through metagenomics. If you're looking through the retrospectoscope like they were, you can take FFP slides and through a fairly onerous process, send them to the CDC for testing. So that's just sort of an illustrative example of if you know that the patient has a T cell immunodeficiency and has a granulomatous skin disease, rather than going through that long diagnostic odyssey, you might seek out a specific infection like rubella. And so that is just emblematic, and I'll go through a couple of the infections that we tend to think of as being sort of hallmark of certain inborn errors of immunity. That's the list of EBV susceptibility genes. Some of them have their own sort of names of the syndromes. And testing for EBV is both serologic and through sort of targeted PCR. The one reason I bring it up here is that a number of institutions, including our own, actually do a little bit of a specialized test where we will sort peripheral blood cells into T cells, B cells, and NK cells, and do a qPCR from those, because these inborn errors of immunity, when they have severe EBV disease, it's still in the B cell compartment largely, but in EBV HLH, such as there is a garden variety, which is much more common in Asia for reasons that are not super well understood, it happens that actually infect T cells and NK cells. And so, you know, the nature of the immune dysregulation might determine where that EBV is actually a reservoir. And that is not a test that is sort of widely available. Susceptibility to HPV is a common feature of a number of severe T cell defects, again, sort of listed in the table there. Clinically, it's not quite ready for prime time. We don't actually test for HPV. We just assume based sort of phenotypically that things look like HPV, and that's what it is. But it does turn out that there are specific types of HPV that are maybe more specific to certain immunodeficiencies. You know, the gamma type being maybe more common in CXCR4 disease or WHIM syndrome, and maybe also in some other T cell defects like DOC8 or GATA2, which is not a T cell defect, I recognize. But the technology to do this is largely done through something called rolling circle PCR amplification, which a number of places are working on rolling out, but is not clinically available. Mucocutaneous candidiasis, there's a number of genes associated with loss of activity of IL-17 pathway. And, you know, it usually goes the other way. If you have a patient with mucocutaneous candidiasis, you're looking for the gene. But in these cases, if you happen to know that these patients have the gene, you might be looking for it. Finding the candidiasis in mucocutaneous areas is not always so hard. The hard to grow strains are actually more associated with invasive candidiasis, which is actually not associated with these defects, but more neutrophil defects. And then finally, it is insane for me to be presenting on Mendelian susceptibility to mycobacterial disease at this meeting with some of the folks who are here, but nevertheless, you asked me to do it. So here I am, and that is the list. And you know, mycobacterial detection is really hard. These are incredibly fastidious, slow-growing organisms that are not always tuberculosis. And so the current testing includes sort of a all of the above approach, and we still are sometimes worried that we're missing an infectious cause of a very low abundance of mycobacterium in a patient that maybe they don't have an incredibly severe susceptibility, but if they're just not fully clearing it, that could result in a granulomatous disease. Maybe that'll be the next rubella story. And then finally, enterovirus is not one virus, it's a family of viruses that particularly associated with agamma-globulinemia, which is BTK deficiency. A substantial number of them can develop invasive enteroviral disease, especially in the CNS. And so there's a number of ways of detecting that, both by qPCR, if you know specifically what strains you're looking for. But if you have a patient that has a sort of the right encephalopathy phenotype within, you know, one of these inborn errors of immunity, you might look broadly for enterovirus within these tissues. And so then finally, what you might consider inborn errors of immunity that are not always what we consider immunologic diseases, like sickle cell, like cystic fibrosis. You know, at least cystic fibrosis patients, a lot of the problems they deal with are largely infectious. And so I would consider it a sort of barrier or a structural immunodeficiency, at least in 2024. There's a long list of genes associated with both CF, largely because they have resistant bacteria that grow up because they get so many rounds of antibiotics. They also get non-tuberculous mycobacterium, but also CGD, chronic granulomatous disease. There's a number of hard to grow or anaerobic or fastidious bacteria that unless you know to look for them, you might very well miss them. And so I'm trying to move quickly. One of the slides didn't make it through, which was basically, so I'll just summarize it. There's not a lot of primary evidence for the use of these metagenomic or large multi-pathogen panels for the diagnosis of infectious disease within primary immunodeficiency. There's a bit of literature in specific syndromes like sepsis, and there's a bit more in HIV, which is maybe the closest sort of surrogate we have. And that data is basically shows that it changed management in a substantial number of the cases that are reported. So I bounced that just last night off of our special immunology clinic at CHOP, which is the clinic that manages children with HIV. And I said, well, are you starting to use metagenomic sequencing? And the answer is no. Some of the barriers to that are surely cost, both for inpatients and outpatients. There's a lot of gatekeeping to the access for those tests. But they also pointed out that, you know, the infections that the HIV patients get are the same infections that they were getting 10, 20 years ago that we have good sort of molecular tests for. And so they haven't felt like there has been an unmet need yet. But the corollary to that is they may not know what they're missing, and they might have patients that have indolent infections that they just haven't found because they haven't looked for everything. So in summary, you know, there are a few situations where individual inborn errors of immunity would cause you to look for specific testing. I think rubella is maybe the best example of that, but I illustrated a few others. But really, the sense that I have from working with our immunologists and our infectious disease and our multidisciplinary group is that it's really classes of infection. If you have a immunodeficiency where there's substantial T cell defect, there's sort of a panel of T cell deficiency pathogens, and you're looking for all of them, and it's not always, you know, a one-for-one kind of thing. It's really, you know, you're looking fairly broadly for susceptibility to those kinds of infections. So, you know, basically we try to aggressively seek out getting good samples and culturing broadly, and especially if you're worried about mycobacterial disease, culturing for a long time. And I think we're still figuring out what the role for metagenomic sequencing is. There's been substantial investments, both commercially and at large academic institutions, in making that available. We increasingly make use of it, but and the data, although somewhat biased because most of it is funded by companies that are selling these tests, show that it changes management in a substantial number of patients, but there's really not a pipeline for access to that kind of sort of expanded sequencing of pathogens, whether it's for inborn errors of immunity or whether it's for just garden variety patients or HIV patients. So with that, I want to thank the broad team of people who are tired of me bothering them about how we would sort of get to this question. And I'm happy to talk more about this program, which obviously I feel very strongly about, and plug that we have a symposium that's free. Folks are welcome to join. Thanks very much.
Thank you. Moving on, we're going to have Stephen Kingsmore from Rady speak about NICU and PICU sequencing and variants predisposing to sepsis.
Thanks, Terry. Great to be here. If you had met me in the late 80s or early 90s, I would have described myself as a cellular immunologist. My first R01 was from NIAID, and my first Nature paper was positionally cloning the Chiari-Kagashi gene. Bet a lot of you didn't know that. All right, so what are the current opportunities to save lives and avoid suffering in infectious diseases by whole genome sequencing? So for those of you who are immunologists and not genomicists, you might not be familiar with this literature, but there is now strong evidence that a genome sequence can save lives and avoid suffering in children with a very broad range of presentations. This is the literature, the evidence base as of last year. And the statistics speak for themselves. In fact, there has never been an intervention quite like the genome in critical illness. I really said that, and I believe it. When you dig into this, you'll find that there's not a single cohort study nor a single clinical trial which has looked at genome sequencing in infectious disease. It's entirely left out. So what is the evidence? Might there be any evidence that a genome could do the same thing in infectious disease? So 3 cases. As I said, there's no cohort studies, so I just went back through our records and picked out 3 informative clinical studies. And the first one gets to, should recurrent infection be a new indication for rapid diagnostic genome sequencing? So let's just run through this together. Baby girl is admitted to our intensive care unit for prematurity. She's small for gestational age, and she has multiple congenital anomalies. She's dysmorphic, but genetics can't come up with a unifying diagnosis. She is in hospital for a long time. She has surgical repair of her congenital anomalies, and she has, during that hospitalization, recurrent respiratory infections, both gram-positive and negative. And these are very severe, as evidenced by sequentially requiring being put on a ventilator, on ECMO, for cardiac and respiratory support. The family are asked on several occasions about palliative care being an option for this baby, but there was no etiologic diagnosis, and they held out hope that she would grow out of this. So finally, at 7 months of age, a genome was ordered. At that time, the baby was in another of these septic shock events and required both inotropes and ventilatory support. The diagnosis was Coffin-Cyrus syndrome, and this is indicative of a broad set of genetic diseases that are not typically regarded as being immune deficiencies, but if you do a search of OMIM under recurrent infection, you'll find there's 884 such diseases. This is one of those. That the diagnosis was made, and she succumbed later that day. So rapid genome sequencing at birth would likely have allowed the family to have made a very different decision regarding their baby child. So this study would suggest, yes, we are missing out by not having recurrent infection being an indication for rapid genome sequencing. Okay, second study, or second case. This is a little baby boy who's admitted at 19 months of age to the PICU with septic shock, and the pictures show purpura fulminans, if you are not familiar with that. I'd never seen it before. His twin brother, and he was a monozygotic twin, so twin brother had died 1 day earlier at an outside hospital. Our hospital's located 11 miles from the border with Mexico. We have a lot of cases like this of families who try to get across the border to get admitted to our hospital. In this case, the twin brother had died on the other side of the border. The story was very similar in that we have a history of recurrent respiratory infections requiring courses of antibiotics. I don't have time to go through those findings, but for those of you who are immunologists, I know that you're just having a mental high there, and already you're thinking about what's the differential here. So let's get to it. Indeed, a genome is ordered, and it shows that he has X-linked A-gamma globulinemia, and most likely so did his monozygotic twin, explaining both of their presentations. But really interestingly, one thing we don't normally think about with a rapid diagnostic genome is that we get whatever else is in blood. When we purify blood for rapid genome sequencing, we don't like wash the cells, spin them down, stuff like that. We just take an EDTA blood tube or a blood spot and turn that into DNA and sequence it. And so here's something interesting. He was actually culture negative, if I go back, upon admission. Now, he was presumptively treated for Pseudomonas because of that skin rash, but he was culture negative. And in fact, what we found by aligning the reads that did not align to the human genome, we were able to show that he had Pseudomonas. And you can see the distribution of alignment of those reads to the genome in the graph on the right. He actually had a good outcome. Horrible hospital course, required debridement of those lesions followed by skin grafts, but he did well on IVIG and aggressive anti-pseudomonal antibiotics. But again, a rapid diagnostic genome done earlier in the clinical course could have avoided this hospitalization and the death of his twin. Okay, third case, even more interesting. We got it into our minds. Now, this is kind of before Carius came along and metagenomic analysis of cell-free DNA became something that's now evolving a standard of care for inpatient infectious disease workup. And here we just said, hey, let's take a look at unaligned reads in general. Let's align them across a host of pathogen genomes and let's see what we find. And one of the findings that really jumped out to us was cytomegalovirus. And believe it or not, about 4% of 669 critically ill children who received rapid diagnostic genome sequencing had reads that were 100% aligned to cytomegalovirus, and the Q scores were such that, you know, they were high quality alignments. And for a subset of those newborns, there was sufficient clinical information to show that they were symptomatic for cytomegalovirus infection. And for some, we were able to show that there was a very nice correlation coefficient between the results that we found and qPCR. So this is really an unexplored area. We don't know the limits of this. So I've shown you a bacterial genome. I've shown you here a DNA viral genome. We have no idea how good a host genome is at metagenomic analysis. Okay, moving on then. Terry asked me to be very, very implementation centric, and sometimes I listen to Terry. Make the most of it, girl. The policies, there are now this tapestry of policies, particularly for Medicaid across the country, related to rapid diagnostic genome sequencing. Each state that's colored here has its own policy, and none of them are common to two. And then on top of that, Anthem Blue Cross Blue Shield has policies which cover most states now. And again, their policies are different. These policies are complex, but the point I wanted to make was 2 points. One, it took cost effectiveness studies to actually see that happen. Showing clinical utility in and of itself, that second slide I showed you with 44 studies did not get the job done. It was real world evidence of cost effectiveness and actually net cost savings that drove Medicaid policy directors to write policies for their states. Not all of those policies are effective. Some of them are wholly ineffective in as much as they provide no payment. Very interestingly and topically, and you're all going to love this, it is Republican states that are reimbursing rapid diagnostic genome sequencing, and Democratic-led legislatures are saying, we're not going to cover the cost of this test. But nevertheless, it's a policy. I won't go into that any further. Okay, so what might this clinical research and implementation study look like that we might want to do in infectious disease? Well, here's a study we published last week in American Journal of Human Genetics, which unfortunately was too late to get considered for one of the top 10 papers of the year. Darn it. Probably wouldn't have got in anyway. Here what we did was we said,
Next year.
Roll babies. Was there a comment there?
Next year.
Okay, I'll take you to that on that. Here we said, let's only enroll babies for a rapid diagnostic genome who everybody says this kid is not genetic. They don't smell of genetics. they're not going to get a genome. So we enrolled 120 babies and we ran diagnostic genomes in the pink on the right. And lo and behold, 30% of them had genetic diseases. I took a look at this on the plane up here yesterday, which was a bumpy ride, believe me. Anybody give me an amen to that? Thank you, Weather. Three of that 30% diagnostic rate were actually recurrent infection or diseases that are associated with immune deficiency. Again, these are not classical immune deficiency diseases, but they fall into this second group of genetic disorders for which recurrent infection is a prominent clinical feature. And just to point out the middle blue area there, which is one of the ending points, which is that there are now multiple groups worldwide enrolling lots of babies for a genome-based newborn screen. And there are hundreds of primary immune deficiency genes on those lists, since they are remarkably, just about all of them have effective therapies. So they're high on the list for addition to newborn screening. So last slide, so here are some hypotheses I'd like to throw out that we might want to test with grants. Hypothesis one, that rapid diagnostic genome sequencing actually does have sufficient clinical utility in infectious disease to warrant coverage policies. And I've thrown out some ideas there: severe sepsis, septic shock, sepsis with opportunistic etiologic agents, sepsis without a normal response to an adequate antibiotic regimen, TRK positives in the newborn screening area. Great opportunity here to do what I asked provocatively of Jennifer. Could we do better than 3.5 months? And so if you marry the TRK positive with a rapid diagnostic genome, you can actually have that diagnosis in a week in terms of the molecular etiology of TRK positivity. Number two, just exploring this area of whether you get a free metagenome in a rapid diagnostic genome. You know, the alternative is to do something like a Carius test. There's no doubt that that's effective, but maybe we get a free analysis whenever we do a host genome. And very interesting to contrast and compare the two. This is just an unexplored area. Certainly for something like cytomegalovirus, we might also be wanting to add that to our newborn screening, as others have said here, and other congenital-ish infections that are probably massively underdiagnosed. As I mentioned in the newborn screening, we have 884 genetic diseases that we think we ought to be adding to genome-based newborn screening, since they are all associated with recurrent infections, and all of them have effective interventions. And then lastly, another unexplored area is getting outside of children's ICUs and getting outside of the NICU and saying, how applicable is this, say, in adult populations? Again, we have no idea. There's only one paper looking at the cardiac intensive care unit at rapid diagnostic genome sequencing. It found 20% positive findings in that population. It was only 50 subjects. And so it's insufficient really to understand what that might mean in terms of generalizability. Thank you.
Right, that was a really interesting set of presentations, so let's open for discussions. Down 80% down this table on the side, I can't read it.
Yeah, we may have to ask the people on this side of the table to kind of lean back a little bit so that our two moderators here can see. So there's a question way at the end near Bob.
Oh, just went down.
Oh.
Oh, so over here down.
I really enjoyed all the presentations. Stephen, just a question for you. I was very impressed with the slide that shows the effective and ineffective strategies. We're certainly very, very interested in the clinical implementation and sustained use of effective interventions. But to what extent are the ineffective interventions also continuing to be done? Because another part of our interest in de-implementation of low-value strategies.
Yeah, so that slide was referring to policies, and that's my subjective opinion of whether it's effective or ineffective. There isn't published data on this yet. Most of those policies are just kicking in. And so there isn't historical data to judge this. But at least in our state, California, which was the first to have a Medicaid policy, so great, but there is no attendant reimbursement for the hospital. So they say it should be covered under the DRG, but as we all know, a hospitalization is only 97% covered by the DRG under California Medicaid anyway, so adding cost is not going to help. What we found is that there is no uptake, right, outside of 2 children's hospitals. Really, California remains not a state that has converted to a NICU regime where rapid diagnostic genome sequencing is done in a substantial proportion. We don't know the ideal proportion yet. There are some innovator hospitals like ours, like Mayo, like Riley Children's, where up to 1/2 of NICU admissions are receiving a diagnostic genome. They are showing cost effectiveness even at that scale. And so that's another question. So it'll take some time to sort out what the policies ought to look like, but a big gaping gap, as I mentioned, is there's no mention of infectious disease as an indication.
We're going to turn to Mark now.
Thank you. This is a question also for Stephen. The CMV data is really extraordinary, and I was wondering, and this relates to one of your extended research questions, as to whether or not you looked at other torch infections, rubella, toxoplasmosis, which would seem to be the most likely ones based on our understanding of their teratogenicity to look for.
We've not done that, Mark. I think it would be an exceptionally interesting question. The data's out there. I mean, it would not be expensive to do this on a large scale against a much broader array of pathogens.
Okay, question over here. I believe it's Inka. Can't see your name tag.
Yes, thank you. So I think I have a question for Scott and both Stephen, or questions and comments. So some of the approaches that you showed, and I thought that was really, I didn't even think about, human whole genome sequencing and picking up pathogens in those samples as well. But I think, as was pointed out, the institutions that are doing the current sequencing, both for pathogens, metagenomic type of approaches, are mostly academic institutions and maybe some hospitals that have a research arm in it. So the big question is, going to the sequencing first approach and figuring out what those patients have in terms of infection. I think there are also, issues around regulatory aspects and getting reimbursements for approved diagnostic tests. And we have a session on this later on. But what are your thoughts on that aspect of it, like around the regulation of some of those technologies? Either one.
Are you talking about the patient sequencing or the metagenomic sequencing, or both?
Well, I get the metagenomic for pathogens, I assume, and then I guess the same if you're picking up with the human genomic sequencing, the pathogens as well. But let's start with the pathogen metagenomic type approaches.
Yeah, I mean, I think that some of it is, I guess it's sort of the snowball is still at the top of the hill, at least for my institution in metagenomic sequencing. It's a thing that we talk about at a much higher proportion than we actually send. And then I think our accumulation of anecdotes of times that it's been useful is not high enough yet that we're sort of pushing. But I think that traditionally that's kind of the trajectory that these kinds of things have taken, is that the academic centers find it to be clinically useful and then have the resources to sort of fight for getting it done, and then push to, both from a research standpoint and from a logistics advocacy standpoint, get it adopted more widely. So I'd be interested in others' sort of clinical experience with the utility of metagenomic sequencing in practice, because I was surprised that even reaching out beyond our sort of group where we only deal with the really puzzling cases, that it's not caught on in some of our special immunology clinic or our transplant clinic.
Down at the end of the table on our side.
Yeah, I had a follow-up to that question. One of the things that I've seen in some of the guidance is a pretty strong distinction. So I guess this is probably also for Stephen and Scott. A pretty strong distinction between the inpatient setting and the outpatient setting, particularly for metagenomic tests and when to do it, with the one exception being among folks who are immunocompromised, where there are tests that would typically only be done in an inpatient setting, maybe, but for somebody who's immunocompromised or has a particular genetic condition, that maybe those metagenomic tests might have more utility in the outpatient setting. And so I wonder if you all think about that or have an opinion on that.
Well, certainly the emergency department comes to mind immediately, right? And could we speed it up enough so that we could predicate admission decisions on results? Wouldn't that be wonderful?
Yeah, I mean, I think that's a factor of the, like I said, I was not giving a metagenomic sequencing talk. But a lot of the data that support the use of those is exactly about sort of turnaround time, and that you can get results much quicker, especially for organisms like fungi or mycobacteria that take a while to culture, that I don't think has really factored into decisions to say, yeah, we could recover that organism by culture, but there's both value and I guess cost savings to having that answer anywhere from a couple days to a week sooner.
This is work. Good morning. Liliana Brown from NIAID. We do pathogen genomics. Inca is in my group as well. I have a question for Dushan. I think I said it right. I don't know, but you had a wonderful presentation about RNA and how you can see different expression and how that can lead to different, disease conditions. So my question is more using RNA sequencing for diagnostics rather than relying on genomics. It would also address some of the data that I saw on Stephen's slide where 2 reads are going to help you diagnose a disease, which would be incredibly difficult, I think. But in RNA-seq, you might be able to see more of that pathogen signal. So just your comments on the reality.
Thank you for the question. And yes, that's how I was named Dushan, but I go by whatever is easiest. So in terms of host genome understanding of susceptibility to infectious diseases, I would go DNA first. And in the families where it doesn't make sense, or there is this form of incomplete penetrance, which is actually way more common than we like to admit, or we report in literature because it's mostly shrug, we don't know what to do. Absolutely, we need to go on the RNA level because we, and hopefully others as well, are documented that this discordance can happen. And because it is RNA, often that allows for sequencing of not just the host, but something else. So it can be a catch-all moment if designed appropriately. Of course, it gets very complicated very fast because we are talking about somatic commitment to transcriptional of one allele. Then that means it's in a specific lineage. Let's be simple and just talk about the immune system now, specific lineage of the immune system. So I think we are a few years away of being able to do this well on a single cell level, where we can then pseudobulk the parts and lineage trace based on mitochondrial DNA burden and infer what is this lineage that may be more committing to maternal or paternal. So I think it's still early days there, but absolutely going forward, I think we should be developing it.
We're going to have Jeff and then Jennifer.
Thank you. Stephen, always great to see your data. I'm wondering if you've had any, Stephen, if you've had any experience or success in working collaboratively with a payer or payers to design your clinical trials so that you agree on the endpoints, and if those endpoints are achieved, then a coverage decision is likely to be made in favor if the data is supportive of that? Or is that a completely naive way of thinking about it?
No, it's not at all, Jeff. You almost never say things that are naive. With Anthem Blue Cross Blue Shield in California in particular, we worked for about a year with their medical director, who was very open to suggestions in terms of what a policy might look like and where there were gaps in evidence. And having established that relationship, he then went to bat to get a national policy across all of the blues, and has continued to be in touch with us to look at modifications, knowing that a first policy document is not going to be perfection, and where should it be tightened up. So that was an exceptionally good interaction. More recently now, we found that the same is true of Medicaid medical directors, and that this is a group of people who really are looking for hard scientific evidence. You know, they really read the literature, they sweat the details, they produce these 100-page long synopses of all the published literature. And the onus is on us to publish well, and have well-designed studies that they can really draw conclusions from. And then to reach out to them. And we've seen this now happening state by state, where we now just routinely will reach out, you know, as legislators get interested in their state adopting something. We'll typically have an outreach, and you know, having government relations folks seems to be a good way to do this. They know the legislators, and are able to broker that discussion and then get passed along to Medicaid medical directors.
Jennifer?
Yeah, this may be for Scott or perhaps Stephen, but in patients with immune deficiencies, getting a tissue diagnosis is absolutely critical, and yet you know, we in actual clinical practice, we know people want to use the available sample or the and so we find a lot of the time people are getting a bronchial lavage aspirate when we know the yield from that is dismal compared to actual tissue. So I wonder, I mean, the NICU whole genome sequencing is always blood. Have you thought about other kinds of samples that might have better yield depending on a phenotype? And I think that is something that, especially for immune deficient patients, you should think about up front.
It is dogma that we feel like, yeah, that we get much better yield out of tissue. And I wonder if a lot of that is because our diagnostic methods are maybe more antibody based, because I was impressed as I was looking through some of the metagenomic literature that at least for in HIV, the diagnosis of PJP from BAL and paired blood was pretty much 100% concordance. And much, much better than silver stain or other, you know, more traditional PJP methods. So I think that, you know, using more sensitive techniques that are looking either for specific pathogens or more broadly at nucleic acids might overcome some of that dogma that, you know, that might be where the investment needs to be, because it's probably easier and cheaper than trying to revisit, you know, going back into tissue blocks and doing these things.
I think we need a lot more research in this area, Jennifer, honestly. We don't know the kinetics of DNA emia, really, right? And particularly cell-associated DNA emia. The beautiful thing is you're not relying on culture, so you can have dead bugs and you're still going to pick them up. That's awesome. We know that we have exquisite sensitivity. We can pick up a single molecule, you know, and we can have a bar in terms of its match level, so that we're certain, even with very few counts of positivity, that we're seeing something that's not an epiphenomenon. So I feel good about that, but honestly, we just need a bunch of research to understand what are the kinetics, what is the sensitivity, how does that vary based on the type of etiologic agent, and the site of infection, right? DNA EMEA is going to depend a lot on things like, are there barriers to, DNA getting into the bloodstream? It's a great question.
Next, we'll go to Ivan and then down to Bob Nussbaum.
All right, thanks. So my question, and maybe this is not just for the speakers, but for the people in the room. So I love this meeting because it gets together genomicists, immunologists, and infectious disease specialists. And so at our institution, the gatekeepers, as Scott mentioned, to getting cell-free metagenomic sequencing are the infectious disease specialists. And I guess the question is, what do we have to do, or what should be done to maybe get them to open the doors a little bit more for this kind of testing in immune deficiency patients? I mean, we have so many patients with immune deficiency, genetically diagnosed, who come in with like fevers, signs of infection. I mean, you swear there's an infection there somewhere. You can't figure out where it is. We want to send this test. They say no. And yet they're also, get this, the antibiotic stewards of the hospital. So, after 2 days, they're like, stop all antibiotics. I mean, perhaps the most frustrating disease, and Steve Holland will appreciate this, is CGD, where I mean, they're sick. They have all signs of infection. We cannot find the bug, and we don't know what to treat them with. So, like, what can be done, or what do we need to do? And like Scott alluded to, there are no studies, and this is what they tell us. There are no studies that show that doing this test has any kind of benefit in immune deficient patients. So maybe that's where to start.
I mean, that would make the absence of evidence argument, like, there are no studies. That doesn't mean that it's not true. It just means that, like, we have to do the studies. But, I have this argument with my ID people. I'm like, just because we haven't done the study to show that it's true doesn't mean that it's not true. And so,
So it might be helpful later on for y'all to define what kind of studies you're thinking of. Okay, I mean, because we're kind of talking about, well, we need more research on a variety of things, but if think about lunch, what kind of studies are you thinking?
You guys are just ganging up because Jeannie left the room. I know that. But I really want to echo that. I think as they say, the absence of evidence is not evidence of absence. I would just put one caution in there. The whole reason to do genetic diagnostics is because we think every disease is different. And I think it is true that every immunodeficiency is a little different. And there are the ability of cell-free DNA diagnostics to identify pathogens in chronic granulomatous disease is markedly inferior to the ability to do it in other, say, T-cell defects for a whole variety of reasons. And we need to generate that database, and it is in part incumbent on those of us who are doing it to put it together. It'd be great if the companies that have an interest in this created studies. And I'm not trying to defend my ID colleagues necessarily, but they are correct that we don't know. And that is really an urgent call for us to get the hell out there and find out. And whether we have to do it by pooling clinically relevant data or setting up prospective studies, I think is an issue for us to discuss.
I really like that study. I mean, I did the study, so of course I like it. Where we only enrolled babies who everybody said, no, that's not genetic, right? They're not going to get a genome. Doing something similar with metagenomics would seem to be the way to go, right? That's quite humbling for the gatekeepers, right? When they realize that their paradigm and their filtering tools are no better than tossing a coin, right? And it may or may not be true. It may be that they're great at calling who should get that test or shouldn't. It's a great way to do it. We have the same phenomenon with genetic testing, with host genomes, where the typical gatekeeper is the medical geneticist. And again, it's been sobering and life-changing for them when they realize, that was Coffin Cyrus. Why didn't I pick that up? I had 8 months to look at that baby, and I didn't think it was. So I think we're going up this learning curve, which comes from genome first, metagenome first, you know, where we are relearning as physicians the fact that clinical presentations are much broader than the textbooks tell us.
All right, so let me go to Bob Nussbaum, who's next question.
This is actually for Dushan. I wanted some more clarification. So first, it looks like some of the monoallelic expression looks like it's parental origin imprinting, but there are others where there are multiple siblings who inherit the same variant from the same parent, and yet they vary in their expression. And I'm wondering if he has any insight into what the mechanism is for this. And my second question is, for the ones that show parental origin effects, are those genes sitting in regions that have been suspected of being imprinted? And are there neighboring genes that also show monoallelic expression?
Right, so it appears that imprinting must be the whole organism, so parent of origin. Whereas in the random autosomal monoallelic expression, none of the genes lie in imprinted gene regions. They are, by definition, must happen somatically, otherwise they're not random autosomal molecularly called, because then they are parent of origin. And they are seemingly stable, and unlike imprinting, they're not 100% mother or father. They can be, the percentages that we came up with is above 80% single parent origin expression levels. We've done the calculations for 90, 95, but it's also true at 55, 65, and 75. And it's a completely arbitrary point. And where that cutoff should be used in terms of known genetic causes of diseases and penetrance is probably a sliding scale.
So if I could just follow up, so are there, do these represent then somatic errors of epigenomic effects around these genes? I mean,
Yes.
does the same gene always show some
No.
monoallelic effects?
No, it doesn't. So yes, these are de facto, by definition, somatic epigenetic errors, if you think, because they're not biallelic, which is what biology should be. What we do know is that maybe it's not an error, because the healthy New Yorkers that we tested, about 0.5% of testable genes in any individual can undergo it. Collectively, it's about 5%, and it's not always the same gene across, but there is a propensity of certain genes. But we're still not at a saturation to understand if it's a specific class. They're not organized genomically in any fashion that we or others can understand. So TBD.
Next to Inca. I'm sorry, I don't know your name, sir. Yes, thank you.
Thank you. So circling back to the question that was asked before about the clinical value and the gatekeeper question. So I mean, one thing that'll definitely help is when the test is not $2,000. So that's a barrier, which will come down in time. Yeah, sure. It's just commenting on the fact that one thing that'll help is when the metagenomic test doesn't cost $2,000. And that's something that should come down obviously over time. But one of the other challenges is that is demonstrating the incremental value. And there are some papers that have done that more so in an adult immunocompromised population. So maybe not necessarily the neonatal population with inborn errors of immunity, for example, but in transplant patients, severely immunocompromised patients, the data is there to demonstrate that there is incremental value. But it really, you have to end up restricting it to those who are really highly likely to have atypical organisms that you're unlikely to find using conventional methods. And it's also worth noting that it's not a good enough test to replace other testing. So for example, there is a partial overlap with what you get from culture. There are things that metagenomics misses that end up growing in culture and other methods. So it doesn't end up becoming a replacement, it's an add-on, and that just further challenges the economics of it.
Terry?
Yes, so I wondered if some of the genomicists who know this better than I in the room could speak to Dusan's question about a human variome project. There is such an animal from the Hugo, the Human Genome Organization, I think it is. And we also have the impact of genomic variation on function, the IGVF program that's addressing it. But Jonathan, maybe you could speak to that, or I saw you nodding, so, but it may be someone, or Bob, someone else.
I don't know the details other than to say, I think you're right, that there are already some nascent projects that could be sort of tapped into for that kind of idea. Yeah.
Yeah, it might be interesting, Dushan, to take a look at that project in particular, and also the IGVF, and see where you're seeing holes or gaps in what's available there.
I mean, I would put forth, what I heard you discuss was what I perceived as a, since you made it analogous to the Human Genome Project, hit the accelerator and try to be comprehensive. I don't believe, our IGVF program, impact genomic variation of function, I think we should still regard as a exploratory pilot, figure out what we're going to do phase. I don't think we believe it's ready to fully hit the accelerator. And we would fully hit the accelerator would be bigger than our institute could do. So I think we are, as an institute, that's very high on our priority list to try to lead an exploratory way, because we know that's an incredibly important area of genomics that fits very much in the wheelhouse of what our institute should be doing. But I will acknowledge it's not probably at the scope and scale of what your enthusiasm sort of implied. But it's something we're certainly hoping to help figure out the path forward, and also develop the technologies, the computational needs, and the strategies for eventually hitting the accelerator harder.
If I can just say something, is Liliana again. If there is going to be a project studying the variation of human genome, I would urge you to call it something else than a variome, because we already started the human virome project, and at least the word would be very close and confusing. But also just a note to mention, for example, for SARS-CoV-2 at NIAID, we've done a lot of work studying just within the receptor binding domain, the implications of variations in a very small region of that gene. And it's a lot of work from a lot of very many different groups. And that's in one single pathogen in a very small region of a protein. So it's not as easy as simply annotating a specific mutation. It takes a lot of work, and yeah. Anyway, I'll stop there.
Dan?
Yeah, so I'm online. I'm at home with coughing and sneezing. Terry thinks I have pertussis because she's seen a case, and I'm not going to argue with her. Maybe I do. Sorry, you don't want me there. We have spent a lot of time. We're spending a lot of time developing assays of variant effect for, dare I say it in this meeting, cardiovascular genes. And one of the things we're learning is that every assay assays a specific protein function. So when we start to look across literally tens of thousands of variants in our favorite genes, what we find is we can assay specific protein functions, but then we have to take one step back and say, well, did we capture everything that's pathogenic or benign? And that's going to be an Achilles heel to the variome project, or whatever it is you want to call it. The other two things to say about that approach are that eventually we'll have enough information that we should be able to use AI approaches to infer the rest of function. And it's the marriage of AI, structural biology, and actual deep function at the level of thousands or tens of thousands of variants is going to be the next big frontier for this. And I'm glad to hear Eric's enthusiasm. We're equally enthusiastic.
Bob? Is our next to Bob at the end of our table? Sorry.
Yeah, I have a question, I think for Dusan, regarding the monoallelic gene expression. Did you look at differences in DNA methylation that were allele specific, and did that play a role at all?
We did. So DNA methylation does play a role, as does H3K27 trimethylation. And we've also looked at H3K4 methylation. And the first two play a role, but it's actually more complicated than I thought, isn't it always? Even in the clones from the same individual that have committed to alternate or reference allele. In some clones, when we downregulate DNA methylation, we can actually now switch back to biallelic expression, right? So that's evidence that DNA methylation plays a role. But when we do the same thing in a different clone from the same individual that's at the alternate allele, nothing happens. So it seems like these marks are necessary but not sufficient, and sometimes they suffice. So it appears to be a fairly complex epigenetic regulation and decently leaky, because the level of monoallelic expression, unlike in imprinting where DNA methylation just shuts it off completely, right? So you just don't have it. Here, it can really be 50 shades of gray from 50 to 100.
So I'm wondering if we could ask the three speakers from this session. We had asked each of you to focus on sort of one thing, whether it was pathogen first, phenotype first, or sequence first. Some of you followed instructions, some of you didn't. But no, at any rate, and obviously, the answer is yes, we want to do them all. But are there ways of identifying, when is it the pathogen that really points you toward immune deficiency? When is it the phenotype, and when is it the sequence? So maybe you could comment on those, please.
Can I start? So I still consider myself relatively young, which is wrong, but I host that belief. Thank you for that. But part of that is because I'm very privileged to sort of be surrounded by the really paramount leaders of multiple fields who, like Dr. Puck, who have been seeing patients as the whole field has evolved. And the refrain that I hear from a number of them is that despite the exponential increase, we're still lousy at guessing what gene or even genes it's going to be. Every time. Thank you. So, and it's not because, these are some of the most brilliant people I've ever worked with, right? So, and they're paying attention the whole way. And so I think that, part of this is just that the phenotypic expression of inborn errors of immunity is very incompletely described, in part because that's work that no one wants to fund. It's not necessarily a hypothesis-based thing. It's, we have to build databases and populate them with phenotypes, and that's not it's hard work that costs a lot that nobody wants to fund. But I think part is just that these things are really pleomorphic, and so I think that favors, for me at least, a very genotype-first approach, because we have not iteratively gotten better at predicting what the genotype is going to be.
So I'll agree with Scott here. It's very hard for me to understand any reason that when a child is in NICU or PICU, doesn't that get whole exome or whole genome done. I think that should be a standard of practice at the price point that we have right now. It's just the first thing that gets done in parallel to everything else. And I even think bigger than that, but in these cases, and in adult setting, I would go as well, especially with the explosion of somatic variations that have adult onset as well. We absolutely should be doing that, because it's still the very minority of all of us who will ever end up in ICU of any sort, right? Which means something is very wrong with us.
So to me, genome first means that we are doing screening, and phenotype first means we're doing diagnostics. And I think we need both. I think we need a belt and braces approach. I think they both have different pretest probabilities of positives. And so the test formatting is completely different, as is the cost structure, the size of the population to be tested. I think there's a strong case to be made for both.
All right, I'm getting the nod from Terry that I think can we wrap the session, or are there final comments?
If there are other
or-
comments, I mean, we have till all the way till 12:35.
Can I ask a question? So we were just talking about how there's a big gulf between what the cost is for research sequencing versus, you know, the clinical test, right? So I just sent out a trio whole genome to be sequenced with the Broad, and it cost a little over $1,000 for all three. And that is continually coming down. Yet, you know, it's impossible to know what anything costs clinically because there's what's charged and what's paid, but it seems to be in the many thousands. And I understand that there's a lot that happens, you know, isolation of DNA and then the big bioinformatics that must happen. Although, to be honest, that looks like a lot of that is just sort of automated. And so I'm just wondering, what are the barriers to shrinking the cost of clinical sequencing when the cost of the actual sequencing part is so precipitously dropping?
Oh, I can go with that one because I live that nightmare day in, day out, every day. So we need automation, right? Clinical genome sequencing is incredibly manpower intensive. And we've talked about the VUS issue, and we're talking dozens of hours with incredibly highly paid people and 3 analysts to interpret a genome. And we're still only, you know, diving into the top of the surface. If you pay for $1,000 genome, you get $1,000 value. You pay for an $8,000 genome, you get $8,000 value in terms of the extent, the thoroughness of the interpretation. How we fix that, honestly, is automating interpretation. We're not that far off from it. There are now really good algorithms that are about 95% good, right? So as a first pass, if you want $1,000 genome, these tools are awesome. That's kind of how the Broad is doing it. So it will come down. Part of this is it's still an esoteric test, way underutilized. Enough on that. I could go on all afternoon.
If I could weigh in on that as well. A lot of this is the confusion, and not to say that the questioner had that confusion, because I think he clearly didn't, but we conflate cost and charge. And what it actually costs to do something and what we charge for that same thing has a lot of different components to it. And one of the things that we've encountered at the institutional level is that with send out laboratories, many times the institution that's sending out the test applies a multiplier for charge, which if it's a relatively low cost test is no big deal, but if it's a test that, you know, costs $1,000, then that adds up quickly. And that, of course, has to do with how we do insurance reimbursement and the fact that different insurers pay different amounts based on charge. So it gets incredibly complex to deal with the charge issue and what is relevant to the patient and ultimately the clinician who's going to have to deal with the patient. And those are not amenable to sort of generalizable solutions for the most part. And so that's more of a policy issue than it is, you know, something that's intrinsic to the actual testing process itself. And it's very frustrating.
I might also add to that the difference between inpatient versus outpatient testing and way those things get funded in healthcare. So if you're doing an inpatient genome, that hospital that's doing that test is probably going to pay directly to the sequencing vendor, the commercial vendor. And that's going to be a small drop in the bucket if you're a NICU patient and you're there for 3 or 4 months, right? And so from, you know, Stephen's, his group has really sort of pioneered this and shown cost of the genome, according to the cost of the NICU stay, it's a no-brainer. And most institutions can see that logic and start to think about how they can just pay for the genome because it's going to save them money. As an outpatient test, it's getting billed to insurance, and that insurance might be a private insurer that will or will not cover under certain circumstances that test, and it might be a you know, public, you know, Medicare where there's this patchwork of decision making. So it just gets really complex at the level of outpatient testing. Who's going to pay for it? I would also wager that the Broad Institute's probably getting a much different deal from the sequencing companies on reagents and machines than other groups do. So there's a lot of subsidy that's happening behind the scenes probably that also is affecting what's the apparent cost that you might see as the investigator.
Could I suggest, I know we're just before lunch, but we have two people at the end of the table who have a lot of experience in this, Bob and Jillian. Could we maybe take those two, Lucas and Jonathan, and then we'll stop? Okay.
I was just going to add an administrative detail to Mark's to call out that from a cost and pricing perspective, many of the prices for tests in this country are based on what CMS sets the rates for. And for exome, there is what I'm pretty convinced is a typo in the fee schedule that leads to the cost of an exome being $30,000 per the CMS rate, and specifically the comparator code. So if you do a trio, that is priced at $12,000 per comparator genome for parent. It's a little bit better for genome. It's about $15,000 is what our government has priced genome at. So I think that's also something that somebody could look at if they wanted to reduce the costs of exome and genome.
I just wanted to add one other area that adds significantly to the cost is the billing department. The billing department for example, at Invitae, I don't know the exact numbers, but it's comparable to the size of the entire variant interpretation group. In fact, even larger. And between having to do pre-auths and letters of medical necessity and appeals adds tremendously to the cost of trying to deliver these results.
Okay, we'll wind up. Great. Before we applaud all of our speakers and presenters, lunch is outside. It's a little bit sticky because people who are paid to attend the meeting, your lunch is already paid for. You should have gotten an email if you're not paid to attend the meeting that you could pay for lunch, and you had to do that in advance. So if you're not sure if you've paid for lunch or not, you probably shouldn't eat lunch, at least not here. But at any rate, perhaps Jenna Cohen can help you with that. We will be back here at have it right now 1:20 for our next session, and I think that will do it. So many thanks to all the speakers and presenters. So please, everyone, if you could take your seats, we're going to get started again. Okay, everyone, if you could take your seats, please. We are going to start. Please wind up your conversations and take your seats. Our next session is on viral infections and host genomics. Viral infections and host genomics, you don't want to miss this. There you go. All right, good.
Well, now everybody outside will think they're missing something.
That's
They're
right.
Excited.
That's right. So our moderators are Gail Jarvik and Casey Overby Taylor. So Gail and Casey, please take it away.
All right, well, we'll just jump right in with our first speaker, who's Andrea, and Tales from the Genome.
Thank you. Can you hear me? Okay. Thanks very much for this wonderful meeting and this invitation. So what I will tell you is a little bit of how we've been using genomic medicine to elucidate the host determinants of severe human papillomavirus diseases and try to infer novel therapeutic paradigms for management of those diseases. And I think everyone already mentioned some of those points very quickly. What I refer to with HPV disease is a pretty broad spectrum of clinical manifestation, usually starting with benign lesions involving any cutaneous or mucosal surface, usually skin warts or verrucae on the skins, or papilloma and condyloma when they're on the mucosal surfaces. If they persist, they can evolve with some different degree of dysplasia and eventually evolving frank squamous cell carcinoma. Despite pretty much everything we know about primary and secondary prevention and treatment, we still have that HPV diseases cancers are 5% of the whole number of cancers diagnosed worldwide and 30% of all the infections related cancers. And this is illustrating very quickly what is the natural history of an HPV infections. The incidental infection occurred through microabrasion in the epithelium, infect the basal layers. The infection can be cleared or can persist, evolve again in dysplasias, and cancers occur when virus is going to be integrating in the host genome, overexpress the virally encoded oncogenes and cause transformations. And the point of this slide is that there is essentially specific host functions that are going to control the fate of each of those transition. They can clear the initial infection, they can clear the dysplasia-induced regression of it, and I'm going to show you that can even clear cancers. And the work we do is essentially focusing on identifying those immunological checkpoints to correct them whenever we can or leverage them for therapeutic use. So this natural history at the individual level is reflected at the population level. So if there is a given population exposed to HPV, there's going to be a number of individuals that are going to have benign infections, dysplasia, and cancers. And of course, they're going to follow more or less the natural history throughout age. We're going to be focusing in the program we focus on a phenotype first approach in which patients with very severe disease with refractory warts or aggressive dysplasia or cancers in earlier age are sequenced and worked up immunologically. And this is the type of work that we and others have conducted and led to identify a number of specific functions associated to control of HPVs. Many are residing in intrinsic antiviral responses in the keratinocytes or DNA repair. Some have specific myeloid function and antigen presentation involved. And some others in effector function of T and NK cells. All of those somewhat pleasing in function of the adaptive or innate immune system. So pretty broad type of functions that are necessary to control those viruses. And I'm going to focus on three sort of paradigm of genomic medicine applied to HPV infections, IL-2RG, ZAP70, and IL-7. Thanks again for Jennifer for introducing this fascinating topic, but this is a patient, one of ours with a nasal squamous cell carcinoma associated to HPV-16, one of the more oncogenic one, small skin warts, sequenced and found to have an heterozygous mutation in IL-2RG, again, T gene for development of the immune system. I'm not going to enter into this for this audience, but a gene that is placed, as Jennifer was pointing out, on the X chromosome. This individual had an heterozygous variant. And the reason why that happened is because the variant was present in the germline. The CD34 stem cells had the mutated allele, but a reversion to the wild type allele occurred in a common lymphoid precursor, leading to complete reversion and normal T cell development, while the NK cells retained the mutated allele and they were defective in their cytotoxic activity. So this is a patient that, for the instability of the somatic reversion, as well as for the ongoing cancer and the defect in NK cells, was transplanted. He underwent complete regression and cure at this point of his nasal squamous cell carcinoma, as well as his skin manifestations. This is how his NK cells look like. We talked earlier this morning about functional validation. This is an assay that looked at how the cytotoxic function of NK looks like, was completely flat in these patients before transplant due to also poor expression of perforating granzyme in those NK. Upon transplantation, brisk recovery of NK cytotoxic function and granzyme and perforating expression, and resolution of that phenotypic defect, which resulted also in complete resolution of what was the imbalance in the skin microbiome of this individual. We alluded to the importance of metagenomic sequencing. This is how it looks, the skin distribution of viruses, bacteria in a healthy individual. 80% of your sequence are bacterial sequence in green. The darker green are actually viruses. And those are all HPV viruses in the different type of HPVs, alpha, beta, and gamma. This is how this patient looks like before transplant. The majority, or almost 90% of all his reads were viral reads of alpha and beta HPVs, and upon transplantation, this was restored in the normal bacterial viral distribution, as you can see. So a case that somewhat illustrates the importance of IL-2RG, but the importance of actually NK function to control HPV diseases. And another example of the HPV genomic medicine and the relevance of TCR signaling for controlling of these particular viruses offered by this other case in which ZAP70 variants occurred. This is a patient that was actually originally reported by Ivan a few years ago. This patient eventually, more recently, developed a very aggressive squamous cell carcinoma in her forehead, refractory to any surgical and medical intervention. She had some skin warts and some small oral condyloma, and she had a compounded heterozygous variant in ZAP70, a gene, an adapter that is absolutely important for transducing the signal of TCR and leading to T cell activation. This patient, cancer, that's, I'm not sure if you project well, but that's how the cancer looked like. Squamous cell carcinoma looks like a nest of cells that have clearly an invasive behavior approaching the muscular layers rather than staying on top of the skin, embolizing the vessels. Those nests that you see in the vessel should be empty and having red blood cells, not cancer cells, and perineural invasion. So this was a cancer with very high-risk metastatic spread behaviors. And this cancer, one sequence shows the presence of high level of transcription of all genes of HPV beta 19. This is notoriously not oncogenic virus, but under the lack of pressure from the immune system and generating fusion transcript at least 5 sites, so integrating in the host genome and overexpressing all the oncogenes of HPV, especially classic E6 and E7 that lead to P53 inactivation and squamous cell carcinoma formation. So this is, so far, is the first case of beta HPV integration and cancers caused by these virus. And this is again a patient that with bone marrow transplantation underwent complete resolution of her cancer and complete resolution of all the other skin manifestations. And I'll conclude with one more novel combined immunodeficiency. IL-7 deficiency has been recently reported by a group of investigators. There is so far 6 patients that have been diagnosed with this combined immunodeficiency. All of them have some level of, pretty aggressive HPV skin disease, evolving to squamous cell carcinoma and other more typical opportunistic infections. IL-7 is incredibly important for T cell development in the thymus, but is also necessary for the homeostatic maintenance in the blood and in the periphery of T cells. And these patients, this is a normal single cell RNA-seq of a healthy individual. This is how one of those patients looks like. Complete lack of CD4 T cell subset and mates, and the lack of lymphoid precursors in the lymphoid cells in the skin. So there is completely devoid of those resident memory T cells. This is a condition in which it's tempting to think to substitute what is lacking, and so adding IL-7 in culture lead to, of course, proliferation of the T cells and anti-apoptotic effect of the cells of these IL-7 deficient patient, as expected in a healthy individual. And so it was tempting to think to develop this in an in vivo approach, and this is the molecule we're currently clinically developing. This is a human recombinant IL-7 fused to an Fc neonatal receptor that maintain a long-acting function. This is a dose-escalating protocol in which actually the first patient of this protocol was dosed in the clinical center yesterday. So we hope that this will lead us to success in the treatment of those diseases. And I'm going to conclude with this sort of summary slide, emphasizing the point that genomic medicine is important to understand and solve that case, as we were saying before. But the thing that is most important for me is what it tells me in terms of managing that patient. It's not only understanding with gene, but it's what I have to do for management. And of course, bone marrow transplantation is good when we know what gene and we know what type of platform, and if we have a safe platform to transplant it. But there may be other interventions that are useful for those diseases. IL-7 could be one. Checkpoint inhibitors have been already approved for squamous cell carcinomas. Therapeutic vaccination for HPV is being developed. There is, of course, ways to potentiate the intrinsic antiviral effect of keratinocytes. TLR agonist and imiquimod is the classic, but there's many others that are developing. And it's important for us to know what a patient may have and what may benefit with. CXCR4 blockade for WHIMS was approved a few weeks ago, the first oral blockers that could be useful for things beyond CXCR4 gain of function and WHIMs, and of course, interventions that are emphasizing effective function of NK and T cells. So monalizumab, IL-15, adoptive T cell transfers. So that's the armamentarium that has been sort of built in the probably in the last, I would say in the last 5 years, thanks to genomic medicine approaches. And just my thanks to the clinical center and all the groups and collaborators that have helped us in this work.
Thank you. And we have Jean next.
Okay. Is this yours? Okay. Hi, everybody. Thank you so much for the invitation. And today I will tell you about the work that we have done to understand the role of CCR5 in the context of flavivirus infections. Okay, so CCR5 is a chemokine receptor, and its main role is in leukocyte migration. You can find this receptor expressed on a wide range of leukocytes, primarily a subset of macrophages and K cells and T cells. And leukocyte migration is initiated by binding of its cognate ligands, CCL3, CCL4, and CCL5. CCR5 deficiency is rather common. There is a 32 base pair deletion mutation that is found in the coding region that ultimately results in a truncated protein that basically never makes it to the cell surface. So people that have this variant don't have expression off of this allele. It's pretty common. The allele frequency is between 10 to 16% in individuals of European descent, and is found in homozygous frequency at about 1 to 2%. And we know the role of this in the context of HIV. HIV uses CCR5 as a co-receptor for entry, and the importance of this receptor in humans was demonstrated many decades ago by looking at individuals that are CCR5 deficient. These are people that have 2 copies of the Delta 32 mutation, the homozygotes. In a random blood donor group of people, the frequency is about 1%, but in individuals that are HIV exposed but remained uninfected, the frequency is increased to almost 5%. Conversely, when you look at people that are HIV positive, you basically can't find CCR5 Delta 32 homozygotes. So in the context, as we heard this morning in the introduction, sometimes these benefits come at the cost of making you more susceptible to something else. And that is actually the case for CCR5 Delta 32 homozygotes. So I'll tell you a little bit about what we learned about how being CCR5 deficient may make you at higher risk for flavivirus infections. So flaviviruses are a group of pathogens that you can find globally distributed on the top. And these are all viruses that are transmitted by vectors, mosquitoes and ticks. And as you can see, dengue virus, yellow fever, Zika virus, West Nile, Japanese encephalitis, and tick-borne encephalitis are distributed basically all around the globe. They cause millions of infections annually, lots of morbidity, lots of mortality, and there are no therapeutics for any of these at the moment. So I will tell you a little bit more about what we know about West Nile, because West Nile virus is the primary flavivirus that circulates in the United States. So when you look at the outcomes of infection, about 80% of individuals remain asymptomatic or subclinical, and about 20% will develop symptomatic disease. These symptoms can be really severe, oftentimes leading to hospitalization. So West Nile fever, as well as the more aggressive neuroinvasive disease, meningitis and encephalitis. And the mortality rate among the people that develop symptomatic disease is about 5%. So we start primarily because we found that CCR5 was really important in mice. And so when you infect mice with wild-type mice with West Nile, you see about a 30% mortality rate, but in CCR5 knockout mice, you see 100%. And so this gave us evidence and reason to want to go forward and to see if this was true also in human beings. And so what we did was we were able to get some samples from West Nile virus infected individuals that ended up on the extreme end of this spectrum, these people that had severe disease, symptomatic disease. And what we did was we looked for CCR5 delta 32 homozygosity and compared to random blood donors, where again, it's about 1%, we found that this was increased to almost 5% in individuals that were West Nile virus positive and had severe disease. We then wanted to look at the other end of the spectrum, people that were West Nile virus infected but remained subclinical or asymptomatic. And these samples are very hard to get because they do not come to clinical attention. And so we worked with the American Red Cross that screens for West Nile virus in their blood supply, and we were able to identify people that were asymptomatic or had very few symptoms. And what we found was measuring the number of symptoms, we found that people that were asymptomatic and reported 0 symptoms, basically the frequency of delta 32 homozygosity was 0, as opposed to the frequency found in people that reported 1 or more symptoms, which was elevated. And when we looked at the people that had symptoms in greater granularity, what you can see is that the frequency of delta 32 homozygosity increases with increasing reporting of symptoms. So what about people that have 1 copy of this mutant allele, CCR5 delta 32 heterozygotes? And so what we did was we also looked at this population, and what we found was that they basically phenocopy wild type. So they do not have a greater number of symptoms compared, like we observe if you have 2 copies of delta 32, and they look identical to wild types. And this has also been something that we showed in mice. We basically had wild type knockout or CCR5 heterozygous mice and found that basically if you have half the amount of CCR5 expression on your cells, that is enough to give you a wild type phenotype. So basically you need to really have no CCR5, at least that's what this data shows, to be at increased risk for flavivirus infection. So what about other flavivirus infections? So, we and others have looked both in mouse studies and in human studies and found some studies have found protective role for tick-borne encephalitis, but there are of note studies that have shown no association, and overwhelmingly the mouse data supports the role of CCR5 for a wide range of neurotropic flaviviruses. And I want to also note that the delta 32 allele frequency is very low in populations in where some of these flaviviruses circulate, such as in Asia, where JEV, Japanese encephalitis virus, circulates. And many of the other flaviviruses like dengue, yellow fever, they just circulate in places where we wouldn't be able to test for CCR5 delta 32. And so how does it actually work? Well, I think we know from our studies as well as many other studies that it is basically leukocyte trafficking, which is the function of this receptor. You can see here that if you don't have CCR5 in mice, you do have the inability to clear virus from the brain, which is where the virus ultimately ends up. And what you can see here, if you look at the brain sections, in red are circled leukocytic infiltrates. So you can see in the wild type mice, a lot of cells traffic into the brain, and these leukocytic infiltrates tend to co-localize with viral antigen. And in the absence of CCR5, you have just a lot fewer cells that get into the brain. So it seems like CCR5 deficiency just impairs the ability of effector cells to get to where they're needed to clear the virus. So as many of you may remember, in 2015 and 2016, a new flavivirus, not a new flavivirus, but a Zika virus, which is reemerged and caused a multi-country outbreak. And Zika virus is very much like all the other flaviviruses, but it has a couple of new features, which is that it has been shown to be sexually transmitted because you can find the virus in seminal fluid, but also the virus can transmit from mother to fetus. So if the infection happens while you're pregnant, the fetus is at risk. And these are 2 new features that are not known to happen with any of the other flaviviruses. And so we really are interested in this. The spectrum of outcomes for Zika virus and maternal infections is as great. So 70 to 80% of individuals or maternal infections result in no transmission of the virus to the fetus, but 20 to 30% do. And among these, you also see a wide range of outcomes, fetal loss, as well as congenital Zika syndrome, which includes microcephaly. But then you do see that most babies are born healthy, but as we've tracked some of these babies, it has been shown that a subset of these do have some neurocognitive abnormalities as we track them into early development. So this has sort of made us question or think about whether there's a role for CCR5 in Zika virus infection. And we have preliminary data that sort of suggests that that's true. And it's really, I'm not going to share that data today, but it's made us really think a little bit more broadly about what the role of CCR5 might be. Because we've always thought about this as really controlling cells migrating into the brain, which is protected by the blood-brain barrier. But it's possible that the role of CCR5 could be a little bit broader and control things, other barrier-protected tissues that are protected by the blood-testes barrier or by the placental barrier. And so things to consider, we don't know how much CCR5 is required to give you protection. We know that 50% or about half the expression is enough, but how much loss of CCR5 do you really need to make you more susceptible? We don't really know that. And this is important because CCR5 delta 32 isn't the only mutation. In CCR5, there are other mutations that can control the amount of CCR5 density on a cell. And of course, you may know that there is an FDA-approved CCR5 inhibitor called Maraviroc that is on the market, and as well as several in the pipeline, monoclonal antibodies, which are all thought or being considered for chronic use. So if CCR5 blockers can mimic a CCR5 delta 32 genetic mutation, how much would that put you at risk for increased susceptibility to some of these flaviviruses? And of course, we would love to be able to know if other flaviviruses are affected or the loss of CCR5 would put you at risk for other flaviviruses as well. And as I mentioned with Zika, it's made us really think a little bit broader about how CCR5 is protecting. CCR5, knowing your CCR5 delta 32 status, I think, is important, not only just for HIV, but also because it could put you at higher risk for neurotropic flaviviruses like West Nile and TB, where these genetic studies have been done, and could be broader than that and could include other flaviviruses as well. We just don't know. Of course, like I mentioned, that there might be a possibility that the loss of CCR5 could also put you at higher risk for poor pregnancy outcomes for flaviviruses that do impact pregnancy. And, you can learn your CCR5 delta 32 status by commercially available genetic testing. With that, I will end. I just want to also say one more thing is that, this is pretty important, particularly even if we live in a country where West Nile is the only flavivirus that circulates. This is also important for people that are living in endemic regions where flaviviruses are, which is basically all around the globe. And this is important for travelers as well. And individuals that are at increased age and are immunosuppressed to some degree, this might be very important to know. And with that, I will end by thanking the people in my lab, but also most of this work was done while I was here at the NIH and Phil Murphy's group. I'd also like to thank the American Red Cross and state departments of health that provided samples for us to be able to do these studies. Thank you so much.
Thank you. Moving on to Priya.
Okay. Can you hear me? So make sure. Okay, thanks. So I'm going to be talking to you today. First of all, thank you for including me. My career has always saddled somewhere between genetics, NHGRI, and NIAID. And so I'm going to speak to you today with both hats on, but talking to you more about population level work versus individual. And we're going to talk about it through this idea of viral infections, specifically hepatitis C. So just as a reminder, I don't think this group needs to hear it, but I think it's important to be reminded that we're not only interested in treating those at risk of disease, but also in preventing and disrupting transmission, because that's really important on the larger scale of infectious diseases. And the underlying presumption that a lot of people have, not necessarily people in this room, is that everyone who's exposed will be infected and everyone infected will have a similar outcome. And this is why so much of infectious diseases has centered on important population measures. That means clean water, sanitation, and vaccines, which are phenomenal. But I do think that things are a little bit different than what we perceive them to be. And that's that some never get infected despite being exposed, and those exposed don't always have the same outcome. So there's something that's driving that heterogeneity, and we believe, and that's why we're gathered here today, that it's likely genetic. So there are really good examples, I think, that we can think about. The best really is the Cutter incident with poliovirus, where you had activated polio vaccine that was accidentally administered instead of inactivated to school-aged children. And in these 200,000 children that were injected, 40,000 had infectious symptoms, 200 ended up with paralytic polio, and 10 died. So this is where you have the same inoculum, you have the same age child, and so you've restricted your variables, and yet you still see differences in what those outcomes are. And then we all have lived through a pandemic, and we know that in the early days before vaccine with the same variant, we saw multiple different outcomes in individuals from death to asymptomatic infection. And so what is driving that heterogeneity? Clearly factors in the pathogen themselves, and on a global scale, that's really important. So the dose that someone's getting, how that's transmitted to them, the variant, repeated exposures starting at a young age. And then host factors, obvious ones, age, sex, comorbidities, and co-infection. But I'm going to walk you through this example with hepatitis C virus, where an individual could be exposed to hepatitis C. Most commonly today, that would be through a bloodborne mechanism, usually injection drug use, but it could have also been from blood transfusions. And the virus would incubate for about 50 days. On average, you'd have some symptoms of malaise or jaundice or dark urine. This occurs in about 20 to 30% of individuals. And in the United States, that's about 19,000 new infections per year. And then that can lead to HCV infection. And then once you're infected with HCV, you actually have two paths, one of which is spontaneous resolution, and 15 to 20% of people will just spontaneously resolve their HCV infection. And the rest will go on to a chronic state of persistent infection, where factors like age, race, HIV co-infection play a role in that. And then if you are chronically or persistently infected, you can go on to cirrhosis and then hepatocellular carcinoma or liver failure. So we're most interested in that first level of who's resolving infection. So it's sort of taking the opposite, not who's most likely to have a severe infection, but why is it that some individuals naturally resolve that infection? So before I can get there, I just have to remind everyone, and Jennifer did it earlier in her talk, but there's one thing that makes infectious diseases just really different, and this is probably most aimed at the geneticists in the room, and that's that we have to deal with exposure, right? So it's not just about exposure in the cases where you presume there has been exposure, but also in the controls. So did everyone have the same opportunity to be infected and to have disease? And then how do you define that disease or infection, both in the cases and the controls? This seems really simple, but in the age of biobanks, it's really easy to just take access or get access to large numbers of controls and use them. And so I had a doctoral student who worked on this particular question of what would happen if you use population-based controls in this simulation instead of using well-characterized controls. So the yellow boxes are the well-characterized controls, and we'll start right at that very top box on the left, and the red is population-based controls. When the presumed prevalence of exposure is 100%, that means measles walked into this room and we all got exposed, there's no difference between the well-characterized controls and the population-based controls. But as you move down that panel on the left, it goes from 100% exposure to 75 to 50 to 25 to 5%. You get this inflation of your test statistic. That red box is moving to the right. Right? And so we're now seeing things that shouldn't exist. We start to find genes that really aren't real. We start to see things that are complete, basically, we start to see spurious associations. And so if we look to the right, the one on the left was 20,000 cases and 20,000 controls. The argument has always been just increase the number of controls you have and you will control for it. But actually it stays all the way. And this has implications for how we look at population level genetics and infectious disease work. And so that brings us back to hepatitis C and why it's a really good example of where you can clearly identify your cases and your controls. So in this case, our marker of exposure is HCV antibodies. That's detectable throughout your life. It doesn't distinguish between chronic or resolved infection. It just tells me that you've been exposed to it. And that's that blue line on the right in the graph. Versus acute and persistent infection, where you would see HCV RNA, at least in the first 6 months, and then after that it becomes undetectable unless you have persistent infection, in which case you persist, and that's that purple line that you see that exists overall. So, I'm sorry, that green line that exists overall. So now I can see that you could have spontaneous resolution. I know you've been exposed versus individuals who have persistent infection. And then we conducted a genome-wide association study. So this is a Manhattan plot looking at the minus log p-value on the left and then the chromosomes along the bottom. And we find, this really huge, large effect that is well known and well studied of lambda interferon. It's IFNL3 and IFNL4 on the end. HLA, also a clear contender to have a role in hepatitis C infections. And we identified a new gene, GPR158, in chromosome 10. So the key with this is a meta-analysis of of a large consortium of data that comes from around the world, acknowledging where HCV is actually occurring, both in Europe and the United States, and including African ancestry individuals. And we saw that when we stratified by individuals who were of African ancestry, that's the red, versus European ancestry, that's the blue, that when you increase the number of favorable alleles, that means your probability of spontaneously clearing infection, that your odds increased as appropriate, right? So these are independent effects, and it's dose response and related. And you see that in both ancestries that we look at. Now, individuals in the United States who are of Africa, who self-report as Black, are more likely to have chronic hepatitis C infection. And yet we're showing genetically that if you carry these alleles and the allele frequencies differ, you actually clear at a really high rate. We also looked at sex-specific genome-wide associations because we know that there are sex-specific differences. This is a Miami plot with the top being females and the bottom being males. And we still see that lambda association and we see that HLA association, but we actually see in males this association that does not exist in females with this specific gene. So something that is specific to males overall. And then we evaluated both the lambda region with a lot of fine mapping in an effort to try to find the causal alleles, as have many, including several folks here at the NIH, and the MHC region. And we had more success with the MHC region where using trans ancestry meta-analysis, we were able to localize this region and identify binding HCV peptides, and key amino acids that would play a role. So we took a really large region identified by GWAS and were able to identify specific peptides that were critical. And we believe this sets the stage for enhancement of HCV vaccine efforts. So in following Terry's instruction, outside of discovery, why do these findings matter? I think there's importance of the clinical translational aspect. Initially, post the original discovery of lambda interferon, it was immediately used in screening individuals because it predicted spontaneous resolution. And it was especially helpful when the therapy that was being administered was interferon therapy because you could see that those individuals were more likely to clear. It's also important for development or prevention, like I just mentioned with MHC. And then, of course, this helped to validate some of the findings we've seen that explain sex differences, ancestry, and other clinical and epi factors. But the real lessons, I think, for this group that we've learned from HCV is that population infectious diseases are just like any other complex disease. They're polygenic. There are multiple genes that underlie them. They're often independent. Our sample sizes in infectious diseases tend to be limited, but our effect sizes are really large, which means that we're really just finding the low-hanging fruit. And with more sample size and more information, we could find much more. Diverse populations matter. I didn't highlight it enough probably, but we only found a lot of what we did because of the cohorts that we utilized and because of the consortiums that we built that were international. And then, as I mentioned already, phenotype is key. This goes back to basic genetics, right? We need a strong and clear phenotype in both the cases and controls, or you can't find anything. And so you have to consider exposure. And this draws some questions on whether or not biobanks would be the most useful source of our time, or if it really needs to be existing or new cohorts. And so huge thanks to all of our team internationally that has worked on this. And I will pass this on.
Thank you. And Ryan?
Talofa lava, malole suifua laulele, ile mamalu o e ua poto mai, a fifio mai, matala mai a ao. I'd like to thank everybody for inviting us to give a talk for me to represent the research that we're doing in the Pacific with Pacific peoples. And if I could just reorient us for a minute to this part of the world and center an often marginalized people. Pacific Islanders are rarely represented in research and especially rarely in genetic research. And when they are represented, their data is usually aggregated with Asian individuals. Polynesia is located right here in the center, the Polynesian Triangle. There are 3.2 million Polynesians or individuals who identify as Polynesian living on the planet Earth. Many of those individuals live in the United States, New Zealand, or Australia. And then the largest of the peoples within the nation, the Polynesian nation, the Maori are the largest, followed by the Samoans at just over 500,000. Okay. My research team works with the communities in Samoa and American Samoa. Located right here, the Samoan Islands are divided into two nations. There's independent Samoa there on the left and the US territory of American Samoa on the right. Okay. I represent the Olanga study group. The purpose of our study group is to try to sustainably and substantially reduce disease burden in the Pacific Islands, and especially as a model for ways to improve health in lower and middle income settings. Now, our research team is particularly focused on NCDs, so it's a little unusual for me to be at this meeting, but we had data that was relevant in an immune deficiency that appeared among the people of Samoa. My group has multiple cohorts that we've recruited from the islands, but the one I want to talk about in particular is Soifua Manuia, the Good Health Study. We have just over 3,000 genotyped individuals that were recruited in 2010. Okay. And of those, 1,300 were whole genome sequenced as part of the TopMed project about 8 years ago. And so this means that we have a resource that isn't really available anywhere else on the planet. We are custodians of a data set that gives the allele frequencies for variants that are present among Samoan people and could be inferred to be present among other Polynesian peoples. And so there are no known individuals of Polynesian or Pacific ancestry represented in Nomad, despite its size. Okay. And so we often get a request from clinicians or from other researchers asking about allele frequencies so they can do investigations into variants of unknown significance that they see in patients or in research participants with Polynesian or Pacific ancestry. Okay. And so back when Twitter was a thing, you know, a genetic counselor just put this out on Twitter and someone connected her to me. And then within the afternoon, I was able to give her allele frequencies to help her with clinical interpretation. And then there's her update. Okay. We were contacted in 2021 by two clinicians in New Zealand who had a case of a child who died of severe viral disease shortly after vaccination with a live attenuated MMR vaccine. And it turns out that her brother had also died after vaccination a few years earlier. Subsequently, 4 additional kindreds were identified where a child had developed severe viral disease, 2 deaths, 2 that survived, and then 1 child who was tolerated the vaccination quite well, but much later in life, she developed severe viral infections. What's notable about all these kindreds from New Zealand is they're all of Pacific ancestry. The hypothesis was that these cases looked like they were individuals that had symptoms similar to known defects in the interferon-1 signaling pathway. And so the focus was to look at these 7 genes in whole exome sequencing. Now, there's a list of some of the very rare variants that are associated with immune disorders in this particular pathway to see if that's what's going on in these kindreds. So after whole genome sequencing, indeed, it was seen that the proband is homozygous for a truncation of the protein, and so was her brother, and her parents were both homozygous. And if you look across the rest of the kindreds, you see a similar pattern where homozygous individuals developed severe viral disease and their unaffected relatives were all homozygous. Okay. And I'm going to skip through these slides pretty quickly. I don't think this audience really needs to walk through the science behind establishing it, but it expected to produce a truncated protein. That's demonstrated here with a positive control. It's not expressed at the cell surface. In the patients, MX1 induction fails when exposing patient 3 fibroblasts to type 1 interferons. And so in control patients, we see a response, but in patient 3, we do not, nor in the positive control. And then MX1 induction can be rescued with vectors overexpressing the wild type, but not in vectors overexpressing the mutant type, nor in the positive control. So what's demonstrated here is homozygosity for IFNAR1 and a change from the glutamate to a early stop codon is likely causal for these severe viral disease cases. And that's when our group, my team in particular, was brought in, was what is the prevalence of this variant? And the allele frequencies, this particular variant is present in NOMAD. There's 1 allele out of 100 or 1.5 million alleles in version 4.1. But in Samoa, among 1,300 individuals, there were 32 alleles. We saw no homozygotes in our data set. And then in a data set from French Polynesia, about a 10-fold lower allele frequency was observed. Okay. What this means that among Samoans, we would anticipate that 1 in 41 individuals would be a heterozygous carrier, and 1 in 6,400 individuals would be homozygous and potentially affected. We look across the Pacific. The size of the circles represents the sample size from which the estimate was collected. So in Samoa, we see a allele frequency of about 1.25%, and not too dissimilar among the Cook Island Maori. And the allele is also observed in a few other samples from French Polynesia and Fiji, but then is not observed in a few other samples, but many of which are small, so they might not be large enough to observe something with an allele frequency quite this small. Okay. For comparison, these are the population prevalences in the United States of several conditions on the recommended uniform screening panel: sickle cell disease, cystic fibrosis, congenital adrenal hyperplasia, MCAD deficiency, PKU, and X-linked adrenoleukodystrophy. And so among Samoans, the population frequency of this allele is high enough that screening might be something that you'd consider for this population or this community. Okay. And estimates would say that this is about the number of homozygotes that we would anticipate just among Samoans. And there's about 500,000 Samoans on planet Earth, just among Samoans, with some ranges depending in certain communities on whether they report being Samoan or of multiple ethnicities. Okay. As a reminder, Samoa had a severe measles epidemic in 2019, which plays a role in our thinking about the phenotype. In October of 2019, the outbreak began. On November 20th, they began a nationwide vaccination campaign. They vaccinated 95% of the population in 7 weeks. At the time, this was considered unprecedented to be doing it at a national level. This was 2019. Such that the epidemic ended by late December. There were over 5,000 cases of measles and 83 deaths, most of whom were children under the age of 4. And there are circumstances why this particular epidemic occurred. But what was unusual was if this population has 3,400, wasn't a single adverse event recorded with the measles vaccine during the course of the campaign. So what does this say about the prevalence of this particular variant? It meant we were very cautious about, we were thinking about sort of publicizing this particular variant and publishing it in the literature. Okay, a couple of updates since the publication. Additional cases have been observed in New Zealand. Approximately half of those cases are now observed before MMR vaccination. So upon exposure to just wild yellow fever virus or chikungunya or RSV or influenza. When Pacific individual prevents with severe viral disease in New Zealand, such as severe bronchiolitis, they are tested for this variant. Originally, whole genome sequencing or targeting sequencing were the approach, but there is now a genetic test in New Zealand at $40 New Zealand dollars, about 23 US dollars. Okay, this is the ordering form. Okay, regarding screening for this population, so New Zealand's approach. Okay, and to remind, New Zealand has 1.2 million Polynesian individuals. About 1 in 5 New Zealanders is of Polynesian ancestry, either Maori or Pacific. And this is also one of the few nations where I think indigenous peoples have significant roles in governance, in particular health governance. So they were incorporated into decisions about screening. So should we do universal screening? And the consensus was no. Community screening of Pacific people, also no. There were concerns about driving vaccine hesitancy. If we started screening all people for this variant, would people stop getting the vaccination for their children out of concern that they might be carriers? Screening all respiratory admissions, no. But what the nation has implemented is testing of specific individuals with particular presentations. Okay. And to date, no cascade screening has been done of the family, so there's really been no opportunity to try and locate sort of unaffected siblings. Okay. And to take this outside of the Pacific, we can rotate the world a little bit more and look at the Arctic. In the same issue of the Journal of Experimental Medicine, a variant was identified among the Inuit with similar patterns in some families, and similarly demonstration that it was located in the interferon-1 signaling pathway. Also rare and nomad, 2 out of 1.6 million alleles, but an allele frequency of 3.4% in the Inuit and 2.4% among Greenlandic individuals. So here we have 2 examples of genetic variants that are not extraordinarily rare, existing in certain populations, which are generally underserved and marginalized. As a footnote, the original kindred the family has undergone in vitro fertilization, and through pre-implantation screening has recently had a baby boy who is wild type, and is quite healthy. The paper was published in 2022. I'd like to recognize in particular, we incorporated the feedback of 4 clinicians and advisors from Samoa, in particular in crafting the public health messaging associated around this paper. And then finally, I'd like to thank the Olonga Research Group for all their work and our funders. Thank you.
Thank you to all our speakers. We're open for questions, and I see one over there. Or is that a flag up? Can you read that? Yeah. Okay. That's fair.
Thanks. That was an extraordinary session. I want to just ask one quick question of Ryan. It was quite impressive that in the measles population vaccination campaign, you didn't see any cases of severe disease. You know, in BCG vaccination, it's very clear that if you give BCG on day 1 of life, the risk of getting severe disease is high. If you give BCG at 3 months of life, the risk of getting disease is really quite low. Do you think that had any role? That is, was in the aggressive campaign, were children getting vaccination at much later ages and therefore less likely to develop severe disease? Or was there a change in the lot of vaccine or something else?
I don't know, and I'm not a clinician. I don't know enough about the details relating to that, but I think one of our hypotheses was that measles killed the children who were susceptible. So the virus itself during the outbreak might have killed those children before they were subject to the vaccination campaign. So they might have been early deaths during the outbreak, in addition to adults who were affected. So that's sort of where we were coming from with it. But I don't know if enough information about whether or not lot number or anything like that has changed.
Let's see, is that George? Is that your?
Yeah, I just wanted to agree, really fascinating session, and a quick question for Priya and another for Ryan. Priya, when I listened to your session, I kept thinking of Jeffrey Rose and the classic paper on sick individuals and sick populations and the whole issue of resilience. And I didn't hear you mention anything about social and environmental influences when you have the same age group, race, gender, people in Chicago versus people in, say, New York City, where exposure may be the same, but you have completely different responses in terms of ability to clear. What do we know about that?
Yeah, so I think that ours was predominantly, so yes, perfect. I also think of Rose, and I think about the message that Jeffrey Rose makes about really focusing on populations, and genetics sort of turns that a little bit and says there can be personal aspects to this along with populations. In our case, we're looking at injection drug users, followed often in large cohorts that have similar protocols. So I think we may have eliminated some of the other aspects that you would have for other infectious diseases. But I do think those are especially important when you're comparing them. So we don't see big differences from that social environmental side in this particular scenario.
Maybe a very quick one for Ryan. The responses you showed to the questions about sequencing and those no responses, were they the responses of the community members or those of you experts working with them? I mean, I wasn't sure.
Yeah, I believe those are the decision that He Watu Ora, the New Zealand Health Authority, made about whether to implement community screening, universal screening, community screening. So those were the no's and the yeses was their approach that the public health officials in New Zealand chose to take.
By the way, Eric Green is really supporting a meaningful community engagement approach to genomics and genomics medicine. We haven't started the effort yet. I'm sure Terry will be telling you all about it once we get started, but we would be really delighted to hear the community members' perspective as opposed to either the leadership or the scientific community. Really very, very important in community engagement efforts.
Hello, I was intrigued by Jean's presentation and your comment that that CCR5 mutations are actually on 23andMe. This may seem like a silly question, but what is the role of companies like 23andMe? And I mean, given the real desire for a lot of patients and individuals to control their own genetic testing and to have that information, I mean, can that play a part in a lot of the efforts that we're talking about in terms of increasing genomic medicine and testing?
That's a good question. Yeah, I think those commercially available testing platforms are useful. A lot of people use them for ancestry and things like that, rather than trying to figure out what their polymorphisms are. And so, but they do offer value. If you know what you're looking for, you can look and see whether you do have, for example, a mutation in any particular gene that's on their panel. Yeah, I do think that there is value in knowing that and having access to that. And I think if you, yeah, I mean, particularly people that know what they're looking for, can certainly search through their database and their own data to find that information.
Stephen and then Bob.
Question for Ryan. So I work at a children's hospital in San Diego, and as you probably know, sick babies in Samoa would go first to Kapiolani in Hawaii, and then they wind up at Rady. And so we do a lot of genomes of so-called Polynesians, and see a lot of this issue of how do I interpret this genome. It's really tricky. My question was, now that we're getting into genome-based newborn screening and we want to qualify a variant set for use in diverse ancestral populations, how much, genetic diversity? You showed one example, and so first of all, for that one particular variant that you've done very nice studies on and have looked all across Polynesia, first question was, is your conclusion that within the Polynesian islander populations that there are significant differences in allele frequency or not?
I think the jury is still largely out on that. I think there are differences between different island groups, particularly the degree of admixture that you see between different island groups as well. There's some evidence that there is in particular a jump in allele frequencies from West Polynesia, so Tonga, Samoa, Tuvalu, Tokelau, and Niue, and then pretty much the rest of Polynesia, that's Eastern Polynesia, that there was the founder effect of jumping from that one set of islands to the other set of islands might have created some allele frequency differences there. But beyond that sort of hints at that, I think the folks in the Pacific, we haven't really developed a consortium yet. There are Pacific Islander and Polynesian geneticists who are now junior faculty starting to move up, where that's going to be a priority for us to do. I think the jury is still out on just to what degree we see founder effects that lead to allele frequencies between the different ethnicities within the region.
So just teeing on from that, have people measured fixation indices in these specific populations, and are they significantly different globally, or do they all fall under that, less than 0.1 difference?
Trying to figure out how to answer that question. I'm trying to think about how to not answer this in like a dichotomous way, like are they different or not? Because there's really not an answer to that. I would say no other population on the planet serves as a good imputation panel for Polynesian individuals. So predicting allele frequencies from, say, East Asian individuals is abhorrent. So the degree to which, say, fixation indices indicate a significant divergence from the population, I don't know that anyone's done that study, but I'm going to say that they're high enough that we consider these populations dissimilar from a genetic level.
So Bob, and then Terry.
I had a question for Dr. Lim. So it looks like populations that are exposed to flavivirus have lower CCR5 delta 32s, and it's protective against HIV in populations that have a higher frequency, such as in Europe, they're not as exposed to flavivirus. But prior to HIV, what do you think is responsible for the increased CCR5 delta 32 allele frequency in non-flavivirus exposed individuals? I remember reading something about, well, maybe it was plague, maybe it was bubonic plague, maybe it was this, maybe it was that. What do we actually know about that right now?
Yeah, that's a great question, and one that I spend a lot of time thinking about. Yeah, there have been lots of theories about what was the pressure that sort of caused the enrichment, particularly in Europe. And there is definitely this climb that you see, particularly when you go towards Northern Europe. And I think the thing that the theory that I feel has the most weight is one where they talk about some sort of hemorrhagic viral plague that basically came multiple times over the course of multiple centuries, rather than one plague that happened and killed off 50% of Europeans would really not do the enrichment that you've seen. And so I think that they don't know what the pathogen was, but it was probably something where CCR5 was either very important. It was definitely has to be that the CCR5 was definitely important. So I would say that like it was probably something, a viral hemorrhagic pathogen that hit Europe across multiple places for centuries.
Is it conceivable that it could be genetic drift in the absence of purified selection from flavivirus?
Yes, so there have been at least one very good study that was done that seems to that has proposed that is possible. But I would say that probably the field generally thinks that there must have been some sort of pressure.
Yeah, again, for Ryan, I'm a little curious when it seems as though when you screen for this, you would, I mean, the treatment is just don't give them a vaccine, but you'd be screening for those that already have life-threatening infections. So what's the action then when you find it?
The first thing that comes to mind is make sure everyone around them is vaccinated. So herd immunity, protect the individuals, right? So it's interesting the contrast between by vaccination without ruling out an immune deficiency seems like, well, that's, if we could do that, that would sort of perhaps improve, especially, potentially vaccine uptake. On the other hand, the best way to protect these individuals is to vaccinate everybody in the community so the virus can't make it to them. Does that answer your question? That's sort of like where my mind goes first.
But not exactly, because obviously the kids that they're surrounded by would be the ones you'd want to vaccinate, but also if they're siblings, they'd be the ones most likely to also carry the variants. And so it just struck me as a little bit odd that you wouldn't test everyone to catch those, asymptomatic ones.
And I think it's a matter of resources right now in New Zealand. I think it's a matter of resources in terms of it's not risen to the level of being I'd presume a health priority to sort of do cascade screening or anything like that.
So one thing that we might want to think about in terms of future implementation or development is are there ways to develop really cheap tests for this at the point of care that might only be for these variants, for example? And is that something we should talk to our technology development program at, within NHGRI or in other settings to develop? Is that something that you think might be worthwhile?
I don't know, because I don't know how soon newborn screening with a whole genome or at least a whole exome, like if that's coming sooner rather than later, do we need to invest in do we need to invest in technology and delivery systems for all these individual variants when we might be able to just capture it all?
Well, interesting point. On the other hand, if they can't afford a single variant test, how in the world are they going to afford a genome test? So I don't know. Those of you who are in the technology field, and I know we have some folks here who have feet in both camps, might want to comment on it. Or Jennifer, you just raised your sign. Maybe you want to comment specifically on that?
Yeah, well, one thing to mention quickly is New Zealand was the first country after the US to institute nationwide newborn screening for SCID. And so I think that was very forward-looking of them, and quite interesting that seems different from what you are describing, although I know that the Maori population is involved in their health decisions.
There's government administrations change and priorities change, and there's been a recent shift in New Zealand.
And then another thing I did want to also mention is that there are places that have implemented TREC screening, which is obviously a PCR test, and have added in particular regions where there are variants of significant incidence, they add these things that would not necessarily be detected by TREX. And one of them is in Canada in the Northwest Territories where there's a frequent ZAP70 incidence, and ZAP70 is yes, it's a T cell immune deficiency, but it affects T cells after the stage of T cell recombination and TREX. And so they have added this particular variant to their, to the newborn screen to pick up those cases, and have done it successfully. And I think there are other places in the world that do that as a step, between here and way before whole genome sequencing.
This is a question also for Ryan. So, I completely agree that under or non-represented populations need to be. I mean, we need the database information for what is common, what's not common, but that could be a double-edged sword in the sense of, if you go back to that Twitter exchange you showed, right? You mentioned, oh, well, this variant is present in 2% of, I think there's Maori population, so therefore it's a polymorphism. It can't be disease causing, and yet you showed with the interferon alpha receptor variants, right? So both in your population and the Inuits, that those MAFs were around 2 to 3%, and yet those are not polymorphism. So I mean, how are you, even though you have this population data for this small, essentially bottleneck population, how are you going to distinguish, what is a founder effect versus what is common?
We get things wrong. Science, right? That's a really hard question. That's a question we're sort of always asking ourselves as we walk in. My mentor always told me, know your assumptions, right? Like, so when we deliver that genetic information about the allele frequencies, know that you're assuming that just because something appears to be polymorphic, you're assuming it's not going to be involved, but we could be wrong. And yet that's sort of the standard by which we are often looking at databases like Nomad, isn't it? Is, oh, this is common enough. We don't think of European ancestry as being a founder population, because of admixture, but, and it's been, white people have been pretty well studied. So we don't think of it that way, but that is, you're right, it is a double-edged sword, because we might have ruled this variant out if a priori it was known that it was common, this common.
So I'm going to call on Dusan in a minute, but I'm going to interject nomenclature, which people know I want to do, which is a polymorphism is not synonymous with benign, right? A polymorphism is 1% allele frequency or higher. It can be pathogenic, and we have many examples of that. So just a nomenclature point, but.
So to add to this discussion, and maybe to what George mentioned as well, when we are forming policy on return of genetic results that may impact infectious diseases here, or in this case, vaccination decisions, right? I think we have to discuss among ourselves as to what is the best way to do that. How do we include the vulnerable populations in that decision? How do we make sure that we do it in a way that it doesn't become a political fight, but it becomes a rational decision for the betterment of health of the population while protecting the vulnerable? And I don't have a solution for it. I just think it's something we constantly have to keep talking about, evaluating, because even the ethics of genome sharing and consent has been changing over the past 2 decades, and we have evolved with it. By no means we're doing it right now, nor we were 20 years ago, and I don't know what's going to be done there, but I think we have to keep talking about that at a lot of these things, so we don't end up in a situation that becomes a political choice as opposed to a rational medical cost, population benefit, and inclusion decision.
David?
Yeah, so this may be more of a comment than a question, but thinking about
implementation. And I think the concept of windows of opportunity is really important. As Terry kind of just put up, this conversation around, do you do things that you could do now with a few number of variants when maybe not too long, it'll be the whole genome. And I was also thinking about, we'll talk more about this tomorrow, when Priya was talking about interferon and the genetics of interferon, which was incredibly relevant very recently for hepatitis C, but now with incredibly effective therapies and nobody being treated with interferon and trying to move toward a single shot cure, the genetics may wane for that. And some of that also comes up with CCR5 and other things. So I'm not sure how much of the talk today has to do with, talks have to do with windows of opportunity, because you're talking about fundamental genetics that affect susceptibility to an infection. But as we talk about interventions, whether that's a genetic test intervention or a therapeutic intervention, I think that's a really important thing in terms of thinking about how to prioritize with resources and all that. So not a question, but more of a comment.
Any other speakers want to respond to that comment? No disagreement, certainly. Bob, go ahead.
Thanks. Also by way of a comment, which is, I think both pharmacogenetics and infectious diseases are really great examples of where allele frequency has a lot to do with exposure. I know Steve Holland has published this really nice work on severe cocci, where the allele frequency of the variant that confers susceptibility to severe cocci is actually high enough to make 25%, I think, of all Amish heterozygous for it, and yet there's no cocci in middle Pennsylvania. So it was ruled a benign variant for many years because of its high allele frequency, but it's clearly not when you look in the right population. I think we're going to see more and more of that. In fact, the whole idea of the current clinical classification system, where a single allele frequency cutoff is used for recessives and dominants, like two different ones, but just one for recessives, one for dominants, probably is not subtle enough to really reflect the realities of whether variants are pathogenic or not.
Yeah, I completely agree, Bob. And I think that Jean's talk also speaks to the other aspect of infectious diseases, that selective pressures likely have created environments where you're going to have some benefit to some of these alleles and risk based on the other pathogen that you encounter. And I know in other research that we do, we see the same thing. We see some protection against infectious diseases, but it puts you at much greater risk for autoimmune disorders. So it's just balancing that, like which side are we looking at?
Dusan, did you have another question, or okay, any other questions? Otherwise, I have a question for Andrea, which is, you talk about HPV, remind me of the amazing success that vaccine has been to reduce these cancers. But we have increased vaccine, and we had another mention of vaccines as possible treatments, but there's this increased vaccine hesitancy now. And how does the medical community try and counter that, I guess, is my question. Or what are our options here?
Yeah, vaccine is incredibly effective. It's changing and shaping the circulation of the oncogenic variants. But the problem of some of the immunocompromised population is that, in addition to the issue of vaccine hesitancy and in addition to the problem of actually getting the vaccine, the response to the vaccine in this population is not great. So we measure it in a population of T-cell lymphopenic individual. Their antibody response to the Gardasil 9, it's greatly impaired. So whenever there's going to be a background of T-cell deficiency, we know that it's not going to be an effective one. And the other issues with HPVs is how, and we don't know yet how is shaping and changing the population of oncogenic ones. There's 450 classified HPVs so far. So the oncogenic one, fortunately, are a minority. but it could be expected that could be a shift that is including non-oncogenic variants or not included in the current platforms. And the other thing is that in our immunocompromised population, you start having, or you have cancers like the case that I showed you, in which you're actually having cancer with the integration of a beta HPVs, which is definitely not even on the radar. So far, cutaneous cancer genesis has been attributed only to EV exposures, and the beta HPVs have never been implicated. The more we start looking at sequencing the tumors, and this was to go back to Jennifer's comment about sequencing in tissue beyond blood, the more we're actually seeing integration of variants that we didn't think that were important. So yes, definitely vaccine, but there's probably going to be more.
Any more comments?
Yeah, if I may. And we do have a fair amount of time, but we don't have to take it all. We can, we can stop and just finish a little bit earlier. But I am a little bit curious when it comes to particularly like the hepatitis B and C stories. Are there interactions between the host genome and the genome of the pathogen? Because there's been, I think, a lot of work done on sequencing of the pathogen, and David may talk about it with HIV, et cetera, tomorrow. But can you comment at all about how the specific variant you're infected with may affect, may interact with your own genome?
Yeah, thanks, Terry. That's exceptionally important and something that we've tried to do for quite a while. It was really hard with hepatitis C because we're using often historic samples to do the work that we do, because you've got to build the consortiums, follow individuals, know their outcomes. But we are doing that with hepatitis B. And so we're able, because hepatitis B in parts of Africa can occur at much younger ages, so we can get both the sample and the sample of the pathogen and the individual. But I do think that that is the complexity is, and we've seen it in viral diseases, but that complexity of the viral genome and the human genome having that interaction. And it could be really for any pathogen.
Well, because one of the things that comes to mind is cystic fibrosis, and as I understand it, in cystic fibrosis, each patient sort of gets their own Pseudomonas strain that kind of encodes on them and stays with them for years or life. And is that genomic, or is that just whichever one happened to establish itself first? And it's probably a little bit of both. And I don't know if there are cystic fibrosis Pseudomonas people around the table or not, but might be another sort of model of that.
Scott?
Yeah, that just got me thinking, because I'm a rheumatologist. Some of the biggest susceptibility factors for lupus and for MS are things like EBV, but the mechanisms of that are totally unknown. And so is there an opportunity within biobanks, like you kind of did for CMV, to look for, some of the genetic characteristics or interactions between people who are reactivating things like EBV or even CMV and susceptibility loci for autoimmunity?
I do think that's where biobanks could be useful, is EBV, CMV, like pathogens where there is high prevalence and presumed that the entire population has been affected. I think you really can do a lot of work, and I think that's what we saw with MS and EBV, that work that came out of large-scale studies. So yes, I think that's one of the better benefits. And there could be other pathogens. You just have to think about what is circulating by a certain age in a large portion of the population.
Is Liliana again from this corner over here. We're just going to respond to some of your questions regarding pathogen adaptation to living inside a host. So many bacteria actually undergo very severe dramatic genomic reduction once they become fixed into a human host, especially once they're carried for a long time. We saw also in COVID, there's a specific idiosyncratic evolution of the pathogen inside the host and even inside different tissues. But whether those trajectories are the same, different human hosts, are questions that we're currently exploring through our genomic centers as well as our systems biology programs. I forgot what. Oh, I was going to mention. We do know, for example, in commensal pathogens, they also are commensal organisms in your microbiome. They are also sort of unique to each person. And I think that kind of source data is going to give us a little bit of better understanding of if there are human genomic or other variables that do make and have an effect on what genotypes can exist of those bacteria in your gut. The issue with most of our bacterial studies or other pathogens, fungal, viral, is that they're very short duration, right? So we can't see that large scale genomic adaptation and selection for other pathogens. As you would so CF is a great case, but it's also cystic fibrosis. So it's not necessarily the right model to study human genome pathogen genome interactions.
So I had a comment for the speakers around this being able to capture exposures. So we brought up the biobanks a little bit, and I'm wondering, since it's important to try to find people who like may be resistant, for example, like what kinds of surrogates for exposure might be relevant to better capture in biobanks, so that some of this work could some of the research could be more effectively pursued. I don't know if that makes sense. So, for example, on the topic of exposures, for example, there are examples on finding controls who were exposed but didn't actually get the conditions, and so if we were able to know whether somebody was involved in drug use, or like some kinds of surrogates that could say that somebody's more likely to be exposed. Are there opportunities to better capture like these like surrogates of exposure in biobanks? And has there been any work in that space?
Yeah, Casey, I think it's a really good question. I think that's the problem, is there isn't a surrogate of that exposure that we have, right? So we diagnose disease, biobanks can work for that. We diagnose infections, but we don't know if you've been exposed or not. And so without that surrogate, it makes biobanks really difficult to use, because the assumption then is that someone has been exposed, but if they haven't, then you're not comparing apples to apples. So that's where cohorts have been really beneficial, because you can follow people over time and sort of determine whether or not they've been exposed through frequent testing.
But correct me, and Jennifer actually has a question, but I'm going to jump in anyway. So my understanding is that the Ohio River Valley, almost everyone who's lived there for any length of time has been exposed to histoplasmosis. So wouldn't that be one of many kinds of markers for exposure? Or living in Arizona, you've probably been exposed to whatever it is in Arizona. Is it blasto? I don't remember. But at any rate, you know, and trying to, is it cocci? Okay, sorry. Yeah, this is why I'm an internist, not an infectious disease person. But at any rate, you know, maybe, you know, coming up with some of those kinds of surrogates would be a very helpful thing, at least for the research, and maybe as markers for the patients.
Yeah, absolutely. I think that, you know, you can make some assumptions based on what you think is circulating in a given area. It's just harder to use a biobank to do that, right? So if you think of like the UK biobank, we can't make the assumption that the people in that cohort have been, in that biobank, have been exposed to certain things.
I mean,
I was just going to bring up how complicated the whole field of vaccinology is, and you know, we've been sort of talking about vaccines as if they're, you know, one is equivalent to another, and yet we know that, you know, the reason MMR are live vaccines is they don't work if you don't have live vaccines. They don't give lasting protection. And on the other hand, now we have a whole new technology with RNA vaccines. Maybe we should be looking at how we can make vaccines safer for everybody, because we know that a certain number of people are going to have variants that cause big problems when they get vaccines.
Hi, this is Inka from NIAID again. I have a question, and I noted this earlier, but Liliana reminded me. So there was, I think it was Andrea who said that in his talk about treating the immunodeficiency by, I think it was transplant, and then looking at the microbiome and the microbiome change before and after treatment. So I'm wondering, how common is the microbiome looked at after transplantation or after changes in immunodeficiency, and if this is commonly done, and how the microbiome might change before and after, because they could also have implications for subsequent infections, I guess.
Thank you. Probably Liliana can answer and can add on. So what we did was probably refer to skin colonization by HPV. So that's pretty ubiquitous signature. All of us are harboring some type of HPVs. And so that is pretty stable on our skin. And on the type of immunodeficiency that we studied in NIAN, we've been seeing a very strong signature to enrichment of those HPVs in, for example, DOCA8, GETA2, IL2RG, or other type of immunodeficiency. So that is translated in a somewhat more stable change. I don't think there is enough granularity to look into what type of taxa are more or less expanded. Those data are coming through now that there is more metagenomic pipelines. And in the particular case that I showed you, there was enrichment of the alpha oncogenic HPVs that should not be on the skin, should essentially be populating and colonizing mostly the mucosal surfaces. So in immunodeficiency, you also shift the regular tropism that you would be expecting for these particular viruses. But I don't think we have enough data to be systematically characterizing different immunodeficiency and shift in bacterial species for what I know. Although the skin, I think, going back to sequencing of the skin and tissues and other opportunities is an incredibly accessible source. If we're able to identify, and going back to the concept of exposure and signature of exposure, if we're able to identify also through the skin particular features and signature that can be complementing data that are going to be genomic data, especially at the metagenomic levels. I think the skin reflect your immunological status very well. It's populated by any immune cells that is there. And from what we've seen, the skin microbiome does reflect a lot of what your immunological condition is. But again, we haven't systematically done that. Julie Seger has done a fantastic job with ID Kong, but still it's early. I don't know, probably Liana want to add something.
And to that, I mean, we're hoping that the microvirome that the NIH Common Fund is starting will give us some additional answers, at least what the virome looks like in healthy human populations. So a good comparator, hopefully.
Yeah, and just building off of Jennifer's comment about making vaccines safer, I remember a time many years ago when the argument was made that every clinical trial of any drug should include genetic, at least genetic material stored and tested later. I would ask my NIAID colleagues, is that common in vaccine trials? Is DNA stored and examined in vaccine trials? Inka, maybe that's a question for you. I don't know.
So do you mean human?
Yes, yeah. So to look for, in case of adverse effects, then obviously one could do the GWAS or whatever studies. This is discovery research, so sorry, but I'm just asking that question.
I'm not an expert on this. I would think very restricted. I mean, of course, and we know that some people respond better to vaccines than others, and I think there was a study that I remember that related to the circadian rhythm, and you have a better vaccine response, I think, in the morning than in the evening. I'm not sure how detailed people actually look at human genomics and and see how they respond. I mean, I would think B cell, if you have B cell issues and others, then you would have less of a vaccine response. So I guess the common players would likely be good candidates to look at, and maybe somebody else has a better response than me.
So I just might suggest for the meeting that you and Lucas are planning, maybe that would be a question that Lucas
I mean,
wants to
I know
respond.
who I could ask potentially. This is Maggie
Oh.
over here.
Oh, hey.
Sorry, from NIAID. I'm from the autoimmunity and mucosal immunology branch and Division of Allergy Immunology and Transplantation. There is a large cohort for COVID that includes a lot of healthcare workers that had samples before they got COVID, and then after they either contracted the disease, had the vaccine. And so have they done that? I actually don't know because I'm not personally involved in that, but that might be one that's recent enough where maybe they do have that information or have the samples stored for that. I don't know.
And not like precisely, but I don't think it's routinely done in vaccine studies. I think you generally try and measure like actual, correlates of protection. Obviously I think you run up, particularly I think in larger clinical trials and sort of the economics of that. People really want to save costs, and sequencing is probably one of those costs that probably, but back to the earlier section about biobanks, one thought I had, and I'm sorry, this is also very in a bit of discovery. I remember in graduate school, there was a discussion we had in my group about looking at, understanding populations that have a large amount of data and samples biobanked and have been exposed to certain pathogens over time, and then comparing those to populations that have recently been exposed to that pathogen hadn't been previously, and looking at maybe differences in outcomes. Dengue is one of those that comes to mind because I remember that there was a study back in the 90s of a dengue outbreak in Cuba, where there was a big, with dengue, there are 4 serotypes, and if you get infected with a different serotype after being previously infected, you're predisposed to have more severe disease. And Cuba's an interesting population because you have a mixed European population and African population. And there was this big wave, I think dengue 3 in the island. People got mildly ill, no big issues. And there was a big secondary wave of disease about 2 years later, if I recall, from a different serotype, and that you really saw quite dramatic differences, in severe disease distribution, based on sort of people's ancestry. And in my lab, or one of the labs I rotated in, they had biobank samples from Nigeria over years and actually found that dengue and Zika had been circulating in those populations for a long time, but didn't really seem to observe severe outcomes. And so understand like what populations have been exposed in the past, what populations might be newly exposed, might be a way to leverage those biobanks to actually have more power to detect those genes that might be involved in the variation of phenotypic clinical presentation.
Yeah, thanks. I did want to come into this exposure discussion with the All of Us research program, wondering, I'd love to hear more from this community about what kinds of exposures would be useful to incorporate into our data set. Just so you know, we are going to collect residential and occupational history over the lifespan of participants as best as we can, and have geocoding to a number of linkages and geocoding to a number of databases, including starting off with the Environmental Justice Index. We collect social determinants of health data, and there's a number of other data types that I'm hearing this community would think would be important to understand the manifestations of certain infectious diseases. So maybe it's during this meeting or afterwards. I welcome the opportunity to learn more about what we can do to enhance your ability to do the kind of research we're talking about here.
So I was going to mention it before when the databases were discussed about. So maybe something to think about that we can all incorporate in both cohort and maybe the biobanks, even the large scale ones, is seromics. Because with FIP-seq now it's not that expensive to do it, and you can find what you're not looking for, right? Which is what we also want to know about. What is it that we are exposed to, but we don't know that we are not routinely testing for. So I think that can be a rich beginning, at least in the pilot program that has that type of depth.
What's the type of sequencing?
I mean, FIP-seq is one of them. There is a few different ones. Basically it's a library that has an antigen to it, and then you basically catch the antibody. It's basically a high throughput ELISA, if you wish, that's sequencing based and decently inexpensive.
Yeah, that would at least show history and overall serology.
Okay, I think you're done. So super, thank you. We're about 5 minutes early, so why don't we save a little bit of time and come back at 3:20? So please be back at 3:20. Okay, if y'all could take your seats, we'll get started again. Please take your seats and we'll get. Introduce you. Okay, we're going to get started again. So we have George Mensa and Inka Sastala who are chairing the session. George and Inka, take it.
Hey, thank you, Terry. Welcome back, everyone. So we have our first talk this afternoon is virtual. So we have Dr. Roshini Abraham from Ohio State University, and the title is, or maybe it's different than what I have, current status of clinical use of host genetic information. No, wait, it's the evolving landscape of diagnostic immunology, past, present, and future. Roshini?
Yes, hi, can you hear me?
Yes, we can hear you.
Okay, thank you. I apologize that I cannot be there in person. It has been a wonderful day so far, and I'd like to thank Terry and all the members of the planning committee and the moderators for having me as part of this session. So I oversee a clinical diagnostic laboratory, and so I've been asked to talk a bit about how the clinical laboratory, particularly the clinical immunology laboratory, plays a role in the diagnosis of these inborn errors of immunity. And I think from all the talks we've heard today thus far, it's quite clear that our understanding has evolved with regard to these inborn errors of immunity. We've gone from a Mondrian concept to a Pollock concept. And so that requires that in the clinical laboratory, in the diagnostic setting, that we have a range of tools available to help clinicians with diagnosis and monitoring of these patients. And I know the title evolved a little bit, and it was talking about diagnostic testing, past, present, and future. And we've come a long way, I think, in the clinical diagnostic immunology laboratory, but we still have a ways to go. And when we think about clinical testing, particularly immunology testing, we always have to think about our reference cohort, much like one word in the genomic setting. And this is an area that hasn't received much attention in the clinical diagnostic laboratory in terms of developing robust reference cohorts, because there's so many different factors that can influence the immune response, both phenotypically and functionally. And I feel that this is an area that we ought to pay more attention to in terms of developing robust immune profiles across the age and taking all these variables into account, because we use this data to interpret patient data. And when we also think about clinical diagnostic laboratories, and when I say clinical diagnostic laboratories, I specifically mean immunology and not chemistry, microbiology, and all the other clinical labs. We think of it, it's good to think of it like a pyramid. You know, the base is broad, and as you get to the top, you've got fewer and fewer. So you've got a lot of clinical diagnostic labs that offer some basic immunology testing, whether it be lymphocyte subsets, immunoglobulins, etc. And then as you keep climbing the pyramid, you come to fewer and fewer labs offering more and more esoteric testing that is not widely available. But the goal of bringing tests into the clinical diagnostic immunology lab is to make it broadly accessible to physicians, and also to meet all the stringent regulatory requirements that various regulatory bodies, including the FDA, requires. And I don't think it's well appreciated all the time, the amount of money and resources that it requires to validate a single clinical test, an immunology test, to bring it into the clinical domain. And that's partly why the whole clinical lab immunology enterprise is shaped like a pyramid rather than a square or a circle. So what I hope to do today is to use some of the tests we have developed in the clinical laboratory domain to either functionally evaluate variants of uncertain significance in IAI genes or to help physicians identify targets for immunotherapy, etc. So I'm going to start with this particular case. And that is a 13-year-old male who was referred to our hospital in Columbus, Ohio. He had a history of recurrent infection since early childhood, but his first pneumonia was the year prior at the age of 12. And when taking his history and looking at him phenotypically, he was found to have microcephaly. He also had microcephaly at birth. He had normal immunoglobulins with IgA, selective IgA deficiency at the time. He had a normal karyotype, etc. And he had some genetic testing. But he was referred to our hospital because he developed the pneumonia and he had another immunological evaluation elsewhere and was now found to be profoundly hypogammaglobulinemic. But he also had some increased IgM levels, absent vaccine responses, and some B-cell lymphopenia and NK lymphocytosis. His T-cell counts were normal. So he underwent genetic testing, which identified compound heterozygous variants in the gene encoding the nibrin protein associated with Nijmegen Breakage syndrome. And he had one of the variants, which was a Slavic founder variant, the 657 661 deletion, which he inherited from his mum. And he had another variant that was novel, but considered pathogenic, which was present in his father. His father was adopted, so there was no real family history on the father. So I had started developing a test to look at DNA double-stranded break repair response when I was at the Mayo Clinic. And when I came to Columbus, I completed the validation, moved it into the clinical laboratory as a clinical test. And in this particular assay, we look at phosphorylation of 3 key proteins that are associated with the DNA double-stranded break repair response, which includes phosphorylation of ATM after induction of DNA double-stranded breaks, as well as phosphorylation of the histone gamma H2AX. So you can see that he has some impaired phosphorylation of ATM, but his gamma H2AX looks normal. However, when we look at the kinetics of the DNA double-stranded break repair response, we find that, and this is data from his T cells, that his B cells and NK cells looked exactly the same, and that he had failure to dephosphorylate the gamma H2AX, indicating that DNA double-stranded breaks were unrepaired or there was ongoing DNA double-stranded break damage. And if for those of you may be familiar, nibrin is one of the key proteins involved in DNA double-stranded breaks, both due to homologous and non-homologous end joining. So it was quite clear, and phenotypically, actually, he had the classic bird-like faces as well. So it's a rather a bit of a shame that he was diagnosed only at 13 years of age and not early in life. And as I was wondering, you know, what this meant for him, because I was concerned that he might wind up getting a malignancy. Well, 4 weeks after that diagnosis, he was found to have a T-cell lymphoma, and for which he received chemotherapy. But the problem was after chemotherapy, he got multiple bacterial infections which were treated, but then he developed pulmonary aspergillosis with necrotizing pneumonia. And so the clinicians, the ID clinicians reached out to me and asked if I could help do some immunophenotyping to identify whether he might be a suitable candidate for immunotherapy. And so based on long-standing evidence, as well as some work I'd done at the Mayo Clinic, I'd looked at HLA-DR expression of monocytes, because the loss of HLA-DR and monocytes is associated with a state of immune paralysis. And so this boy had low DR expression of monocytes. And so it was determined based on the NETIA protocol to treat him with recombinant interferon gamma. And then we measured changes in HLA-DR expression. It went up and down over a bit, but then stabilized by what I refer to as time point 11 over here. And then by then he was clinically much improved. He was discharged and had recovered from the aspergillosis. And we've had a few different patients where we've used this approach of looking at DR expression of monocytes to identify individuals with immune paralysis and to select them for treatment with recombinant interferon gamma to sort of reverse this scenario of immune paralysis. Now, from a laboratory perspective, there are many different assays one can do. One can measure HLA-DR expression on CD14 monocytes and look at the mean median fluorescence intensity. Or one can do it actually a little more robustly by using what we call molecules per cell quantitation to actually try and analytically standardize the measurement of this particular marker across clinical laboratories. But regardless of which way one does it, this is a potential way of identifying patients who might benefit from treatment with recombinant interferon gamma therapy or GM-CSF in the context of refractory infections. My second example is a 2-month-old female who was referred from the outside, who had hepatosplenomegaly, failure to thrive, identified to have pseudotorch syndrome, and USP18 deficiency, which is a type 1 interferonopathy, is associated with pseudotorch syndrome type 2. And she was found to be a compound heterozygous, with one of her variants being a frame shift and the other a missense. And so a couple of years ago, I had developed a test in the laboratory to quickly assess the type 1 interferon signature on CD14 monocytes. I can, during the discussion session, explain why I chose this particular marker. But long story short, I was looking for a marker that was easily accessible to physicians, that I could turn around rapidly and do it on whole blood, on a small amount of whole blood. And not surprisingly, this patient had a strong type 1 interferon signature with strong expression of Siglec-1 or CD169 on the majority of her monocytes. And she was started on treatment with JAK inhibitor therapy, and we've been monitoring her periodically since. She did show an improvement after 4 weeks of JAK inhibitor therapy. And I'll show you some data in a moment, but we also see a type 1 interferon signature in the context of viral infections like COVID-19. But these signatures tend to be transient, as you can see in the example below. This is a patient during the middle of a COVID-19 infection. She had a strong type 1 interferon signature, which resolved within 6 weeks, and then even 9 months later, she was essentially normal. So in part, with that assay that looks at type 1 interferon signature, I also look at a type 2 interferon signature using a marker called PDL1 or CD274 on monocytes. And here's an example of a young male with hepatosplenomegaly, elevated ferritin, soluble IL-2R, etc., who happened to have perforin deficiency HLH. And so as we were getting ready for transplant, the decision was made to treat him with emapalumab. So I looked at a number of different markers to look at CD274 expression. And as you can see, PDL1 is a molecule that is induced by interferon gamma. And so you see a strong interferon gamma signature on this patient, which is also supported by the CXCL9 levels that are high after treatment with emapalumab. That normalized, and he was slowly starting to drift again when he had his transplant and has now been stable. We also look at some cellular biomarkers in these HLA patients because they have increased activated T cells, particularly in the CD8 compartment that express HLA-DR and CD38. So we use a number of different markers to evaluate our HLH patients. We use this PDL1, CXCL9, the activated T cells, and we use that to both diagnose as well as monitor response to treatment. And this particular child had a male sibling who was also affected, who unfortunately died of COVID-19 prior to transplant. So I've looked at about 600 patients with a variety of clinical contexts for both type 1 and type 2 signatures, and about a quarter of those have a positive type 1 interferon signature. As you can see over here, far fewer patients have a type 2 interferon signature, and most of these are HLH patients, though there are some patients with some autoimmune cytopenias, etc. And we've looked at ROC curve analysis and found that you can get really good sensitivity and specificity of CD169 for type 1 interferonopathies, a variety of them, with the cutoffs that are shown over there, and the same for CD274. So I won't go into the details of the type 1 interferon pathway because you heard a little bit about it from Ryan in the previous talk, but nonetheless, there are many different pathways of type 1 interferon induction and receptor signaling. And there are about 38 monogenic or Mendelian disorders associated with type 1 interferonopathies, and about 17 of those are associated with to do with nuclear acid substrates. And there's been some data to suggest that Aicardi-Gouttier syndrome has been identified by newborn screening for X-linked adrenoleukodystrophy. Aicardi-Gouttier syndrome is a prototype type 1 interferonopathy. However, I think there are some challenges with introducing newborn screening for Aicardi-Gouttier that we can discuss later.
And 1 minute, Roshini.
Yes, almost close to being done. And the other is going back to the discussion regarding using combinations of markers. So this is a study from Carl Allen and his group where they looked at different plasma proteomic data and identified certain cytokine or chemokine signatures that could discriminate in context of inflammation between HLH and sepsis. And they showed that CXCL9 IL-6 was a good discriminator of HLH versus sepsis, though, of course, if you added another 8 more classifiers, you could discriminate and improve the sensitivity and specificity. One of the topics that Terry had asked me to briefly touch upon, because this is a topic in itself, is that we are currently under significant threat in the clinical laboratory in all areas with regard to new FDA regulations that go into effect May of 2025. And right now, the FDA is quite implacable. So clinical laboratories are preparing for this, but I think as a field, we stand in great danger of any of the innovation that we are trying to bring into the clinical laboratory being defeated by these FDA regulations. So I think as a mandate for diagnostic and clinical laboratories, we try to improve the repertoire of testing, and we can use that for functional evaluation as well as phenotypic assessment. And oftentimes we find a combination more useful than a single marker, but the regulatory oversight is indeed a significant threat. And as we've heard from some of the lectures today, timing of immune assessment is very important. And patients who are pre-treated, can alter the interpretation of the immune signature. So it's ideal to obtain a baseline. As for the future, well, it's bright or grim, depending on your perspective and depending on whether the regulatory bodies have the final say or not. I'd like to thank the people in my laboratory who try to translate research findings by validating them and bringing them into the clinical domain. And I also thank my former lab at the Mayo Clinic.
Thank you very much, Roshini. We would need about 10, 20, 30 seconds to switch from the remote to Ephraim's presentation, but it looks like we're almost there. By the way, we're not going to hold those last 10 seconds against you, but we're going to count on you getting us ready. No. All right. So Ephraim is going to tell us about the the available technologies for assessing host response. Ephraim, over to you.
Thank you very much. I'm going to start with a little bit of maybe a philosophical preamble before we get into actual technologies, which relates to the way in which we think about technologies and use technologies as clinicians. And I'll just sort of jump in here. Most of you have not been sort of industry, so I didn't know whether I needed a disclosure slide, but there you have it nonetheless. So I come from Danaher as well as on the academic side at Duke and the Durham VA. But Danaher, if you've not heard of it, is a large company that is itself comprised of many companies, some of which you may have heard of, and they're sort of listed there at the bottom. So I'll jump on to phase. So diagnostic reasoning is intuitively Bayesian. We think about how likely a particular diagnosis is, and then we initiate testing in order to help inform what our differential is. And a lot of that is dictated by the history. I mean, the history alone can give you a diagnosis in the majority of cases. And then you move on to an exam, and then you move on to testing. And you go through this stepwise process, and ultimately to get to some diagnosis where you have sufficient diagnostic confidence in order to act and take the next step, whatever that may be. And so we intuitively think that way, but that's not, but we don't hold our tests to the same standard. We sort of force clinicians to that we do statistics and p-values and sensitivities and specificities. And this then becomes the bar, the standard by which we hold all of our diagnostics. We want things to be nice and simple. We want a troponin algorithm for everything that we do, not just in infectious diseases, but everything, right? If the patient has had chest pain for more than X number of hours and you do a troponin and it is less than 6, then you're okay to send them home. If it's between 6 and 20, repeated after 2. So we like that. It tells us exactly what to do. Medicine is increasingly complex. And I mean, only going to become even more so as we introduce genomic information and other multimodal information. So clinicians rely on algorithms to help them make sense of what it is that they're supposed to do. But then, when you think about and you ask them, well, how are you using diagnostics? They're not all, most of the diagnostics we use are not troponin-like, which is itself imperfect, although everybody says they want the troponin of infection, the troponin of whatever. And so when we think about what this looks like in the context of sepsis, where is our troponin for sepsis, and sort of the most extreme form of infection? This is a little dated. There's actually a more recent review, but I just didn't bother updating the slide. But these are all of the biomarkers that have been looked at for sepsis. And they divide them up into different physiological measurements, coagulation-related biomarkers, vascular biology-related biomarkers, and so on. And of all of these biomarkers that have been evaluated, they all have some relationship and some correlation and prognostic value in sepsis. And I can tell you that probably, well, 0 of them are used in clinical practice. So clearly it's not that we need a biomarker. We have biomarkers, so why isn't anybody using them? And I'll highlight a couple of examples of tests that would probably in no way pass muster today. Well, we know they wouldn't pass muster today, but yet are actually utilized. And so how do these tests break through this paradigm? And I think it's largely because they've been around since before any of the physicians using them were born. So they just grew up with them. I could add white blood cell count to this list. Is almost as good as flipping a coin to figure out if you have an infection. But I can't tell you how many surgeons I've said, well, did you check the white count? When I tell them, I'm seeing a patient who has pus coming out of a wound. Well, did you check the white count? Like, I don't care what the white count is. So ESR, right, discovered more than a century ago. CRP, another one, right, that's now almost a century old, that we actually, and CRP is actually used relatively commonly to help guide whether or not antibiotics should be necessary, a little more so outside of the US than in the US. And the reason is because even though these tests, we know implicitly that they're not super specific or sensitive, that still have value. And that's particularly at the extremes. And this is going to be a point that comes up again a little bit later. But if you look at low values of ESR, this is in the context of distinguishing osteomyelitis from diabetic ulcers. But low values and high values can be informative. It's that large number that's in the middle that, of course, is difficult, but that's okay, right? We see a value in the middle, and we kind of shrug our shoulders and move on. But if it's really low, we're like, oh, okay, I feel better about that. Or if it's really high, it can tilt you into that degree of greater diagnostic certainty. So that was my sort of philosophical backdrop. So now let's get into what some of the technologies are. So these are all FDA cleared. I'm only focusing on US, so not sort of IVD or CE mark, or IVDR or CE mark. But these are, and I also will apologize that these are not all genomic or transcriptomic in nature, but they all are host response diagnostics. So this was a relatively recently cleared one. This is actually a lateral flow assay that measures qualitative levels of CRP and MXA MXA is increased in response to viral infection, CRP in response to bacterial infection. It's nice, it's cheap, 10 minutes, and the indication for use is as an aid. You'll find all of these are aids in the diagnosis of. None of them are definitive. Aid in the diagnosis of bacterial respiratory infection to differentiate from non-bacterial etiologies in essentially adolescents and adults in an urgent care or ED population with less than 7 weeks of acute respiratory symptoms who've also had fever. And this is qualitative, so it's very much a yes/no sort of a scenario. But you can see I'll focus on likelihood ratios, which again sort of speak to how useful these may be, and it's actually pretty good, right? So a likelihood ratio of 8 when positive, 0.08 when negative, and this is specifically to help determine whether or not someone has a bacterial or a viral infection. So not too bad. This is another one. This is the MEMED, an Israeli company that measures 3 proteins, immunoassay proteins. These are quantitative. And those 3 proteins, those levels are fed into an algorithm that then gives you a score. And that score essentially correlates with the likelihood of infection. And the score is basically from 0 to 100 over here, you sort of see on the left. And now we're kind of getting into that stratification, right? Where if you look at patients who are in the highest band, you have a likelihood ratio of 34, and in the lowest band of 0.1. But again, you've got a large number in the middle, but even so, there it's fairly decent, 5 and 0.13 in terms of positive and negative likelihood ratios. And only around 10 to 15% of patients fall into that middle category that's sort of considered indeterminate. This is an interesting one. This is host response as measured in sort of a cellular biomechanical form. So as patients deal with infection, their white blood cells become activated, and macrophages and monocytes in particular. And so what they do is they almost create like a little flow cell. They pass blood cells, and then they exert an external pressure on them, and they look at the squishability of the cell. And over on the left, you can see what sort of a normal white blood cell would look like if you exerted some pressure on it, and what a sepsis patient white blood cell, how it might become deformed in the context of that external pressure. And they use that essentially as a biomarker for the presence of infection. And so their indication is an aid in the early detection of sepsis with organ dysfunction arising within 3 days of testing in patients showing up to the ER. And you can see similarly, they have these bands, 1, 2, 3, and depending on which band you're in, the likelihood ratios go up. Here, maybe not as compelling as what you saw before, a negative likelihood ratio of 0.31, a positive of just under 3. But even so, an added tool, good enough to get adopted into use? I don't know, time will tell. Similar sort of concept, this is the sepsis monocyte distribution width. Here they're looking at the distribution of size of monocytes in a population of cells, which increases due to cellular activation in context of sepsis. And here, similar sort of indication to identify patients at increased risk of developing sepsis within 12 hours of presentation. And here, there is only a single cutoff. You don't have bands, but you could see in when the likelihood ratios, if you're sort of above that level, 2.4, below that level, 0.3. Or the point estimates, around 2.5 and 0.3. Now we're getting into gene expression, and you'll hear more about a gene expression approach in the next presentation as well. But here they're measuring the levels of 2 genes. One is upregulated in sepsis and one is downregulated in sepsis, and they combine those to basically create a score, which, as before, gets broken up into 4 different bands. And you can see, in this case, these are the probabilities of having sepsis, depending on which band you fall in. So it's worth noting that if you look at patients in band 4, 20% of patients who test positive in band 4 won't have sepsis, and 11% of patients who are in band 1, the lowest likelihood of sepsis, will actually have sepsis. So most people would look at this and say, well, wait a minute, you're going to give me a test that's going to tell me that I'm going to miss sepsis 11% of the time? I'm not willing to accept that. But again, they forget that what they're already doing is so bad that this has got to be an improvement. But one thing to note, this is, I think, sort of interesting, is these signatures were largely derived in patients from Utah and the Netherlands. So you can think about what they have in common. But if you just take a bunch of healthy Black individuals, more than a third of them will test positive for sepsis. completely healthy. And slightly lower, but still perhaps unacceptably high population of Asian background patients will test positive for sepsis. So again, something to note in terms of the derivation of these things. This was an interesting one recently cleared by the FDA. This is a clinical decision support tool. And they basically take more than a dozen different parameters that you can identify in the medical record, as well as some biomarkers, and create effectively a score or a likelihood of somebody having sepsis or progressing to sepsis within some period of time of presentation. And here, again, your likelihood ratios on the low end, 0.13, on the high end, 15. It's pretty good. But again, it's going to be a fraction of all of the patients that you test are going to fall into either of those bands. Okay, a little bit of a preview of what's coming. In the bacterial versus viral space, there's a lot of activity. I won't go into each of these, but the one on the left, Inflammatics, is what you'll hear about in the next presentation. Cepheid, one of the Danaher companies, is also developing a gene expression tool for bacterial versus viral discrimination. And another company, Biomeme, previously been affiliated with, also developing one. I just want to spend a moment talking a little bit about sepsis, and this isn't necessarily a product, but rather an area that I think is very exciting. I was hoping I'd have time to go to it, so I'm glad I do, unless I'm told otherwise. Oh, perfect. Okay. So one of the inherent challenges, and probably one of the reasons that large table or slide of many different biomarkers have failed in sepsis is because they treat sepsis as a monolithic disease. It would be like saying, I'm going to create a single test for cancer and expect to identify all cancers. And based on that one test, I'm going to treat all cancers with one drug. It's bound to fail. And the same is what we think is the reason that things have essentially failed in the context of sepsis, both from a diagnostic as well as a therapeutic perspective. Sepsis is a syndrome. It is not a singular disease. And if you then assume that it is, and you treat everybody with the same drug or try to diagnose everybody with the same approach, you're going to find that some people might benefit from a particular treatment, some might be harmed, and there's probably a lot of people who derive no benefit at all. And so a clinical trial ends up showing no effect. But what we're finding through a lot of research that's going on is that if you now start to try to define some, basically the biological phenotypes that naturally occur within a sepsis population, now you can develop a potentially new strategy. And so there's different terms, there's some nuances in terms of whether we're looking at endotypes or subphenotypes or subtypes, but it's the same general idea of looking for characteristics that naturally segregate patients or subpopulations with sepsis.
2 minutes.
Okay, perfect. Thank you. There's a number of papers that have started looking at that. I've only focused on ones here that utilize gene expression or transcriptomic approaches, and more continue to come out. And there are also others that have looked at coagulation markers, that have looked at clinical variables. So there's a lot of different modalities you can use to get to these. This is just one published by a group at Oxford that we're collaborating with. They essentially did transcriptomic studies and found that there were depending on where you created the cutoff, 2 groups that they sort of categorized it in. But you could probably almost see 3 or 4 different groups that these patients fell into. But they created the cutoff at a high level, identified 2 subgroups, and then that was all done unsupervised. And then they said, well, is there any clinical relevance? And sure enough, one group has worse outcomes than the other. But you couldn't have predicted which group any given patient fell into based on their clinical characteristics. And then they did something which I think is really compelling, which is they looked at, in a post hoc manner, at a clinical trial of patients who got steroids or placebo. There's a lot of controversy as to whether you should use steroids in sepsis. Some studies show a mortality benefit, some show an increase in mortality, most show no effect, and there's a lot of uncertainty. So the punchline is that when they compared, they looked at these sepsis response signatures among patients receiving placebo, and they found that the one group that generally has worse outcomes, SRS1, indeed had worse outcomes in terms of a lower survival. When they just focused on SRS1 patients, they saw no difference between placebo and steroids. But when they took the group that generally had a better outcome and they got steroids, their mortality went way up, lower survival. So they demonstrated a heterogeneity of treatment effect or a treatment interaction effect between this underlying immune profile as measured by gene expression and steroids. And there's a lot of reason to believe, and there's precedent in other critical illness syndromes like ARDS, that there are a lot of treatments that you can utilize that can demonstrate that heterogeneity of treatment effect when you find the biomarker that helps to now stratify your sepsis population. And just to highlight the fact that this isn't just theory, but this is actually being put into practice, these are the clinical trials that are either active or going to be starting within the next 12 months that are taking a precision medicine approach where they identify biomarkers to stratify patient populations and then essentially treat them and look for that heterogeneity of treatment effect. Another area of future development is in companion diagnostics in infectious disease, target the underlying biology. And these are both targeting innate immunity, vascular integrity, as well as the angiotensin pathway. And there's a lot of preliminary data to suggest that this is likely to be a successful approach.
You're framing me on it. Very good.
This is perfect, man. This is perfectly aligned. Thank you. So, so really, the conclusions are almost taking a step back. One of the challenges that diagnostics developers have is in developing diagnostics that are going to be received by clinicians who've been trained to fight against their Bayesian nature when they look and evaluate and decide whether they want to adopt diagnostics. I'll highlight, and you know, again, I don't know whether Pravesh is going to talk about the timeline that it took them, but from the time of discovery to actually developing these tests, you can at least a decade. So we're really just now starting to see these tests emerging on the market. Host response is a great tool to answer a variety of challenging clinical questions, but ultimately creating a test doesn't mean that people are going to use it. Just because it's there, that doesn't mean it's going to be helpful. And so we're adding that additional layer of expecting clinical utility studies to demonstrate the evidence that ultimately is going to be needed to drive reimbursement and adoption. I think that's it.
So we already had a teaser about the next talk
Yes.
about translating host response-based diagnostics to clinical practice by Pravesh Khatri.
All
Go
right.
ahead.
Thank you for having me and be a part of this wonderful talk, wonderful workshop. And thank you, Ephraim, for setting up the stage that I could get rid of so many slides. So thank you very much. Now, today I'm here as somebody with feet in both academia and industry. I'm a co-founder and a chief scientist for informatics, but I'm also a professor in Department of Medicine at Stanford. And my disclosures, I'm going to talk about informatics related work here. So because everybody else has talked about the difficulty in diagnosing infections, I'm going to talk about why host response. And probably everybody in this room knows this study by Jen et al. In New England Journal of Medicine. All pathogen detection-based tests that were tried across 9 different clinical centers in patients with radiographically confirmed pneumonia. So this is, we knew that they had infection, 62% of the people, no pathogen detected. And then the question becomes why? And the answer to that is immune system. One thing that immune system has been evolving for millions of years is to make sure that any danger signal does not become systemic. That's its one single job. If there is a danger, make sure it's quarantined and get rid of it. And if you had asked me, like Stephen was saying 30, 40 years ago, if you had asked me 20 years ago, I would have said I'm an electronics and communications engineer, not a professor in medicine or an immunologist. But during my postdoc, I started to learn about immune system. I was in organ transplant lab. And turned out that immune system has had this really privileged status during the evolution, right? This is the only organ system that has exclusive access to all other organs, except brain. But then brain is not mobile. Brain has to stay in one place. Immune system's allowed to go walk up to every single organ, surveil, and look for danger signal. It can also expand and collapse as needed. When there is infection, B cell, T cell, monocytes, they all expand. Infection goes away, they contract. We think about evolution over millions of years or thousands of years, but immune system in each one of us is evolving continuously over our lifetime. It's the fastest rate of evolution. And it has memory. It remembers what it saw since you were born until the day you die. So this is this ability of immune system, this privileged status, basically means that we should be, instead of looking for a pathogen, which immune system is explicitly designed not to let it become systemic, otherwise you get sepsis, why don't we ask immune system, what are you looking at? Because it knows over the evolution, it has learned what is bacteria, what is virus, what is pathogen, what is fungi. It knows it, so why look for a pathogen? Why don't we ask immune system, what are you responding to? And what that means is when a completely new pathogen emerges, immune system can actually tell us what bigger group it belongs to. And then as some of the examples Ephraim showed, we could also do risk stratify, treatment prediction. We should be able to do all of this. But the problem is we need to be able to do this very quickly, 30-minute turnaround time at point of care. So the problem in measuring immune host response, and now I'm not talking about genetic response. Roshni had a fantastic slide of all the factors that affect immune response, and genetic information was one of, I think, 10 or 12 different factors that she had, and most of those were epigenetic risk factors: age, sex, comorbidities, lifestyle, all of those things. And that is reflected in the real world patient population, right? We don't get patients only with genetic mutations. We get patients that have different comorbidities. They are different age. They present at different point of their infection. The lab results are not really clear. And any test that we develop based on host response has to work in this very broad immune variation. The other problem is the clinical context also matters. Some patients will have a community-acquired infection, whereas another group might have a hospital-acquired infection. So when you develop these diagnostics, they actually have to work. If you want to develop one diagnostic that works across different clinical contexts, it really has to have these non-linear decision boundaries. And then the question becomes, how do you do this? Do you do single cohort analysis, multi-cohort analysis? And that's where data that exists in public domain becomes very useful. And really, NIH has done this fantastic job over the last 20, 25 years, right? Of all the data that's funded and generated by NIH becomes available in public repositories. And everything that we've done is using this public data. So no data was generated for the discovery. Everything is publicly available data. And not because, as one of my very dear friends says, I'm cheap, lazy, and parasitic, but because I'm frugal, efficient, and symbiotic, right? It's a point of view. But the real point is these individual studies where there is inclusion-exclusion criteria to make studies more or less homogeneous, collectively, they represent the real-world patient population heterogeneity. They come from different countries, different genetic background, different age, different comorbidities, different technologies, different stages of disease, different severities. Think of a traditional confounding factor, and it's present across different studies. If you can somehow combine this into single analysis, then we are off to races, because now we would have very generalizable diagnostics. And that's what, as a computational person, we've developed a lot of methods to actually leverage this heterogeneity, not as a curse, but a blessing. That data already exists. We can directly go and start using it. So we've basically, over the last 10 years or so, our overall analysis is about 25,000 samples, more than 100 independent cohorts coming from all 6 inhabited continents, more than 30 countries, all of that going into a single analysis to say that there is a set of 29 genes where a different set of genes do different things, but they can diagnose presence, type, and severity of infection. All of those signatures combined using machine learning AI into single classifier that can now, when a patient presents with an acute febrile illness, we can diagnose whether they have infection or not, what kind of infection, and what is the severity. But there is another problem, and that problem, 10 years ago, was every time we tried to license this, I never thought I would be a co-founder of a company, was that somebody would come in, license it, and we can go on and keep writing our grants. And then nobody licensed it. And it took us a while to realize that the problem is we had 29 genes, and there is no platform that could measure 29 genes, high multiplex, in 30-minute turnaround time at point of care. So that's where we decided to say, if nobody else is going to do it, we are going to do it. So we started informatics. And to answer Ephraim's question, sort of jumping off of that, it took us about 7 years to build this device, but now we have a device that has 30-minute turnaround time with less than 2-minute hands-on. So the only manual work required is taking the blood sample in a packaging, putting the packaging into this cartridge, and then putting the cartridge in the machine and following the on-screen prompts. Takes about 30 minutes to measure up to 64 genes. Right now we are measuring 29 genes, 32 genes total in duplicate, including housekeeping, but it's fully automated. And the results are 3 scores. So it's not 1 score, it's 3 score with 5 levels each. It's a probability of viral infection, bacterial infection, and probability of requiring ICU level care in next 7 days. You get 5 bands for each of those, but collectively these 3 scores along 2 axes is what a clinician is looking for. Is this infection? If both either viral or bacterial or both scores are high, then you know that it's infection. But what if it's low? Then you know that it's not infection. Maybe it's something else, and you can start thinking about other diagnostics. And then the severity, which basically tells you whether they should go to ICU or you should do additional diagnostics and you could send them home. So overall, 3 scores along these 2 axes allows presence, type, and severity of infection. Does this really work? So this is the registrational trial that is under review by FDA. All of the data is in this manuscript that's currently under review as well, but it's on the research square in public domain. And I'm just going to give you top line result that each of these 3 scores. So first, what the trial is, it's over 1,200 patients across 22 different sites. The inclusion criteria are here. That's basically all ED presentation, either suspected infection and 1 abnormal sign, or a blood culture order placed and 2 vital signs abnormal. And what you see is basically the same result as Ephraim was showing, but you see that when bacterial score is really high, band 4 and 5, the red band, you essentially get specificity more than 90%, meaning when they are excluded from not having bacterial infection, 90% of those would definitely not have bacterial infection. That's band 4 and 5. And sensitivity is, again, 97, 87, 81% in the lower band. In terms of likelihood ratios, that translates to over 5 or less than 0.1 for all of those. Just a few more comparisons compared to, for example, procalcitonin. And this is another thing that Ephraim was showing, how accuracy changes depending on the patient population. So Triverity, the informatics test versus procalcitonin, overall, Triverity head-to-head more accurate. But as you break it down by different patient population, PCT has a higher accuracy in whites compared to Black or African ancestry and all the other races, compared to Triverity continuously maintaining that same minimum accuracy. And that is what, because of large amount of heterogeneous data that was included upfront. Same thing goes for severity, substantially higher sensitivity compared to, for example, lactate. Overall, in terms of what is the value created, what are the advantages if it is implemented in clinical practice using this 1,200 patient trial. So just look at the last column. Reducing antibiotic usage by about 65%, missed bacterial infection picked up about 80% of those. false negative, these are the ones that all the lab tests looks good. Clinicians send the patient home, and we were able to pick up because those patients had band 4 or 5 in terms of severity, that ultimately those patients ended up requiring ICU level care that were actually at presentation were not that severe. And
2
then,
minutes, please.
thank you, I'm almost done.
Great.
And then just one, this is not part of the paper that I showed you, but as Afram was showing that maybe you diagnose, so what should we be doing with these patients? As Afram again mentioned, there is a lot of discussion whether steroid hurt or benefit sepsis patients. So this locked classifier for triviality with the severity band. So the classifier is locked, thresholds are locked, and we simply applied that to first Victus. This is a randomized control trial for head therapy. And we simply a priori decided that band sensitivity specificity is in 95% in terms of requiring ICU level care. If you look in that group versus the other 4 group, what you see, and this is a small study, so sometimes you don't reach p-values, but you see that if you are in band 1 through 4, so less severe, and do not get steroid, nobody dies. Giving them steroid, mortality higher. Versus band 5, there is benefit in those who are really severe. This is one study. Can we reproduce an overall hazard ratio after correcting for everything really significant? So then the same study that Afram was showing, Vanish trial, exactly same threshold, log classifier, same band 5 result, and you see exactly the same result. If you are not severe and you get steroid, we are harming patients. If you are severe, band 5, and you get steroid, those patients benefit. So really we are starting to bring, now we call this triviality because it was present type and severity, but there is a potential that this could also start giving us information about who should be getting treated and who might benefit, who might be harmed with the same thing. So it could be quadvarity instead of triviality. I'll end by summarizing what we started out about 10 years ago, thinking that immune system can do it. And now we are expecting FDA clearance sometime next month for triviality that can now detect any any infection. We can re-stratify patient, make treatment prediction, and you can get answers in 30-minute turnaround time with a single blood draw. This has taken a lot of people. This is not the list of all the people in the lab who do all the work and allow me to say royal we did this. People at Inflammatics, this has been really wouldn't have happened without all of these people really spending all this time. Thank you.
All right, we are at the finishing line here. So the very last presentation will be given by Morgan Similik from the NIAID. And we're talking about bridging the gap, addressing obstacles in genetic counseling for immune disorders. Morgan?
Okay, great. Thanks so much. We've made it to the end of the day. Congratulations, everyone. So Terry invited me to talk about barriers to genetic counseling. I'm the lead genetic counselor at NIAID and co-lead our sequencing program along with Magda Wakowitz. And I'm going to do that. But looking at the agenda, it also looked like there would be an opportunity to talk some more about the experiences on our group, as well as incorporate some of our patient-reported outcome data and other aspects of patient voice in the conversation today. So let's see, forward. So I thought I'd do that and then wrap up with what I see as gaps and opportunities. So to get everybody on the same page right off the bat, genetic counseling is a process of helping people make meaning of genetic information and then use that information in a way that's consistent with their values, needs, and wants. And when it comes to barriers to genetic counseling, that's largely coupled, and especially in the immunology space, to barriers to genetic testing. And I'm showing here a high-level graphic summarizing that these barriers exist on multiple levels for providers, for patients, baked into the healthcare system. And we've heard from many people throughout the course of today that there's several different clinical pathways to genetic testing. Sometimes this happens in the NICU PICU. Sometimes this happens through a multidisciplinary care team. Sometimes this happens through the genetics department or through an individual immunologist. And you can appreciate how, depending on what the pathway is, there's a different mix of barriers that are at play. One of the other things that I want to point out here is that there are likely also some special barriers related to the capacity and comfort in the genetic counseling community with disorders of the immune system. In our professional status survey, there's a handful of genetic counselors, I think several of whom are on our team, who say that immunology is their primary practice area. And then there's a few dozen across a cohort of several thousand genetic counselors that say that they see any immunology patients at all in the course of their practice. And so there's just not a ton of experience. And I think there's some easy wins in the short term to make available more resources than there are at present for the genetic counseling community. One of the last things that I want to add here that I haven't heard in the conversation so far today, but it's really important and it isn't unique to immunology, is that Dusan's group, but others as well, have published on persistent and substantial racial disparities in access to IEI diagnosis. And also that, but also that these minoritized groups are more likely to be hospitalized, minoritized groups with IEI more likely to be hospitalized. And so in our conversation today and tomorrow about implementation of genomics, I think all of us agree that we want to do that in an equitable way and to continue to keep that perspective in our conversation. So this combination of barriers and just shouting out that motivation for any individual case is also in the mix determines who is receiving genetic counseling for immune disorders across the country, and it's not very many people. And at NIAID, we've been able to experiment with exactly how we triage the genetic counseling resources in more rational ways, and I'm going to circle back to that story in a little bit. So to pivot now to talk about our program, which Magda's going to talk about at a greater length tomorrow, I've highlighted in the upper right here our current team of genetic counselors, and this group of people sees patients mostly for immunology-related indications every day of the week, in addition to patients who have other indications. Other indications for testing. And in addition to that clinical care, our genetic counseling group is involved in a variety of activities that I've highlighted on the right side or the orange side of this virtuous cycle that many of you all published several years ago that have to do with understanding how to best improve the quality of clinical care for genomics that we provide to our clinical research participants. So this includes just sort of day in, day out quality improvement strategies, practice innovations, and asking the questions, designing the research, collecting the data to provide an evidence base to inform when we try something new about how we implement, how we return results, how we enroll people, to provide the evidence base about how to do that work better. And this story, of course, starts where it always should, by listening to our patients. So especially when I began practicing in this space and knew very little about immunology, it was really important to me that I listen and that I listen well to our patients about their perspectives about their illness, about their priorities, about genetics and genetic testing. And I did this through a series of systematic reviews, interviews, and surveys. And like all of the cases that people have shared today speak to, and also just clinical practice, and to summarize this work with just the very broadest of brushstrokes, these disorders, of course, have the potential to have substantially negative impacts on our patients' lives, both the quality and the quantity of the years lived for our patients. And to say this in the words of one of our interview participants, she said, "I felt very cheated. I was supposed to go to law school. I was going to do the Peace Corps. I had all of these plans and things didn't work out for me. I've watched my peers moving forward in their careers and starting families, and I felt like my life was supposed to be different. It's been very hard for me to reconcile myself to the fact that this is the way things are for me." And yet, as we all know, most of our patients over time adapt or psychologically adjust to the challenges of their immune disorders. This adjustment or adaptation is complex and ongoing and requires frequent recalibration to the new challenges that emerge as they move through different life stages. Our patients evaluate their quality of life in a multidimensional way that's more than the sum of their symptoms, and they speak to, in interviews and the qualitative work that's been done in this area, to the roles of hope, finding a sense of control and paradoxically relinquishing that control, and the potent positive or negative impacts of the social interactions in their life. To say, in the words of another one of our participants, everything that's happened to me has made me who I am and made me this very determined person. Regardless of anything that goes on, I can do anything I want to do with this level of determination and the amount of fight that I've got. And if you will, it's been in fact these experiences that have given it to me. I've almost died countless times. I've been on gurneys. I've had tubes hooked up everywhere and everything like that. And no amount of stress at work or anything is ever going to put me back in that situation. So now there's nothing that I feel like I can't get past. Speaking to the capacity our patients have to navigate these challenges. Lastly, many studies show us that anxiety, comorbid anxiety and depression are prevalent and consequential for our patients, and they want us to recognize that in them and address that with them. Layer on top of that work on illness experience, we know from our patients that genetic diagnoses matter to them. I've pulled out from some of our surveys and interviews just a couple patient comments to the question, what's been most helpful in learning to live with your illness? And you'll recognize here concepts related to having a name for their condition, having a disease community, having more targeted treatment, the alleviation of guilt that they felt from their disorder. And this qualitative work is in fact further supported by quantitative validated measures of patient-reported outcomes stratified by genetic diagnosis status here, where the patients in orange are the ones that have a genetic diagnosis, and by almost every measure have better self-reported outcomes or faring better compared to those without a genetic diagnosis. And here we're capturing their perspectives on things like being able to explain their condition. And that sounds like a simple thing, but I think this type of thing is very meaningful to our patients, to be able to say to a neighbor or to be able to respond on the playground when someone says basically like kind of what's up with your kid, to be able to have a coherent response and then have the sense of competence that that can give parents or individuals about, you know, how they're navigating their illness. Most importantly, though, you know, people with a genetic diagnosis in this disease community have a greater sense of being able to make decisions that change their future, which is really important, and also a better ability to know who's at risk in the family. And it's in fact that familial dimension of these disorders that have always been of great interest to me, and I think are in fact an unsung complexity of our patients. And as we were talking about earlier, particularly with some of the dominant disorders where there's reduced penetrance and marked variable expressivity, this can be really complex for people. As you all have seen, I'm sure in clinic, genetic diagnosis prompts a sort of iterative reconceptualization of family ties and disease risk, of understanding who might be at risk, and also the spectrum of what it means to be affected. Maybe for a patient, they've been really sick their whole life, but now they're looking at their cousin who has type 1 diabetes with new eyes, thinking, is that possibly a manifestation of the same disorder? In the words of one of our patients on this topic, she said, my whole life I wanted to be a stay-at-home mom. And then when I was 21, I got sick and I went on medication, and my doctor said, no, you can never go off this medication, but I also couldn't be pregnant on it. And that was really hard for me. It absolutely broke my heart and continues to break my heart every day. And eventually I kind of had to give up what I thought my life was going to be and figure out a new path forward. So as this patient spoke to this, you know, initiates a process of reconciling their hopes and their lived disease experiences, where there's uncertainty, layered upon uncertainty, right, about their own health, about if they can be the parent that they want to be, about the genotype of a potential child and what the physical manifestations might be of that child, and also the role of future treatment. Undoubtedly, assistive reproductive technologies are a very important part of the story for some of our families, but I wouldn't say that most of our families utilize these technologies when going through the deeply personal process of planning for a family. Let's see. And so I'm going to circle back here to sort of this question about how we've triaged at the at our sequencing program at NIAID at the NIH Clinical Center. So we see around 1,500 participants a year. 2/3 of those patients are self-reported white, non-Hispanic, and the vast majority of them consent to sequencing, and per survey have very little decisional conflict about that. There's some important caveats there, but that's generally the bottom line. Per survey, most correctly identify limitations to genome sequencing, yet err on the side of being overconfident in the technology. And additionally, most receive negative results, which sort of creates an interesting group of people to think about and listen to a little bit more.
2 minutes, Morgan.
Thank you. So just to highlight some of the work that our genetic counseling team has done to think critically about ways that we can mitigate misinterpretation and sort of best use our efforts. We did a series of projects over a number of years to, one, confirm that the patient portal is a nearly useless way to communicate with patients about the results of genetic testing. Nobody sees it, nobody reads it. And then create a educational website and a link to connect to a genetic counselor if you had additional questions, then evaluated this tech and touch approach with a survey over this past summer positively. And so exactly this type of sort of iterative quality improvement, innovation to our practice with real life outcome data with patients who are prospectively going through the process that lets us continually inform the way that we're providing care and triage, the high touch and low touch use of genetic counselors across patients who have either more complex genetic test results, genome results, perhaps a primary finding, secondary finding, and pharmacogenomics that's relevant for their care, like Magda's going to share a case of tomorrow, and then those who do just fine with an email update and aren't identifying themselves as having more need. So to wrap this up, I think there's a number of just very reasonable opportunities in the short term. One of the things that I think, so there's an opportunity, I think, to discuss patient engagement and the role of the advocacy community in this implementation work. And I think they're obviously key stakeholders and have already done some great work in this area that others in this room can speak to more. The next thing that I want to say is that for genetic counselors specifically, I think there's two really easy wins that are within the sort of framework provided by our national society, sort of situated in our national conference. And then there's also a practice resource that doesn't exist for immunology that Natalie Deitch from NHGRI is organizing among those of us who work in this area, which I think are great resources that don't exist today. Telehealth is going to continue to be a key factor for the issue of distribution. And then the last thing that I wanted to say that has felt like a gap to me that we were talking a bit about during the break is not actually having a very a data-based understanding of who is receiving genetic testing across the country and who's the ordering provider. And I think that data is available through some of the big commercial genetic testing companies. I have an impression, I know what happens at our institution, and I have an impression or an understanding about some of the other big academic medical institutions, but I think there's better ways to answer that question to inform our implementation strategies. So that's really all I wanted to say. I'm going to shout out the people on our team, the folks that support our work, and of course, all of the patients and their families. So thank you guys for your attention.
All right. This is open for discussion and your comments and questions. I think, Bob, I see your card up. You want to go ahead?
Hi, thanks. Morgan, I had a question for you. Do you guys offer secondary findings in your genome, and what's your uptake? 95%.
I know you are not that tired. Yes.
Sorry, I didn't catch the answer to that. So yes, you offer them, and yes, there's uptake?
Yeah, that's right. In our program, we offer everyone genome sequencing, and that genome sequencing is actually an all-in. There's not an opt-out to our research program for, you get the whole genome and you get primary finding analysis, and you'll also get secondary finding analysis. So there are other pathways to genetic testing for people who really don't want secondary findings. And for some people, that's the case. I find it more common for the people who end up at the NIH Clinical Center that they're upset that we don't return more types of results.
I think our experience in Vitae for whole exome was 95% of people said they were given the option
Yeah.
and they took it. They wanted it.
Yeah, thank you. Thank you for that. So my kind of a question, a comment. One is I live in a rural state without much support for genetic counseling. And so I wonder about how available this kind of support is for patients and what you see the major gaps are for making this more widely available. So that's one thing. And then the other thing, the time of sort of getting information and then coming to some sort of acceptance of this is my new reality. Are there strategies that you talk about for how to help somebody come to that point of acceptance? Is it just really so individual that there are no strategies that you can apply generally, or are there?
Thanks. Those are great questions. Yes, I'm also from rural America, so it's a question that's near and dear to my heart. The genetic counselors, like a lot of other specialty care, is absolutely concentrated on the coasts. There's, I think, been some good progress in this area over the last many years, accelerated by the pandemic and telehealth. And there are some companies, including Genome Medical, which employs a whole bunch of genetic counselors and can, provide genetic counseling every day of the week, paired with genetic testing that's available through their company. And so I think it's a reasonable way to close the gap for some of those, for some of that need. But I'm very sympathetic. I know that there's right, and different pathways, right, that there's where I'm from, it's like, well, if you're willing to drive to Omaha, there's a guy that has a 9-month wait list that's willing to see you, who sometimes has a genetic, so it's just like the access issues are really big. So I think that telehealth is probably one of the best approaches. And then, how do you help people adapt to their illness? I think that's a really deeply felt and special question that I appreciate. I very often approach that with, what's been helpful, what's been less helpful so far, what strategies have people used to sort of like come to come to reconcile themselves to the limitations or opportunities that they find with their health. And people very often are eager to talk about this and have a lot of observations and personal insights about what exactly that has been for them. So I find it less that I'm telling them what they need to do and that they're telling me what's been helpful and where they see, I ask people sometimes, how is it different 5 years in the past, and how do you imagine yourself being different 5 years in the future on the inside? And people have a lot to share about what they've learned about themselves and what they've learned about their world and about their community and who's there to help them. And I think people very often, not everyone, I think some people struggle quite mightily with this. And I think, comorbid depression is really an important part of the equation for some of these patients, where it's really hard to find a sense of hope. There's really hard to imagine a future that they feel good about. But very often people have quite a lot to say and quite a lot to draw from, you hear these stories, or even just like in the case pictures, people have been through a lot and learn a lot about themselves in the process.
Do you think a lot of people feel like they're the only one, and that through these, support groups with people who are going through the same thing is probably one of the beneficial ways to help people cope?
Yeah, absolutely. And reviewing some of these patient quotes and preparing for this discussion today, I absolutely saw just so many people saying, for years and years and years, I was the only one I knew that had anything like this. And it was really helpful to talk to someone who knew what I was talking about. And again, talking about the advocacy communities, I think the Jeffrey Modell Foundation, but also the Immune Deficiency Foundation has been a great player, and the patients are very engaged, dedicated, passionate about their stories. I think there's a lot to leverage there.
We have two cards, the other end of the table, but maybe before you start, Carol and Morgan, I just want to say I appreciate the question and the responses. And maybe if we could capture for the report that comes out, in addition to the huge challenges you mentioned in the rural areas, I've recently learned that in urban centers, in the inner cities of our major urban centers, there are deserts for these services as well. We really should be take care of that. Let's start with you and both, and then we'll come back to Ephraim.
So I have a question about the various sort of diagnostic testing strategies and the biomarkers and the predictors. And it sort of gets to the question of how all of that information is going to be documented and stored in the chart, right? So we have this problem in genetics that a lot of the genetic testing reports come back as a readable PDF, but you can't compute on it. And so you can't get structured data. And so there's this push to try to get genetic data into structured form that you can then use for learning purposes and quality improvements. So for the biomarker test, it looks like there's a bewildering array of possible products. All of them have different outputs, different, tiers of 5 bands of predictors and all of that. So is there a vision for how all of that gets structured so that in an EHR system, you could drive decision support off of it?
Yeah, so it's probably a lot simpler than it is with genetic data, just because a lot of these do consolidate all of that information into a single score, but then there's interpretation that comes with it. And presumably as clinicians get used to using a particular test, that it will become a little bit more easy for them to assimilate. It's also unlikely that one particular hospital is going to have 4 different tests for bacterial versus viral discrimination, each with their own numerical ranges and so on. So, you'll have one that your site uses and you get used to that. And so there isn't that complexity. It's not like you'll have one test, but then if you're looking at this condition, it's this, you should be looking at this component. And if you're looking at a different kind of a patient, there's something else that you should be looking at. There's still a fairly these multivariate index assays, there's sort of precedent behind them. So it actually shouldn't be that difficult. And all these instruments are intended to integrate with LIMS. You're not sending this off to a reference lab and then getting your result as a PDF that you then have to figure out how to integrate. So a lot of it is going to be streamlined in that respect.
Thanks. For the gene expression tests, it seems to me that from the data we saw today that we need to have more underrepresented populations and participants as part of the development of those tests and eventually the perfection or the optimization of the models. And when I say that, and I'd love your thoughts on this, I think it needs both individuals that are diverse in terms of self-reported race and ethnicities, because those may be reporting on exposures that could confound the gene expression data, and genetic ancestry, a diversity of genetic ancestry, because those also may directly impact the gene expression. So could you comment on the strategy there?
Yeah, no, absolutely. That's one of the things that we've been talking about for almost 2 decades now. Majority of the diagnostics fail because they don't generalize to broader patient population. That's just single most important reason. The advantage is a lot of data already exists now, specifically for gene expression. If you just go to Gene Expression Omnibus, there are about 5 million samples from different disease, different background. And with the advances in computational sciences, all of that data is now available for analysis. The focus, in my opinion, needs to be on making it more structured, like the clinical data sets are. And then one thing that we need to continue doing, that we've been doing for almost 3 decades now, is as these data are generated, NIH has done a wonderful job and continue to make sure that people put their data in, so that computational people like me can come in and account for this heterogeneity upfront. And it's not just one source, right? We basically need to worry about three sources of heterogeneity: biological, clinical, and technical. Different technologies, gene expression, we've gone from PCR and microarrays to now single cell sequencing. So that's the technical part. Biological, we've been talking about the effect of age, sex, race, ethnicities, socioeconomic status, and then the clinical, all of the comorbidities that we've also been talking about today. And it's going to be very difficult for a single group to make sure that all of the real world heterogeneity is in a single cohort. The best way to do it is for the community to come together and keep putting data out so that 5 years from now we can combine all of that.
I'll add to that if I may. So a lot of I think the strength of the approach that Pravesh has taken is to draw on that heterogeneity of data so that all of those different populations are represented when you do your discovery work. That isn't always available depending on what disease you're looking at. There may just be a sparsity of data or just not well represented. But going beyond that, there's also the importance of validating your test in the populations of interest. And that's absolutely critical. That has to happen. One of the think about an idealized future where you have ready access to a transcriptome. For those who watch the movie Gattaca, right? You get your transcriptome in a matter of minutes from a finger stick or whatever the case is, and then you incorporate an analysis of that data in real time. That would be fantastic, incredibly complex, but that would be ideal. But unfortunately, that's not how our regulatory environment works either, right? You have to identify your biomarkers, lock them down, lock your algorithm down, which then is a barrier to any learning that happens as new information comes in. So I think there also needs to be potentially in the future a change in the way we think about how these multivariate analytes, particularly very high multiplexed or very high complexity analytes or tests, are thought of so that there is a learning component that gets incorporated that can take into account new diseases, new phenotypes, new discoveries, new understandings.
Bob, is that a new question?
Thank you. I had a question for Dr. Khatri. So ultimately, if this Influmics works well and it appears to be actually have clinical utility, in addition to validity, who do you think needs to be convinced to implement it? Is it going to be the hospitalists and the ICU docs, or is it going to be the lab medicine pathologists?
Well, all of them. One thing we are learning is different hospitals have different people that are ultimately making the decision. Our vision is this will be a test in ED, so that when you're trying to triage patients, this could be one of the tests that upfront tells you. And the way we think about this is we think about different diseases leading to sepsis. But in ED, it's people coming in, and then we are saying that which of them actually have infection. So that's where this would ideally be located to start telling people to basically triage.
My usual experience is that pathologists don't like other people doing laboratory tests. I mean, when I was an intern and a resident, I did my own urine analysis on patients when admitted. That's anathema now. They would never allow that to happen.
I'm not laughing at you. I'm laughing with you. Absolutely, that's been our experience as well. And I always tell people, you solve one problem, there will be next problem, and you just have to stick with it. It's as Ephraim was saying earlier, it took us 10 years to get here, another 5 to convince people.
I think, Stephen, you still have a question? And then Terry and Jillian.
Yeah, I'm a little ticked at Terry Manolio because she wouldn't let me present the data that you presented, Ephraim. And that just doesn't seem fair.
You had far more important work to talk about.
She said it was too speculative or something like that. So, nah. It's my job to keep you all amused at this time of the day. All right, so I guess we're realizing that there's a heterogeneity of etiologies behind sepsis. There's this community acquired against a somewhat normal immune system. versus immune deficiencies. I'm thinking that's probably contributing to some of the variability that you're seeing in these biomarker studies, is that the genetic substrate, the host substrate, which you're measuring, is very different in a heterogeneous manner. I'm not sure how to reconcile that, right? I'm not sure how these if we're thinking about an integrated future solution for sepsis, I'd love to hear how you're thinking about that. You know, clearly for targeted therapies for immune deficiencies, we want to pick these things up early and we want to jump on them real hard. So they're going to get quite different therapies when they present, say, with SIRS or whatever. So for actually for both of you, I'd love to hear your thoughts.
Yeah, so sepsis, I would say, has two major components. There's the pathogenic component, the pathogen component, all of which is going to be treated with antimicrobials, and it's a matter of figuring out the right one and narrowing in and using all of the standard microbiological tools that we have at our disposal. The other one, which is the one that so far we have failed to mitigate, is the dysregulated immune response, which exists, I mean, that's in the definition of the sepsis. And the way in which it's dysregulated, that's where all of that heterogeneity comes from. It may very well be that people are immunosuppressed. Although interestingly, patients who we know have immunosuppression for transplant or that sort of a population, they often have better outcomes when they develop sepsis because they're more susceptible, but they also don't have the runaway immune system that others do. And so I don't know that I don't know that we're going to ultimately see success by kind of labeling a patient as being immunocompromised or hyperimmune. I mean, they're all maladaptive in some form or another. And what I'd love to see ultimately is where we start to, the labels are less important, but what are the groupings? What are the abnormal biological pathways? And if we can understand those, then we could potentially identify therapies that target them. Some patients might need an immunostimulating agent. Some might need an immunosuppressive agent or immunomodulatory agent. Maybe in some cases it's the JAK-STAT pathway that needs to be manipulated, or in others it's a different pathway that needs to be modulated. So as we get greater and greater granularity, I think that will open up the opportunities for what we do for these patients. One of the other things that makes it really challenging is these are dynamic. What we know from some of those subtyping studies is patients may be in one category one day, and 24, 48 hours later, they're in a different category. But that trajectory is also meaningful. The patients that start off in a bad category and stay in that bad category do very poorly, much worse than people who start off in a bad category and then improve to a better one. So the trajectory, the kinetics is also informative. So these are not all necessarily inborn and static and constant. I mean, again, they are in certain scenarios, but for most patients with sepsis, these are highly dynamic and changing, which makes it hard.
So only one of us should have been here today. We are like-minded. That would have been mostly my answer as well. And then the other thing is, again, I'm going back to Roshni's slide. It was a second or third slide where genetic background was one of the 11 or 12 different factors that she had. And I think Priya also talked about it earlier today. Correct me, Priya, if it wasn't you, but she mentioned how it was a single strain of COVID, but there was huge variability in the outcome. So we are never going to get this experiment ever again, right? This was the best natural experiment we could have, as a scientist, hope for. And that tells you, and then the other thing that came out of COVID literature is, despite all of this heterogeneity across all of these countries and all the age and comorbidities, the immune response that was triggered in response to COVID was always the biggest magnitude. Everything else was much smaller magnitude compared to the antiviral response. And that speaks to what the immune system has learned over time. That yes, you are going to have all of this heterogeneity, but once there is an insult, there is an infection, that's going to take over. And that's what, at least for Triverity, we've been able to see that you do see a lot of heterogeneity, but ultimately what defines whether you would have severe outcome or not, that's the host response. I would also go back to something that Ephraim was talking about, these endotypes or the different subgroups. We are seeing that if you don't have all the patients with exactly the same immune dysregulation within sepsis, ARDS, burn trauma, all of that, you actually see patients with either only myeloid dysregulation or only lymphoid dysregulation, and you can actually quantify that. And then there are patients who have both myeloid and lymphoid dysregulation. And depending on that, you can start making hypotheses, and we have data on bioRxiv that actually shows that based on the type of immune dysregulation, you can predict what would be the drug that patients would benefit with. And then in longitudinal studies or randomized controlled trials, we can actually show that the patients who benefited from certain treatment, that was because they were treated with the drug that targets the immune dysregulations that they have. So that probably is where the field is going to go in the next 5 years to 10 years.
So I'm going to change the topic. And so Scott, if you were going to speak specifically to this area, you want to go ahead?
Sure, thank you. I think the worst thing that happened to sepsis is the term sepsis. And I think every time I listen to a sepsis biomarker talk, it seems like everyone's just running away from the word sepsis. And then at the end, we come back to it. And so I want to know, like, how these biomarkers that you're measuring are not diagnostic biomarkers. They're measuring specific physiologic defects. And rather than defining sort of AI-generated endotypes that don't actually help anyone describe anyone's physiologic state and time, is there a utility to, using these biomarkers to pull out maybe more like, this looks more like a antiviral response versus a intracellular bacterium versus an extracellular bacterium versus a fungal pathogen versus a tissue damage versus DIC, TMA, all of things are happening at exactly the same time dynamically, and we just call it all sepsis. And then we revert back to algorithms and endotypes, and then we wonder why we can't get anything to work in sepsis. So how are the biomarkers going to help people to understand the physiology as opposed to sort of just bucket algorithm and knee-jerk response?
So you're absolutely right. And again, that's sort of that future state where I think we start to create layers. And you can use the host response to infer what the pathogen is. So, the bacterial versus viral stuff. And we have gene expression signatures to indicate the presence of a fungal infection. So to some extent, that exists in the research space, at least for now, and starting to emerge in the clinical space. But measuring host response and then saying, oh, okay, this is consistent with a bacterial infection. This is consistent with a fungal infection. So that, in theory, is doable. Now, if you could measure an entire transcriptome in a given patient, as opposed to selectively measuring a handful or a couple dozen mRNAs, now you could start to create those layers. That, okay, I see a bacterial response here. You need the models to look to identify those, and it looks like there's this particular pathway is activated and that pathway is activated, or that one is repressed. So as you have more information available to mine, and you can measure it in real, the other thing to realize is that with sepsis, you've got to make decisions in a matter of minutes to hours. You can't send a specimen off and then come back a week later and then decide what you're going to do. So that is an inherently limiting aspect to it. But the other place that we're starting to go away from the term sepsis is to identify treatable traits, right? So Pravesh alluded to sepsis, trauma, burn, ARDS. We're identifying it's still not a very granular level, but these hyperinflammatory, hypoinflammatory, it's not great terminology, but it's painting with a very broad brush. But that actually seems to transcend these different conditions. So it doesn't matter whether or not your critical illness was triggered by burn or pathogen or some other trigger. There's a lot of commonality in that biology, and I think that also opens up a lot of opportunity for therapeutics. And then there's also the identifying a very specific pathway with a particular biomarker. This biomarker identifies endothelial dysfunction And based on high levels of this one biomarker, I know that I need to treat with drug X to target that underlying biological pathway. So it's going to be piecemeal to start off with. And I think that's largely because of limitations in our technology.
And I would just expand on that. I mean, think about breast cancer, right? 5-year survival was abysmal, but then we started with HER2 positive and EGFR positive, and we have 3 mutations, everything else, a triple negative breast cancer. And then even further subtypes in that. And now we have over 90% 5-year survival in breast cancer. So it's not going to happen overnight, but these endotypes, you're starting with one and then incrementally improving that is how it's going to happen.
And speaking to this point too.
Well, and actually, I was just sort of taking the step back to the clinic. So say I get the result from this test, and you know, how do you take the readout from the test to the patient? and I think everybody knows that the risk of sepsis is your patient can die. Well, maybe is that a 1% risk or 5% risk? And you're telling me there's a 10% of risk of sepsis, or maybe it's a 5% risk. How's that going to change my treatment if I actually have a, you know, what geneticists say is a very high burden problem here if it's a potentially lethal disease?
So I would point to some of the data that I presented, right? So there is a risk of somebody dying and not knowing what's going to happen. And one of the best results that we found was we were able to pick up false negatives. So the current clinical practice that is saying all your test looks good, you are not showing any severe symptom, we are sending you home. And in those people, we were able to pick up 77% of the people that shouldn't have been sent home and should have been sent to ICU or at least observed on the floor. That is first, my hypothesis is that's what's going to start convincing clinicians that we are actually saving lives and not missing. There is no diagnostic that's perfect. Even if you look at the FDA data for the errors in emergency department within US, the error rate in emergency in US every year is about 6%, right? So getting to 100% is very unlikely to happen. But being able to pick up these false negatives, start saving those lives is going to start convincing more people and also going to tell us how to improve our diagnostic at point of care.
So basically, you're saying let's use it to include more people, but never to exclude treating people. Is that right?
I think that's one thing. If you ask sepsis and Ephraim and other people can correct me, I'm an electronics and communications engineer, right? So I'm not the phony. I'm the phony doctor, PhD, right? The PhD, PhD. But the threshold for giving somebody antibiotic is very low anyway. Suspicion of sepsis, and its empiric treatment. And what a lot of the clinicians have told us is, who should we not be treating, because we know that we are treating a lot of people that should not be treated with antibiotic.
Right, Terry has been a terrific timekeeper all day. So I'm going to say it before she says it. We have 10 minutes to go, and we may have 2 or more questions and 2 brief responses to the questions so we can get all of you out of here on time. Terry, over to you.
Great, thanks, George. I do have to channel Steve Holland, who is dealing with an outbreak along with Jeannie, but they will be back tomorrow. Steve did make a comment early in our deliberations about the fleets of ships that have been wrecked on the shoals of sepsis research, which is, you know, something to a cautionary tale. But I wanted to change the topic a little bit, and if Roshini is still with us, and very much appreciate your joining us in the midst of an acute illness. You mentioned a lot of concerns about the regulatory environment and how that may change. And we do have a session tomorrow, if you can join us, that Jillian will be leading, that will be looking at that somewhat. But I would ask you, Roshini, do you have suggestions for the kind of research that might be helpful to the regulators to actually make decisions that align with the science?
Yes.
Before you answer, can I tack onto that? Because I was going to ask a very similar question with a slightly different observation as well, which is I also thought your comments were really interesting about the FDA and the way that it may change things for the LDT space. And I thought it was interesting in contrast, actually, to the next two speakers who really spoke about the FDA as somewhere between just a necessary part of the process, I don't know, or necessary reality of life, to even sort of hinting at it as a pathway to access with the FDA. And so that struck me as a conflict. So I'd be interested actually in all three of you sort of weighing in on the FDA and where that stands.
Thank you, Terry and Gillian. I hope you can hear me. And I will be at the session tomorrow. But Terry, you know, currently as we've been preparing for the initiation of the FDA regulations, what I have heard time and again is the FDA is concerned about quality, about clinical utility, and clinical efficacy, if you will. And if you recall, the LDTs had FDA guidelines as well regarding analytical validation, that you had to meet certain analytical parameters in your validation to implement your LDT. And that's partly what added to the cost and the rigor and the time for validating a LDT properly. And the FDA is concerned that clear license holders who are not CAP accredited are taking shortcuts, both in analytical and clinical validations, and that it's not equal across the board, across clinical laboratories. And I think to some extent the FDA is correct, but a lot of their phase 1, 2, and 3 is related to how are you going to deal with complaints? How are you going to correct if there's a mistake? How are you going to register your test? So at least for the first 3 years, it's a lot of quality assistance essentials, as well as a lot of things to do with reporting and ultimately about the clinical utility of your test. The challenge, Terry, I think, is that it's already costs us anywhere from $150,000 to $250,000 to do a proper LDT validation, development, pre-development, development validation before bringing to the clinic. I don't think we, in an academic medical center setting, can afford additional time and money to be invested, and that's where the pushback is coming from our domain. And the other problem is that the FDA considers those of us in the academic medical field to be device manufacturers, and so puts us on the same footing with Ephraim and Purvesh, who have a different approach to this. And I don't think you can lump us all into that same category. And Gillian, I hope to some extent I answered that. If not, I'll try and re-answer your question.
Great. No, that was helpful. I think Ephraim and Pravesh, if you want to comment.
Yeah, I think Roshni's point well taken, but the reason we chose that we need FDA approval, it could have been lab tests, right? One is the scale. If you really want to have an impact on a large scale, FDA approval is required. Hospitals do want to have that FDA approval so that they can administer tests to the patients. Second one, as Roshni was saying, people might say they might be taking shortcuts. And then the third reason, this FDA approval process where we have to do registrational trial, present our data, make sure everything is right, is what Jennifer was alluding to, knowing what is the risk. When we say there is 10% risk, there is data supporting it, and it's not just some made up number. So all of these reasons require that there is, and it cannot be the company that's going to make money that should be certifying these numbers. It should be a third party making this decision.
I don't have a whole lot to add other than, I mean, I'd say that what I've appreciated about the FDA is that they've been, I think, reasonably progressive in terms of how they think about new diagnostics, and they really are trying to find a pathway, recognizing that things are dynamic and changing, but nevertheless, they still obviously have processes that they need to utilize in order to make sure that anything that gets through is essentially meeting the expectations. So, but yes, you know, an LDT route is not feasible for most of these diagnostics, just by how LDTs are defined. It's just, unless you expect your sample to be sent to a single lab like Carius, right, with plasma cell-free DNA sequencing. You send your sample there. They're not trying to put a sequencer in every single lab. Maybe that is actually an ambition of theirs, but you know, there are certain tests that are amenable to an LDT format. But if you want to get your test result back within the time that the patient is still sitting in front of you, it needs to be deployed at the site of care. And if you have a commercial manufacturer of that test, the only pathway is through the FDA.
And I'll add one more thing. That was the Bayesian, all diagnostics are Bayesian, as Ephraim was saying. Ultimately, clinicians, just the sheer number of patients and the amount of work they have, they want some sort of algorithm, an FDA approval where we go through exactly what the thresholds are, how that provides that standardization to make decisions at point of care.
Well, and I might just add, George, because we do have 3 minutes, that it sounds like this may be a role for public-private partnership, or or academic and commercial labs, which you know, there's always sort of this tendency, oh my goodness, if an academic lab is commercializing it, they're only out for profit, and really it may be, you know, getting the adoption and the testing done that can't be done in an academic setting. So something to think about.
All right, we'll yield back 2 minutes to you, Terry.
Great, thank you from the gentleman from NHLBI. So great, we are just about done. I would remind the members of the Genomic Medicine Working Group, you are not done. We will be meeting at 6:30 at 7th State, which is 7701 Woodmont, just up the road. So please be prompt. And tomorrow we will be starting at 8:30, not at 9. So breakfast is at 8, and then we'll start again at 8:30. And any other announcements? Yes, Gail. Oh, I'm so sorry. It's 6. Okay, it's good that I have Gail and Alana here. It is 6 p.m. My sincere apologies. So we'll see you all there at 6. And I think that
Terry, I'll gargle cough medicine for you.
Sorry?
I will gargle cough medicine for you.
Okay, thank you.
And I'll be playing Christmas songs. So good time.
These poor people want to get to their rooms. Yes, Gail. I don't know. So, all right. Thank you all very much.